стартануть стрим.
Лучше на...
Максим, как трансляция на YouTube?
Что?
Как трансляция на YouTube?
Пока не началась.
Пока не началась.
Сейчас, погоди, началась, кажется.
Всем здравствуйте, дорогие друзья. В эфире первый день конференции «Компьютерные методы анализа Cognitome», и мы рады вас приветствовать. Пока мы в тестовом режиме настраиваем звук и трансляцию, если можете, в комментариях напишите, пожалуйста, все ли хорошо слышно и видно. Максим, пожалуйста, напиши в комментарии что-нибудь, я посмотрю, как чат работает.
Запись идет, да.
Отлично, Сергей, здравствуйте. Слышно и видно, это не может не радовать. Так, скоро будем уже подключаться, переключаться на английский язык. Здравствуйте, Полина. Очень-очень вам рад.
Здравствуйте.
К сожалению, OBS у меня не показывает статистику. У нас есть какие-то подключенные зрители? Можешь статистику посмотреть?
Что?
На YouTube можешь на трансляции посмотреть, сколько человек нас смотрит? Если больше одного, то хорошо.
7.
Фантастика.
Я не уверен, что в мобильной версии есть комментарии. Это лучше кому-то с ноутбуком проверить.
Да, Донецк, привет. Очень приятно, что отовсюду нас слушают. Спасибо большое, что подключаетесь. Здравствуйте, Ярослав. Да, спасибо большое за ваши комментарии. Это очень-очень помогает. Dear Professor Fukai, I'm very glad to see and hear you.
Yeah, me too. Thank you for your invitation. Yeah.
Виктор, здравствуйте.
Здравствуйте. Спасибо за приглашение.
Спасибо вам большое, что согласились. Okay, we're about to start very soon, and the head of our laboratory would say well, several words for all the participants before we start the main section.
Извините, но я вас не слышу. Не знаю, как остальные.
Can you hear me?
Now, yes, but please a little bit more louder.
Oh, sure. Yes, of course. My name is Ivan Limonchenko. I am the head of the International Laboratory of Algebraic Topology and its Applications at Faculty of Computer Science, High School of Economics University. On behalf of the organizing committee, it is my happy privilege to start this open ceremony and to open the international conference on computer methods of cognitome analysis. Our laboratory considers this conference as a starting point of a new perspective direction of research of the members of the team of our laboratory, and a very perspective way of science research area at our Faculty of Computer Science, HSE University. We have a number of young participants, young researchers, and it's a big pleasure for me to say hello to them and to welcome also our distinguished professors and members of the organizing committee who are presented here. Actually, when planning this conference, this event, we were thinking about two main goals. Maybe first goal is to gather well-known scientists, researchers in neuroscience and application of mathematical, computer, and physics methods in neuroscience, and to gather all of them in this wonderful event and to give them this opportunity to speak about their current research and make the audience wider, make the number of young researchers who are interested in neuroscience and applications of mathematical, computer, and physics methods in neuroscience wider. And maybe the second aim we were having is that the students of HSE University, in particular our faculty and mathematics faculty, will be interested in this new perspective area of research. They will be involved in research. They will see how to apply the methods of higher mathematics and computer science that they learn at HSE University. So I'm very honored to welcome the experts, well-known experts from all over the world. And it is also my pleasure to see many young participants, many young starting brilliant researchers who are at the early stage of their career. But despite this, they are very nice researchers. Some of them already have their first papers published. And I'm really happy that today they are all gathered together and they have this opportunity, despite all the current issues, problems that we all keep in mind. They have this nice opportunity to be involved in this highest top-level research and to see the well-known, well-established researchers from all over the world and to hear their presentations. So, as I said before, on behalf of the organizing committee, on behalf of the International Laboratory of Algebraic Topology and its Applications, it is my happy privilege to start the international conference on computer methods of cognitome analysis. And today we are going to have only online session. It will be in English. And moreover, tomorrow and the day after tomorrow, we are going to continue this conference. But at that time, this will be offline meetings at HSE University Moscow campus. And moreover, this will be in the Russian language. Well, as I said, it is our first event in neuroscience that we are organizing as laboratory, but I very much believe that this will be a success. And so next year's next, we are planning to organize the next meetings of this fantastic community. And so at that time, of course, this will be only in English, but this time, this is our first event in this series. And so we decided to make English session only today. Well, I think that it's now the time to transfer to our great speakers and to give them the word. So it is my happy privilege. It's my I'm very glad to introduce our first speaker of this conference. It is the research assistant of our laboratory and one of the key organizers, I would say, of this event, Konstantin Sorokin. And he is going to talk about pure mathematics methods of cognitome analysis, pure mathematics of cognitome. Please, Konstantin, your word.
Okay, let's give it a try. Well, first of all, Ivan, can you turn the volume off or insert the headphones on?
Yes, sure.
It distracts me badly. Okay, first of all, I would like to thank everyone who helped us organize this conference, to all our guests. Of course, I can't thank you enough. It's amazing that we managed to come together to work and to discuss new horizons of neuroscience, in particular cognitome. I'm not sure if everyone knows what it is, and I decided to talk about it, but just a little bit, as one of our organizers and, well, a person who inspired all these things would be speaking broadly about his concept of cognitome tomorrow. In general, cognitome is from its origins very highly focused on us, mathematicians, computer scientists, and even physicians. And in general, cognitome is a new concept of representing a mind as a hyper network. Let me share the presentation, as I forgot to do that. Well, probably this will do. Oh no, it won't. Максим, напомни, как? Full screen. Okay. There we go. Okay, I hope it looks okay at the translation too. Yeah, the notion of cognitome started, well, kind of opposed to a connectome. And what that means is... Максим, проверь, если будут... I'm sorry for the organizing issues. Проверь, если будут жалобы в комментариях на плохую трансляцию на YouTube. So it was opposed to Connectome notion, which is in general a network of a brain on its own. And it's well known that some connectomes of, well, for example, C. elegans was developed and studied over and over for many years. But it's not how the neural network of our brain really works. The thing is that the key element of our mind is a functional system, and the theory of functional systems is in general a model that describes the structure of conduct, which was established by Piotr Kuzmich Anokhin. Functional systems were put forward by Anokhin as also as an alternative to the predominant concept of reflexes.
That part of the slides is not visible.
Вот здесь вот слева поправь. Ну, можно по размеру просто даже поправить, если развернешь на секунду. I'm so sorry, we're check.
Я так понимаю, видно вот это вот?
Да, видно вот это вот.
Что сделать?
Просто потаскай ее. Можешь судить, например. Окей? Сделаешь.
Да.
I'm very sorry about that. So, contrary to reflexes, the endpoints of functional systems are not actions themselves, but adaptive results of these actions. In contrast to reflexes, which are based on linear spread of information from receptors to executive organs through the central nervous system, functional systems are self-organizing and nonlinear systems composed of synchronized distributed elements. So the principle of Cognitome takes its origin from functional systems, but Cognitome is a fundamental theory aimed to do much more. It's a theory which is aimed to describe a fundamental theory of a brain. It's aimed to describe a fundamental theory of mind and a bridge between them. So we have a connectome, which is a pure neural network of our brain. But what if we make a network out of functional systems? It is well known that the neurons which are taking part in different functional systems might appear to be the same sometimes. So the intersections of these things are not empty. And moreover, between neurons, we have lots of different synaptic, for example, chemical connections and electrical ones too, which means that it's not a graph at all. It's a hypergraph. That's why a cognitome is a hypernetwork. So graph models, they benefit from simplicity, and we often do some brain modeling using graph models. But as abstract mathematical objects, graphs are quite limited to represented pairwise relationships between entities. But real world and brain, as an example of a real world phenomena, in these systems, they are very rich and they have multi-way relationships involving interruptions among more than two entities, and even among two entities, we have lots of different kinds of relations. Эту штуку. Красный этот.
Что бы я тут не делал, вот это что не меняется, это то, что видно.
Нет, оно меняется просто со временем.
А.
I'm so sorry. So, moving on to hypergraphs, which we are trying to talk about. Hypergraphs are generalizations of graphs in which edges may connect any number of vertices, thereby representing k-way relationships. Additionally, as a finite set systems, hypergraphs have identities related to a number of other mathematical structures important in data science, including finite topologies, simplicial complexes, of course, too. As you can see, I'm showing you an example of a functional system with non-empty intersection, and these things were actually noticed by Soviet scientists back in 1960s and 70s. For example, this was taken from a book of, a paper of Vyacheslav Borisovich Shverkov, a Soviet neuroscientist, where he studied visual cortex of a rabbit reacting to different stimuli. I'll translate everything here. It's feeding stimuli, lighting stimuli, and defeating stimuli. As you can see, we have obviously known, even on those 30 cells, we have non-empty intersections of these subsets. So it appears to be quite hard to describe them numerically. So, and even now, as far as I'm concerned, there are still issues with extending graph theory to hypernetworks and hypergraph analysis on its own. Yeah, speaking of hypernetworks, hypernetwork is a probabilistic graphical model based on hypergraph models and inspired by biological systems. As simple as that. Well, speaking of hypergraph issues and describing them problems, well, for example, extending spectral theory of graph adjacency matrices, which is a basic tool to study graphs, in order of hypergraph, it poses a challenge. So hyper edges may contain, of course, more than two vertices, thereby rendering the usual adjacency matrix insufficient for encoding those relations. Moreover, again, as far as I'm concerned,
Слайды на звук.
так, а если так сделать звук? I'm so sorry for the technical issues.
Не-не-не. Нет, пока. Сейчас.
It has some latency to it.
А?
It has some latency to it.
А аудио input capture работал?
Нам нужен только window capture.
А?
Нам нужно только вот это вот.
Вернулся.
Здесь все настроено. So sorry. So, the nature of intersections and connectivity in hypernetworks is much more subtle than it is even for simplicial complexes. So it's obvious what an intersection of simplicial complexes is as we are used to them, but in case of hypernetworks, it's totally non-obvious. And moreover, there is a fundamental question. What is an intersection of hypersimplices is? And it's called a fundamental question of hypernetwork theory. And it's central to the concept of connectivity in those complex systems. And the way that connected simplices constrain system dynamics also. And well, speaking of hypersimplices and their intersections, whereas it seems reasonable that the vertex list of the intersection should be, at first sight at least, those vertices common in both simplices, it's not clear how the intersection list of vertices should lie in the combination of hyper edges of both origin simplices. The base of the hypersimplex contains many elements of the same level at its top, formed by the description of their relations, and acquires integral properties that make it an element of a network of a higher level. So from that, we can say that a cognitome is a network of a network of a network. And indeed, we should have multi-layer networks as we have different levels of consciousness, for example. It's obvious that the neurons themselves lie below the idea of functional systems, but there is also a layer of cognitome above functional systems. They are called alpha cogs and phi cogs. And Konstantin Vladimirovich, I guess, would talk about it more broadly. So the understanding of consciousness itself, it requires us, I guess, to start thinking of brain not as a connectome, so i.e. as a neural network, but as a neural hypernetwork consisting of those neuronal groups with specific cognitive properties, and we should study how can we construct hypersimplices out of them, and how can we intersect and interact them, of course. As the hippocampal formation is a very active part of a brain, and spatial navigation and episodic memory is very fundamental to understanding the memory and mind properties, we decided to focus our conference on mainly, of course, on those two areas. Of course, we have our great presenters who would go outside that box, and that's also very important. But as we specialize on spatial navigation, we think it's particularly important to understand the mathematical and topological properties of underlying structures in them. And from that, if we understand those simple structures and mechanisms, we would come a bit closer to understanding the structure of mind, consciousness, and specific processes of large-scale integration of cognitive elements of a neural network. So let's speak about a little bit about our studies and our ideas. It's obvious that we are using calcium imaging, while spatial navigation of a mouse in several arenas of different topologies. And we ask ourselves a question: if we can reconstruct the topology from the spatial maps. Of course, there are lots of studies already, pioneering probably by Kurte and Itskov, who studied neural codes, and huge investment in these studies was made by Nachum Ulanovsky and, of course, Yuri Dobagyan, who every year, I think, proposes some new topological theory and application to spatial navigation problem. But let's speak a little bit about the basic principles. So we have signals of neural activities derived from the previous image, video, I mean. So each neuron activates at some point of a space. And of course, we can make some covering of that space. So immediately what comes to mind is a nerve theorem of Alexandrov, which means that we can intersect and open areas of the covering of arena. The covering is obviously made out of these activation areas. So the intersection of two activation areas gives us an one-dimensional simplex, and the more intersections we have, theoretically, the more dimensional simplex we have. So from that, we derive a simplicial complex, and the Alexandrov nerves theorem proposes that it's homotopically equivalent to the nerve of covering, so the simplicial complex is homotopically equivalent to the topology of a space itself. But it's not really fair to reconstruct the topology looking at mice trace. So it's better probably to work with multidimensional data of neural activity. So what do I mean by that? You know, our studies show that in order to study, for example, place cells, it's all about picking the right neurons. And even using some simple techniques like PCA, for example, we can derive some significant results. In order to pick the neurons which we would consider, for example, a place cell, we are using a mixture of spatial and mutual information, which we will provide in our preprint. And it works fairly well. What you see on these pictures is projections of three different mice neural activities, who explored the arenas with three-hole topology. And we picked about, in general, about 30 neurons. And as you can see, at least we have our in the projection from a 30-dimensional space to the plane, we have the neighborhoods of dots saved, of points, I mean. And as you can see, the same is true for two-hole topology, for example. But of course, showing by hand that, well, come on, we have something like a hole there, and the colors are quite similar and smoothly changing there, is not enough. well, of course, mechanisms of persistent homologies come in handy here, and they help a lot, especially in things more complicated than PCA, of course, as PCA it's very smart, but it lacks some important things which we consider important, in particular the graph properties on multidimensional space. That's why we started developing a pipeline to use isomap projection from our multidimensional neural activity data. And as you can see, we were quite successful with reconstructing some of the loops of this space. So as you can see, after the projection, as we wanted it to be, it is a wedge product of S1s, and not all of them are as pronounced as we would like, but as you can see, persistent homologies help us distinct them from each other quite clearly. But you might say, hey, using those isomap things, what is isomap? Let me say a couple words about that. We are making K nearest neighbors analysis on multidimensional data. And after that, as we've made a graph, we are making its projection using multidimensional scaling to the plane. And you would say, well, most likely you've lost a temporal structure when doing a hypergraph, just a graph, sorry, on your 30-dimensional space. And you might have a clue on that, but as you can see, if we pick just a loop on isomap and on isomap projection and use only those points which I picked orange and reconstruct them on the original space, they kind of represent the original loop, which is suspiciously good. On the other hand, of course, we can use Itzkov's idea of time series topology reconstruction. The idea is fairly simple. Mice is not likely to be teleporting in a space. And obviously her mind states, so the conditions of her neural activities, so simply said, the neuron on or off, of different time periods of intersecting the two consequential signals. Let me show you one picture. So let's look at the left one. We are drawing a line, an edge of a simplex if we have two activities of neurons, distinct neurons, coactive at the same time, or in some kind of time, well, range, epsilon range. And as you can see, it is fairly stable as we broaden this epsilon. Of course, we waited until first homologies, which are blue, get stabilized. By the way, though all those studies are made either by me or our colleagues or my students. This study was provided by my second-year student. These all are mine, previous. So we can explore topology using some other different neurons. Of course, as we can use something like TensorFlow and consequently deep LabCut to track them out. Head direction. Okay, we are studying CA1 of a hippocampus. Welcome, head direction cells. Indeed, we can use similar techniques as we used previously, and we can construct the activation areas of, well, in this case, activation angles of head direction cells. This study was provided also by my student Yaroslav. Hello, Yaroslav. I know you can hear me. As you can see, there is another interesting property of head direction cells. They are coactive as the rodent rotates its head. So they react sometimes, and I guess mostly, to the change of state, not to the particular direction. And as we have lots of coactive neurons, it is a good idea to make a simplicial complex out of these things, right? Especially if we have some obstacles in the space. And this study was made by my student Alexander, who managed to reconstruct from the neural activities the kind of topology of the space using only head direction cells, using that method which I proposed for the place cells, of course. And for the two-hole thing, it of course needs more cleaning, but still we have pronounced distinct loops. Another method we are proposing and working on, and by the way, all of them should complement each other. They should not be in argue to each other, and they should complement. So what do we have here? This is also work of another my student Andrei, and this idea is yet to be developed further and further. What we have here, we have several neurons which are considered to be place cells or head direction cells, or in future, I guess, anything else. But in this case, they are spatial navigation related, and we binarize their activities, which mean that if a cell is active, it's equal to 1. If it's not, it's equal to 0. From that, we can consider the neural code as a real code itself, and moreover as a binary code. So we can use it as a binary code for an integer. And as we are using it as an integer, we have some kind of signal after that, as each neural state codes its unique integer number. We can plot a signal which we obtain from this strange thing and make a Fourier analysis, for example, to get to know when the states come in periods. So, for example, when the state, certain state, begins and ends, and moreover, we can extract some loops from that. And how do you think it worked out? Well, it worked out quite badly, as we have lots of different states, which give us lots of, unfortunately, even different loops. Some are, as you can see, low frequency, some are quite high frequency. That's why what comes in handy is topological signal processing. We can make a neural network even learn the most effective phase shift to reconstruct some topology out of that thing. And moreover, we used, of course, using all the neurons, all the collected place cells during the whole experience, which was quite long, it wasn't efficient enough. That's why we decided to take particular loops which mouse traveled around our obstacles and check if we have our neural states looped, and from that, if we can reconstruct a loop in a graph. And it appeared to be that for certain cycles, we have sometimes long, sometimes short sequences of neural states. And even from that, as you can see in these pictures, which were provided by my student Vyacheslav, we can really reconstruct some kind of loops from that. And another essential thing to understand the mechanics of an average level of Cognitome is, of course, combining different methods and showing that they do not they do not argue actually. So what I'm proposing here, my supervisor Anton Eisenberg proposed an idea of a Docker complex, which can be constructed as a combination of time series code, of convex time series code, and our multidimensional data. From that, we can construct a Boolean matrix. And from that, we can kind of construct a simple shell complex, which is called Docker complex. By definition, it's a complex on the vertex set of our in our case, multidimensional data. And its subset is a simplex in a Docker complex, if and only if there exists some subset in our convex time code, such that, well, they both are existent and equal to 1. So in other words, the Docker complex is generated by the rows of this matrix. Originally, also Docker results. Originally Docker proved that the B and transposed B are homotopy equivalent. No. It was proven later. Originally, he proved that homology groups are isomorphic. But later, it was shown that whenever we transpose this thing, surprisingly, it works quite well, reconstructing that. Sorry, it works quite well as a homotopy equivalence of simple shell complexes on their own. So I think I have to make a short conclusion if I have some more time. Oh, I have a lot of time, actually. Okay, we'll have more time for discussions and questions. So the goals I'm trying to propose are the following. Cognitome is quite mysterious thing by the moment, but still there are constructions, in particular hypergraphs and the mathematics of hypergraphs, which can be explored by us mathematicians, and we can try to find those kind of structures in brain. And moreover, using, for example, neuromorphic computing units, we can model them. On the other hand, we have to explore the data using our forget that the things we are exploring are, of course, valuable on their own, but they should be aimed to fit in the, well, I believe, at least, to fit in a hypernetwork structure, and in particular at some level of a hypernetwork structure. So thank you very much for your attention. I'm sorry for some technical issues. I hope that I was a guinea pig to throw at that hard moments. So all the other parts of conference would be as smooth as possible. Okay, dear friends, do you have any questions? It was an honor for me to give you a talk.
Okay, everyone, let's thank our speaker. Any questions, comments, suggestions to our first speaker?
Yeah, hi. Can you hear me?
Yeah.
Yeah, hi. Great talk. I'm curious about the computational complexity of the methods you're using. What sort of roadblocks or computational problems do you come up with? Because when I use some of these techniques, it's an issue. Like it can be exponential, for example, the number of neurons or things like this.
Well, it is.
Exactly. I mean, if you have all the simplicities, it's 2 to the n, right? So, but yeah, please.
Well, the complexity might be n log n. It depends. N squared, and even worse, of course. The thing is, sometimes it's difficult to parallelize the computing, which, well, I think the biggest struggle I'm having. And such things acquire also in mathematics when you use Sage, for example, to compute homologies of some simplicial complex, which we worked on with my supervisor, Anton Eisenberg, for example. You're having like hundreds of thousands vertices in your simplex, and it can use only one core, and there's no obvious way apart from some non-exact ways of computing minors, for example, of the adjacency matrices, to parallelize these things. And unfortunately, we are struggling badly with those things. We are trying. That's why also we are trying to compute those things on loops as well. And of course, it's all about picking the neurons, probably. But we believe we are doing it right as we have repetitive results. So as mouse, for example, navigates around the non-trivial topology, well, we are having some short period of times and, well, at least 20 neurons which were active at that moment of time. We are quite happy about the mechanisms we are seeing. You know, there are also some side things which we are noticing. For example, while an animal explores the space, there are several steps of doing that. So it appears to be that at the first moment it has, during navigating around the very same loop, it has active certain group of neurons. After that, it is a different group of neurons, a little bit intersecting with the first one. And at the very end, the activity, of course, decreases. And the neural group which is active is a little bit similar to the second one, but completely opposite to the one which was noticed by us initially. So it's quite hard to look at the whole experiment at once, because it gets kind of useless, and as you can see, it gives sometimes lots of noise additional. As probably, yes, we're deconstructing the same loop, but several times. And I think there is still a challenge to try to understand how these things, well, how to catch when the neural group is migrating, as mouse memorizes that obstacle, for example. One tool to do that, as I've shown, is to check if our one homologies are stabilized, but it doesn't really show the transition from second to third step. So Tom, if you can ask your question once again, or if you are having another question, you're welcome, of course.
No, it's okay. Yeah, thank you for your answer. Very interesting.
Sorry if I wasn't precise enough.
No, it was fine. Thank you. Yeah.
Hi, I'm Tomoki.
Okay,
Yeah,
please go
Professor
ahead.
Fukai.
Yeah, may I ask? Yeah. Actually, yeah, probably I couldn't follow some part of your talk, but what is the difference between just simple hierarchical graph and your hypergraph? By hierarchical graph, I just mean that it's a kind of networks of networks, maybe with some cluster structure, or is hypergraph you mentioned is different from this notion?
I assume it is, because it has lots of different interconnections between the same. Let me give you an example. In a book which I read about hypergraphs, there was an example. If we consider simplex as a word, for example, let's consider two words, sing and ring. We obviously have an intersection of three elements. But if we use not sing, but singing, so we have ing doubled. But those ing are different. So we can't intersect them on their own, as they are of different nature, although they are looking the same. So the idea of hypergraph is that although things are looking quite similar on the first sight, there is some underlying structure which distincts them additionally. That's what the idea of a hypergraph is. And it's highly asymmetrical in the real world, of course. Correct me if I didn't ask your question.
I don't know. Thank you. Yeah. And just, can I continue to ask?
You're welcome.
Yeah. And one thing is, now there is argument that so if you use, for instance, PCA to analyze population neural data, of after learning, dimensionality is reduced quite a lot. But some people suggested that if the behavior is complex enough, then dimensionality is actually not low and actually high. And can your method tell something about this issue? I mean that whether the dimensionality of learning to be, how do you say, representation of neural representation of learning behavior is high or low? Yeah.
Probably I didn't say that clearly enough, but I didn't say that PCA is a reference result for anything. I show that it kind of saves the relative neighborness of the points after the projection, but still it didn't satisfy us that much. What I wanted to say and what I came to after my researches is that it's not always a good idea to look at the whole data at once. It's better to look at distinct behavioral acts, like an element of Cognitome. So mouse rotated its head and saw something, an obstacle, for example, or something. A mouse traveled around the loop and came back to its origin. Here is some simple behavioral act, which we think we should consider at once. We worked a lot with this high-dimensional data, and we struggled a lot trying to reconstruct some of its properties, but it always was, well, not always, but mostly it was unrepeatable.
Okay. Can you hear me?
Yeah, I can hear, but I think Konstantin.
Yeah, sometimes video stops in my okay, now it works. Okay.
I'm sorry if it stopped.
Oh,
I wanted just to say that I think we have to divide everything to the simplest behavioral tasks and explore not the neurons we picked initially probably, but repick the neurons which were active at this given moment of time. As whatever good we filter out the neural data, for example, if we are working with place cells, of course, we can pick only moments when mouse was navigating around the space. So we have some, well, so-called speed cells involved. And of course, we, by choosing only moving moments of mouse, we are picking the moments when mouse was thinking about, I don't know, tasty cheese or a student walking around and something else. So what I want to say is that it is very hard to pick the neural group correctly for the whole experiment. It's better to divide it to small pieces and even by several ways. As I've said to Tom, you should probably pick different periods of experiment, when mouse was studying, when the functional group was forming, and when it decreased its activity as mouse explored already the space, and even back to the level of simple behavioral tasks. That's how you can, for example, find an intersection of different functional groups and so on. I'm afraid I didn't answer your question quite clearly too, as it happened with Tom. I'm not sure. Sorry, I just
no, thank you. Yeah.
went
Maybe
into the discussion with myself.
Yeah, background knowledge about math is not enough to understand details. But thank you very much.
Yeah, thank you. It was an honor to answer your questions.
More questions, moments, please.
Okay, let's check the YouTube translation. So the question by Alek Kachan, anyone checked the intrinsic dimensionality of neural code? Can anyone give references? Alek, the dimensionality was up to 5, I guess, but I intentionally focus on one homologous, which we can pick from that. That's all. I think that, well, the brain is not effective. So probably we are having couple neurons doing quite similar things. You know, we experimented with place cells, and I had idea that there might be some place loop cells, which means that the area of activation of particular place cell is a loop. And what do you think? It appeared to be that there were several of those things in our experiment. But another interesting thing was that, of course, and it's obvious that we have intersections between the areas of activations of place cells. But sometimes the difference is so subtle that it's almost indistinguishable. And you just notice that one area is headed toward one loop, and the other is headed toward the other hole or loop in the space. But in general, of course, as well, the animal's mind works, of course, as it better memorizes the borders and space closer to loops, and it doesn't like finding itself in open spaces. So it's well, I don't think that it's that much of a sin that I restrict the dimensionality by hand sometimes. I don't think that's too bad. So any more YouTube questions? I guess not. Well, then, dear friends, I want to thank you very much for your kind attention. If you still have YouTube questions, you can send it to chat. If not, I think I'll will make a short break to check if everything technically all right. And we are waiting for the talk of our great next speaker, which is Tom Burns, I guess.
No, I think
Tomoki, of course.
Yeah,
Sorry.
yeah.
Sorry, yeah, Tomoki. We are very excited about your talk. We can test if you can share your presentation, by the way.
Okay. Yeah. Let me check.
Mhm.
Can you see my slide?
Yeah, it works great.
Okay. So should I wait up to about 20 minutes or something? Yeah.
15, I guess. Oh, yeah, 2020, yes. Yeah, let's make a short break,
Okay.
if you don't mind.
Then, yeah, I don't mind. So see you later again.
Thank you very much. Yes, don't worry. We are about to start very soon. The question in chat on YouTube. Don't worry. We'll be right back. Okay. Dear Professor Fukai, can you hear us?
Yeah.
I wanted to say that you are sharing the presentation mode. Probably you would be more comfortable sharing different mode of your presentation, and we would be very happy to hear your speech, of course.
So can you see my slide properly? No?
Not really.
Oh, really? I see. Let me try. Yeah, here in my side, on my side, think of, okay, I'll just remove my second. Let me try again.
I think you
Right.
can switch just your second monitor and the laptop one as
Yeah.
main and secondary. This
Right.
might work.
Is it still, you cannot see my slide?
We can, but it's in presenters mode, so it's
I
half
see.
screen.
Oh, that's not good. Yeah. Wait. Presenter mode. Yeah, how about this?
Awesome.
So it's okay.
Yeah. Let's go. So, dear friends, I'm honored to present you Professor Fukai from Okinawa University, who would give us a talk about emergent individual behavior trails from neural dynamics.
Okay. Thank you for inviting me to the conference. And yeah, so Okinawa OIST, some of you may not know, but this institute is relatively new. And I also moved just 3 years ago from Riken. And we have a lot of nature, interesting birds, and some poisonous, dangerous snake, famous. And yeah, nature is in general very beautiful. And so today I'm going to talk about how we can model neural networks to explain cognitive behavior and based on experimental data. Yeah, and my background is actually physics, and so I'm a little bit afraid whether my presentation is mathematical enough for the audience. But yeah, I think we have plenty of time. So if you have any question, you can just raise a question during my talk. So actually, the word cognitome was quite new to me, and yeah. But anyway, so my lab is exploring so these issues. So the topics, neural dynamics and computation, and mainly we are interested in memory processing by hippocampus and other areas. But we sometimes do something else also. And we are particularly, at least I'm particularly interested in dendritic computation. And because yeah, I believe that neurons in biological system, brain, is much more clever than what we think in artificial neural networks. And yeah, these days, experimental techniques are advanced, and we can, some laboratory can record dendritic activity in behaving animal. And this kind of experiment started to, show very interesting features of dendrite. And for doing these things, we have to know how neurons in the brain encode information. So we also analyze large-scale neural activity data, and by using some machine learning type of algorithm or mathematical methods. And after I moved to OIST, my interest is slowly shifting towards to connect brain to AI. Actually, there are many people who are interested in this, but as you may know, artificial intelligence is rapidly advanced, but still in many aspects, brain can perform more similar property of our brain, which we still do not know, and useful to construct AI. And especially we are interested in spontaneous activity, because as you may know, the brain never stops, always they are active, but AI usually doesn't care about spontaneous activity. And I'm interested in how this particular type of brain activity contribute to memory or inference or even work higher cognitive functions. But today, I'm going to talk about specific modeling study I did mainly in Lichen, and some here. Okay, so before I go into detail on my modeling, I just mention, I want to say, so there are many different, how do you say, levels of modeling. So this is a well-known decision-making, task perceptual decision-making, which Shadring and Newsom initiated. And in front of you, there are fixation point, not you, in front of monkey. And so monkey has to fixate the eye. Then target location appears. Then after that, screen shows some moving dots. And basically the task is to judge which direction, right or left, these moving dots are coherently moving. And if for instance, the monkey judges it is right, then after some delay period, monkey has to make a saccade towards right target. And this task is simple if all dots are moving coherently towards right. But if it is less coherent, the task becomes a little bit difficult. And by changing the task difficulty, these people measured activity of frontal cortex of the monkey. And to model this, there are so many people just kind of use diffusion model, because this is just a binary choice. So monkey has to choose whether right or left. Therefore, you can consider, let's say, diffusion process in 2 dimension, and if, for instance, this is kind of abstract neural activity, and if it reaches to the, let's say, if some neuron reaches to find some threshold of decision, then you may say that the model, our brain decided left. And if other neuron, in this particular case, so other neurons could not reach to the target threshold, therefore the direction encoded by these neurons are selected. Okay, but another level of modeling is, I wonder you can see my pointer. Can you see my pointer?
Yes.
Okay, good. Yeah. So we can really construct recurrent network model, for instance, and give visual stimulus and train this network to properly choose, correctly choose left or right. And I am interested in the latter type of modeling. And yeah, so more bottom-up approach. And there are many of my colleagues are not really interested in this kind of modeling. Yeah, they think so-called top-down approach is more efficient and easy. But there is a benefit of studying biological implementation, I believe. And so I listed the benefits I can think of. One is clinical purposes. So if you want to, for instance, if you know the underlying mechanism, new network mechanism of behavior, then such knowledge might be used for some clinical purposes. On the other hand, the simple diffusion type model cannot do anything about the circuit mechanism. Then by modeling, we may find some coding scheme which is more efficient. For instance, most of the model is based on filing rate. And in many situations, filing rate coding are sufficiently good to explain behavioral observations. However, we still do not know whether, for instance, synchrony of spikes, which I think, previous speaker also talked, is used or give more efficient code for computing. And this kind of question, I think, can be answered only by modeling neural network. And third, maybe reason, maybe neuromorphic engineering. So for this kind of application, actual circuit mechanism will be very useful. And just curiosity. Yeah, as a scientist, I'm curious how brain solve problem. And especially consciousness. I'm old enough, so I have some interest in consciousness. But yeah, anyway, I'm curious about how brain works. And then new ideas about research. Yeah. So I think most of the audience seems to be young. So you do not know what happens in computational neuroscience, let's say after 70s. Actually, I was still high school students, but after the, at that time, computer was progressed, and many people thought there is nothing remain. We cannot learn anything about from the brain. But then later, middle 80s, next wave of research in computational neuroscience started. But I think this kind of fluctuations in interest, research interest, always exist. But in my personal experiences, experimental findings often give some idea that no one thought. For instance, one example I can give you is so-called excitation inhibition balance, which I will show you later a little bit. And before 2000, no textbook mentioned about the balance between excitation and inhibition, but that is nowadays almost common sense among neuroscientists. But these new ideas often comes from experiments. That's why I believe we can learn a lot from brain itself. Okay. But because if we are, at least in my case, I'm not really very interested in very details of biology. because biological system is so complex. So if you start to think some small kind of details about biology, we may get lost in the path of biological details. And that I do not want to. So some balance between biological details and mathematical modeling is necessary. And I think everyone has to find out their own kind of balance. Okay, so today, yeah, I noticed I'm given more than 1 hour. It's a long time. So I decided to prepare 3 topics, and all are related to modeling. And if I do not have time, then maybe I will skip 3rd topics, but let me try. Yeah, I can explain you all. Okay, the first thing is motor cortical processing. Yeah. And actually, this was done a long time ago. When I started, I opened my laboratory in my previous institute. So at that time, and even now, I am interested in how cortical circuits that has 6-layered structure works during behavior. And at that time, there was no optogenetics. So we decided to use so-called multi-unit recording. And that allowed me to record activity of more than many neurons simultaneously. And so this is a silicon probe used for this recording. And so there are 2 sides of tetrode. I don't know whether you are familiar to this technique, but basically here tetrode means there are 4 electrodes that close each other. And basically these 4 electrodes record activity of superposition of activity of many neurons around this tetrode. So you can obtain 4 traces, so-called local field potential by using this. And on top of this, actually, there are spikes of many neurons around the method. It's called spike sorting. We can kind of decompose spikes of multiple neurons into spikes of single neurons by using the information about shapes of each spikes. Okay, so it's a typical clustering problem. And so this silicon probe had 2 tetrode sites. One goes into deep layer, and the other goes into superficial layer. So we can obtain activity of neurons in different layers simultaneously. And the behavior is very simple. So in front of rats, there is a lever. So rats just spontaneously pull and push lever. But the task is, so before rat pull the lever, so rat has to hold this lever more than 1 second at the pull position, then at push position, and then pull. Then if this condition is satisfied, reward is given to rat. Yeah. So it's a self-paced movement. And we also combine another method called juxtacellular, which I will explain you in the next slide. So multi-unit recording allow me to record activity of many neurons, typically around 50, 60, yeah, if the condition is good. However, you cannot identify the cell type. You can guess the cell type, whether, for instance, some spikes belong to pyramidal cells or interneurons. You can guess from the shape of the spike, but you do not know your guess is correct or not. So juxtacellular recording allows you to explicitly identify cell type or even the shape of cell. For instance, this is some kind hole-related pyramidal cell in layer 5B. Actually, what I mean is this neuron is activated when that's holding lever at push position. Okay, and this is another movement-related interneuron in layer 3. So this neuron is activated when that start to pull movement. Okay, so although we can identify only one cell from each rat, but still, so we have many neurons, non-identified neurons, and just single identified neuron. This is the data set we analyzed. Okay, and there are a lot of findings, but I skip. But basically, yeah. So this is multi-unit data, and if spike, so you can judge whether spikes belong to pyramidal cell by the width of the spike. Typically, compared to fast spiking interneuron, the spike width is a little bit broader in excited neurons. And this tendency can be also checked by juxtacellular recording. Okay. And there are lots of findings, but so what we found is actually we expected kind of very layer 2-3 and layer 5 play kind of distinct roles. For instance, if these are the functional cell type, hold related, pull movement or movement, and many things, and we expected these cell types appear in a particular layer, but it was not like that. Actually, both deep layer and superficial layer contains a lot of movement-related neurons. But one difference we found was somehow hold-related neurons appear more often in deep layer. Yeah, but this tendency, yeah, we found this tendency, but other data set that from other group doesn't show this tendency. So this may be a particular in our data set. But anyway, here the unexpected finding was actually just by looking at the activity, functional cell types of each layer, we cannot tell about the difference in function between different layers. So this was a bit unexpected. But anyway, so another finding was kind of, so as I mentioned, excitation and inhibition is actually balanced, always balanced. What I mean is, so let's say this is a data. So this RS, regular spiking, is excited neuron. And so this is a time of pull movement. Then so movement-related activity, excited neuron is activated slightly before the pull movement. Then interesting, fast spiking neurons are also activated simultaneously with excited neuron. And the same tendency appear in juxtacellular recording. And previously, so when I learned neuroscience, the classic explanation about the role of excitatory and inhibited neurons in motor control is when animal starts movement, excited neurons are activated, but inhibited neurons are silent. And when animals had to stop movement, then excited neurons are inhibited by inhibitory neurons. But it was not true. And actually there were around this time, or just before around 2000, 7, 8, there are several reports about the balance between excitation and inhibition. So our finding was consistent with findings from other groups, but this was actually unexpected. And still, I think people are studying functional implication of this excitation inhibition balance in neural code and several other aspects. And now, we wanted to model this, but and for that, we used so-called reservoir computing. And so some of you may know this. So this is a kind of famous paper by Sushiro and Abbott. And so reservoir computing something like this. So you have a recurrent network, and so this recurrent network, neurons are connected randomly. Okay, so it's random network, and it's a synaptic strength scale as K is number of neuron, or size of the network. This, and it means that this is called so-called strongly connected network. If this connection scales 1 over K, then if K goes to infinity, then even if K goes to infinity, each neuron will receive input of order unity. But if scaling is square root K, then without balance by inhibition, so you need this kind of precise balance between inhibition and excitation. Okay, then the information is read out by readout neuron, and this output of readout neuron is fed back to the current network. And typically, you train only this readout connection by using teaching signal. So this is an example. So they trained. So this is a spontaneous activity. Before training, network shows just spontaneous activity, and each neuron shows some random activity. Then during learning, they give some teaching signal, and then you can see that the dimensionality, each neuron started to fire more regularly. Then after learning, even if you remove teaching signal, networks just continue to produce learning behavior. Okay, and they propose so-called force learning that is rate-based. Okay, and we basically use their model. Okay, oops, sorry, why it that? Okay, and actually this is their, but you can introduce some sort of input to reservoir and training arbitrary, for instance, movement by using this network. Here they replicated or produced movement of the body point when the human is running. And so we basically use this kind of modeling. Oops. Yeah, so what we did is, anyway, so what is input? Then remember that, so tetrode, actually, we have, this Chung has multiple electrodes other than tetrode, so actually 8 electrodes, and each electrode can record local field potential of at the different depths of cortex, and the local field potential are considered to have population input to the particular area. So we decided to use local field potential recorded by these electrodes, and but before that we applied so-called independent component analysis, and we found 4 different independent component in the local field potential recorded by 8 electrodes. And the teaching signal was just lever movement. So we know rats, we recorded behavior of rats. So we asked this network to replicate the arm movement of the rat. And then we can use force learning. And basically, this model, very simple model, can replicate a lot various experimental findings. For instance, yeah, here, some particular neuron in the reservoir shows Hall-related activity, meaning, so this dashed line is experiments, and solid line is modeling data. And these neurons are activated when rats are holding lever. And there are some movement-related cells or pre-movement cells, which is gradually activated before the movement onset. So the functional repertoire generated by the model is similar, but how about functional distribution of functional cell types across layers? And so actually all also quantitatively experiments and models show different distribution, but basically we obtained similar distribution. But what we found is, in deep layer, especially deep layer, Hall-related neurons are abundant, but there are not, we didn't replicate that kind of tendency. And so this is the results obtained by using all ICs, but we removed IC1 and IC2 and to see the kind of relative contribution of each IC. And also the distribution or fraction of functional cell types changes, but more in all cases, for instance, if I remove IC2, then there are no pre-movement activity. So each IC seems to have different computation roles, but more or less, the results are consistent between modeling and experiments. Okay. So that simple reservoir model could explain a lot of experimental finding, but there are, of course, some experimental findings that are not captured by that simple model. It is, for instance, this synchronization, phase locking to oscillations. So actually, we found motor cortex shows both gamma oscillation. Actually, there are two types of gamma: slow gamma, which is around 30 Hertz, and fast gamma, which is beyond 60, or even frequencies close to 100 Hertz. And these two types of gamma oscillations are actually nested in theta. So this is a local field potential. So you can see that there are kind of slowly oscillating theta component. So as in hippocampus, motor cortex also show gamma nested, theta nested gamma oscillations. And interestingly, 80% of neurons we found are phase locked to gamma oscillations. Okay, so this is a, for instance, superficial layer, movement related cell. They show clearly phase locked, gamma phase locked activity, and Hall related neurons again, and deep layer also. But there are interesting difference between superficial layer and deep layer. So just look at Hall related activity, these blue ones. So in superficial layer, all cell types are phase locked. However, in deep layer, if you see regular spiking interneuron, which will be pyramidal cells, yeah, some neurons, Hall-related neurons, show activity which is how do you say, anti-phase locking in theta. So these red ones, yeah, these are movement related. And sorry, yeah, these blue ones are Hall related, but you can see two peaks actually, right? Yeah, so this is one peak, another peak appears. On the other hand, movement related cells only show a single peak within single theta cycle. So in deep layer, somehow there are two types of movement hold related neurons, yeah, pyramidal cells. And one easy explanation is in deep layer, there are two types of pyramidal cells and they are mutually inhibiting, right? Then, yeah, as you expect, these two neuron groups cannot be synchronized. And so this simple model is sufficient to explain the experimental finding. However, this model cannot tell the computational implications of oscillatory synchrony. And this feature cannot be captured by our simple reservoir computing model. So therefore, still, yeah, actually, I still do not know why these different cell types exist in deep layers. Yeah, if someone is interested, I'm very happy to provide data. So just think about it. Okay. Anyway, so this was one example. And so the second example is technique is similar, but the different task is different. So we ask decision making. And actually, the organizers specifically showed interested in this paper. So yeah, I'm happy to hear for that. And here, what we did is so we connected neural dynamics to behavioral variance of individual animals. Yeah. So the question was how individual differences appear from the common neural mechanism of decision making. Actually, if you see a lot of paper or most of paper in decision making, animals are overtrained. Therefore, you will see very nice psychometric curve, which is typically sigmoidal function. But if animal is not trained, overtrained, then probably there are a lot of variances in behavior. And we wanted to ask whether that can be addressed by a simple model. Okay. So I will explain our experimental setting. And so basically this is sensory guided decision making. So in front of that, there is two spouts, left spout and right spout. And so some cue is presented. Then actually cue is so two types of cue. One is low tone, high tone, which is 13 kilohertz, and another is low tone. Okay, and we trained rat by using these two types of tones. And animal has to learn if the tone is high, then lick left spout, and if the tone is low, lick right spout. Okay. And then if the answer is correct, the decision is correct, animal is rewarded. Okay. But during the recording, we basically inserted the 5 tones in between 2 familiar tones. And these tones are only used during recording. So rats didn't know what this tone means. So they have to decide which spout they like. And because so novel tones are not used for training, so there is no question about overtraining. So we expected a kind of behavioral variance or individual difference appears. Okay, and activity, we decoded activity from so-called dorsomedial prefrontal cortex, or in other, it is sometimes called secondary motor cortex. Okay. Yeah. Then, yeah, so these are, so we again use the multi-unit recording, and after spike sorting, we obtained, let's say, roughly around 40, 50 neurons per. We did, so we sorted neuron, then for instance, for left choice trial, some sequence, so neurons, some neurons appear, fire in sequence, and during this Q presentation up to the decision point, okay? And in right choice trial, then another sequence appears, okay? And if we, for instance, if we ordered these neurons in the order obtained for left choice, then sequential structure disappear. So this sequential firing is responsible for decision making. And if you use simple PC, then two types of so trajectory responsible for left choice and right choice appears, and they are well discriminated. Okay. And so this is a behavior of animal, and we recorded activity from 8 rats. Well, actually, we recorded from more neurons, more rats, but only 8 rats gives nice data sets. Okay, so you can see. So so actually, this is a low tone and this is a high tone, and these two are familiar, and in between there are novel tones. And some rats show very systematic behavior, so they change the probability of left choice systematically with respect to the tone frequency. However, other rats shows, for instance, this rat likes left right choice very much, but suddenly at high tone, he switches to the left choice. And opposite tendency, this rat shows opposite tendency. So there are a lot of variation. And actually, some rats doesn't show systematic change. So it shows zigzag behavior. And so we call this type of rats, yellow rats, curve as sensitive rats, and blue ones, insensitive rats or biased rats. Okay. And when you see this, probably, I mean, this is quite different from the standard sigmoidal psychometric curve. And you may not think this kind of random behavior can be modeled or captured by simple network. But actually, we found it's not true, and we can. Okay. By the way, yeah, okay, so this is still experiments, and we analyzed whether there is this difference between in that behavior between familiar tone and novel tone, and this is our analysis of response function. So natural expectation is for novel tone is not familiar to the rats, therefore they might needs more time to decide, but actually it was true, but the difference was statistically not significant. So on average, so for novel tone, that takes about 10 milliseconds longer, 13 milliseconds longer than familiar tones, but this is not statistically meaningful. Yeah. So this is, so from this result, we hypothesize, so probably decision-making of animal is just guided by dynamics, neural dynamics, and almost automatic. So in many decision-making, for instance, strategic type of decision-making, many areas are included that has to carefully analyze the value of the, or reward expectation, and it will take a time. But because there is no much difference between familiar and novel, stimulus, we thought this decision making is still at least still just automatic, and probably dynamics in prefrontal cortex are responsible for this decision making. Okay. So what we did is, to get some idea, we studied how separation between left choice and the right choice occur in neural space. And so for that, we used just standard linear discriminant analysis, so-called Fisher's discriminant. So we Fisher discriminant define optimal kind of dimension plane that optimally separate two set of data. Okay, and so this is the results, and so this is a Q-on set, and so after Q-on set, separation starts, and so Q is presented actually 200 milliseconds. However, separation is almost maximized after 100 milliseconds of the Q onset. Therefore, we speculate, we cannot prove, but we speculate that decision of blood made a decision already at this point. Okay. So, and this tendency it doesn't change between novel and familiar tones. Okay. So the decision making is quite quick. So now, what we wanted to model this tendency. So we introduced reservoir. So actually, anatomically, so medial frontal cortex is receiving input from sensory cortex, including auditory cortex. Okay, and then it has projection to motor cortex, and we mimic this. So this is a recurrent network both of excitatory and inhibitory neuron, and some of these neurons receive auditory input. Okay, and then activity of recurrent network is output to read out neurons. And so, but here, because so we cannot use so-called force learning because this is reward-based learning. So we just decided to enhance or strengthen the connection, readout connection, when the rat this model makes correct decision, and weakened this connection when the decision was incorrect. Okay, so simple reward-based learning rule was used. Yeah, this one. Yeah. So this eligibility trace, reward history, and yeah, this is the reward, and this is the history of each activity of each neuron. And we use this simple update rule, okay, and train the network. And actually, this is one example of spy cluster. So when before Q onset, neurons activity of, so these are versions spontaneous activity, okay? Then after Q onset, the some neurons start to increase activity. And actually, this neuron group show activity, higher activity than other neuron group. And then this is a decision period. And decision period, again, neuron becomes activated. And so actually, these neuron group are responsive for left choice or choice of this trial, correct choice of this trial, and other neuron groups are not responsive for that. Actually, so networks just run correct connection between reservoir neurons in the reservoir and readout neurons. And you can replicate sequential activation. So these are rat case. And model also shows choice-dependent sequential activity. Yeah, okay. As in the rats. And these two sequences are nicely separated. Okay. Yeah, this is again a little bit more about sequential activation we found. So so I didn't explain this, but so input to this neuron. So some neuron group are receiving auditory input, and if the frequency changes, so this is the receptive field of input neuron. So for high tone, low tone, yeah, these neurons are activated. For high tone, other neurons are activated. And so if the frequency of tone changes, then group of neurons that are input neurons activated are slightly changing. And of course, they project to neurons in the reservoir. So basically, the reservoir learns a difference, this difference in input, and depending on the input neuron, active input neuron group, they develop different trajectory. Okay, these are what reservoir is doing. Okay, and yeah, actually, if you overtrain, then you can see nice sigmoidal curve. Okay. Then, now the question is whether the simple model, replicates behavioral variance observed in animal. And actually, as you can see, model shows very similar behavior. So this is a sensitive model. So when stimulus or input are changed gradually, then choice behavior also changes systematically. But this blue new model, this model likes left choice more than right choice, just like this rats. Okay. And actually, yeah, we quantified. So we just defined this quality called sensitivity. So we basically fitted this choice curve by nonlinear function. And if choice curve is linear, then we call it sensitivity is high. Yeah, sensitivity S is high. And these kind of insensitive rats show sensitivity of this choice curve is wrong. And actually, these are observed sensitivity of different rats. Okay, and the model also can produce spectrum of sensitivity. By the same Okay. We did it because you may feel because choice curve of each model is so random, there may not be any meaning for this. However, by introducing sensitivity, we can see that actually the model is producing kind of broad range of network model that show different sensitivity. So in that sense, learning it was meaningful. Okay. Now the question is, why this broad spectrum of sensitivity can appear in such simple model? And we thought, because anyway, as I mentioned before, property of trajectories are important. Therefore, we studied sensitivity of trajectory to perturbation. So in other words, we speculated probably robustness of trajectory to noise is different between different rats, showing different behavior. And in that, we couldn't test it. So therefore, we tested it in the model. So this is a trajectory of sensitive model network. And this is a starting point, and this is decision point. And similar trajectory for insensitive model network. Okay. Then at some point, we give a noise, a brief perturbation to this network. Then this is the result. Okay. So sensitive rats. So the effect of perturbation actually was very big. So some cases still the models chose the same response. However, in many cases, this model chose other responses. For instance, if this was the left choice, this is right choice. So perturbation actually gives a very big effect. On the other hand, insensitive rats, so in some case, choice changes. However, most of the case, perturbations that couldn't change the choice of the model. Okay. And so this is quantification. So this is sensitivity of the model. And this is a susceptibility of the trajectory. And there is actually linear relationship between sensitivity of the choice in the choice and susceptibility of neural trajectory. Okay. So what this means? So we reached a very simple interpretation about our results, and it is something like this. So actually, sensitive rats showing this kind of behavior, probably it has very shallow trajectory. So if there is neural trajectory, there is attractor in neural space, and these attractors are shallow, not very deep. It means if perturbation is given at decision point, then actually in the previous simulation, we give perturbation when neural trajectory reaches the decision point. And this noise will significantly influence the development of neural states. Therefore, choice can be easily switched from one to the other. On the other hand, in insensitive rats, these probably trajectory or attractors are very deep. Therefore, just small perturbation cannot change the evolution of the trajectory. And this actually explains behavioral variance. Yeah. So let's say. So as I mentioned, tone frequency difference in Q, auditory Q, actually changes the initial state of the network slightly around this trajectory, right? But because attractor is shallow, small change in initial state, trajectory evolution will be systematically changed by the small change in the initial states. Okay, so probability of choosing this and this will be gradually changed if the initial states are changed. On the other hand, here, because trajectory, if trajectory is steep, such systematic change, gradual change will not occur. That's why these models shows biased behavior. Okay, and actually, this was almost the first, I think, case that where neural activities are related to behavioral variances. So our results were introduced in News and Views in Nature Neuroscience. Yeah. Okay. But anyway, simple model was sufficient to replicate complex behavior. And at that time, many people, including ourselves, asked, then what is that? Is there any difference in circuits that produce systematic decision-making behavior and or sensitive decision-making and insensitive decision-making? And even reviewers asked, but unfortunately, we couldn't answer to that question. But we are fortunate that the reviewer didn't insist on their question. So our paper was accepted. But later, some theorists actually proposed interesting idea. So this is a reservoir. Our model is basically similar to this. Structure is similar. Okay, and only readout connections trained, and we can define actually effective connection. So effective connection in reservoir is, so this J is a random connection in recurrent network, okay? But this readout connection and feedback connection, this is just a vector, right? So this component gives rank 1 matrix. So actually the effective connection for each neuron is sum of original recurrent connection and this rank 1 connection where W is trained. And by studying the property of this effective connection, these people could show why such simple network can have excellent ability in learning. And although we at that time we didn't know, but I see we speak probably if we analyze connection structure, effective structure of effective connection in our network, we may find some differences probably. Maybe in distribution of eigenvalues or whatever. But unfortunately, we couldn't do that. Okay, so summary. So this is about computing with reward-based learning can replicate essential features of decision related neural dynamics in the medial frontal cortex. And a perturbative stability of the trajectory accounts for behavioral variance. Okay, but interestingly, actually, a common learning rule can account for some behavioral variances. So generally, if I see, so if your friend, you have a lot of friends, and each one has different, of course, individual traits. And we think such traits will appear from some differences in hormone balance or differences in neuromodulator or many complex factors affect the personality of human. Of course, this is true. This is obviously true. However, what we showed is, in some cases, just simple interpretation may exist. About behavioral variances. Okay, and also, third topics. This is I did it with rotation students of me of my unit, but I want to leave some time for question, so I will skip this. If you are interested in this, appeared. We published this paper, so just see it. Here we are interested in how synchronization code can emerge from bursting neurons and rate-based learning neuron. Okay. Anyway, so just take-home message is, so simple network models are often successful in explaining complex behavior, as I showed in my talk, but I still interested in complex network in the brain. So because, so why complex but relatively uniform cortical circuit can be flexible in performing diverse cognitive functions remains to be clarified. As you know, of course, cortical circuits are very complex with 6-layered structure. However, the structure doesn't change. The structure itself is uniform across different layers, different areas of cortex. So the similar circuit can handle many different cognitive tasks. And this is, to understand this feature is very, I think, important, not just for brain science, but also for instance, human like artificial intelligence or whatever. Yeah, and I think for that we can still learn from the brain. Yeah, a lot. Okay, thank you very much. Yeah.
Okay, let's thank our speaker. Thank you very much, Professor Fukai, for your interesting talk. I was very intrigued by several things, but before that, I wanted to ask if someone else has questions. Do you hear me?
Yeah, I can hear you. Yeah. Yeah.
Okay, friends, any questions?
Yeah, this is a collaborator. Yeah, I should show this. I always forget to show.
Yeah. Okay. Of course, first of all, I'm interested in saccade slide, which you dropped. But before that, I wanted to ask you the following question. You mentioned that decision-making is governed by a small number of neurons. How do this number relate to the overall number of rich club neurons in the brain? And it kind of feels they might be related, aren't they?
Mm-hmm. Yeah, I think, yeah, I have no immediate answer to your question, but I can say few things. So first from experiments. So some experiments show actually the number of neurons participating in decision making is very small. So if I remember correctly, they suggested something like 10 neurons only. And in another example, so some people actually, so they used calcium imaging data. So they have more than 100 neurons in their data sets. And they trained reservoir network by using their data. So they asked each neuron in reservoir should replicate one of the neurons recorded by themselves. And they asked how much connection, how many connections they have to change to replicate, for instance, these things. And they found actually you need not to significantly rewire connections. You just rewire a small set of new synapses. Rewiring small set of synapses is sufficient to produce, task-related activity. So at least experiments suggest actually important neurons, small number of neurons are important. And as for rich cloud, I can only speculate. Yeah. So here, many models starts uses random network. There is no rich connection, anything. However, it will be interesting, if we can, from right beginning, start from structured network. So instead of random network, we introduce some kind of structure in recurrent network and train it. And this might accelerate learning. And there is a reason to believe that, not believe, but at least this is not really very stupid question, because as you may know, for instance, in the hippocampus, some people suggested there is even before learning, there is a sequential structure, and some or many actually tracer sequences are actually linked with sequences that already existed in spontaneous activity. So it means non-random connection structure is used to encode behavior. I think testing this in simple model will be an interesting question.
Yeah, thank you very much. We have a question from our listener from YouTube. So I'll ask this question, which was actually related to my second one. Yaroslav Korbov, whom you actually probably know, asks you the following. When you mention sensitive and insensitive to certain frequency rats, if you remember, do these different in their genome, or are they all from the same strain?
Sorry, I couldn't do, is it not in chat? Can you repeat the question? So sensitivity and insensitivity. And?
Yeah, do sensitive and insensitive rats were kind of different in their genome?
That's of course interesting question, but unfortunately we do not have any information about the genome of these rats. We didn't do any genomic tests. But one thing, it's not about genome, but I heard from the past, my postdoc, actually you cannot predict. So actually this task was very difficult for rats. So it took on average 3 weeks for each rat to complete. But in the initial 2 weeks, you cannot tell whether this particular rat can learn this task, and even whether this rat is sensitive or insensitive insensitive. So from learning behavior of from initial 2 weeks, you cannot tell. Therefore, I expect there is no significant changes, differences between these rats. Actually, big difference occur only in the 3rd week. Yeah. Sorry, this is answer that I can give him. Yeah.
Okay, thank you very much. And the similar question from myself about that neural population in medial frontal cortex forms the curves, trajectories in response to different stimuli. You mentioned that the path properties are kind of related to the sensitiveness of a mouse, but can you be more precise on how decision making in details forms this trajectory, if you don't mind?
I think what you mean is how decision is made by network. Is that correct?
Yeah.
So, okay.
Because it seems like a very, very, very deep result, and it has more than meets the eye from the beginning of, well, from the first glance.
Yeah. So what we did is actually very simple. So actually training is done only by using these two familiar tones, okay? And let's say if I give this input to the receiver, then recurrent connection produce some kind of sequential activation. And let's say this, so for this, that has to choose left, for instance. Then what reader neurons should do is kind of strengthen input from neurons that are sequentially activated by this particular input. And that is done by just reinforcement learning. If for this input, this particular reader is happen to be activated, reach to threshold activity, then the trial is successful. So we strengthen this connection and weakened connection to opposite other reader neuron from the same set of neurons. And by training this, we can obtain kind of connection between particular sequence responsible for left choice and readout neuron for left choice, and another sequence responsible for right choice and readout responsible for right choice. Then we tested this network by giving both familiar and novel tones and see whether which readout neuron first reaches to the threshold activity. Is it clear?
Oh, well, yes. Thank you very much. Can you return to the dropped slides about saccades?
Yeah. Yeah, of course. Should I show it? But I think it's already time. But
I think oh yeah, we are out of time. But well, as we have break, I think no one minds if you say several words about slides you've dropped.
Okay, then just very briefly. I mean, it's not big. Yeah. Thank you. Anyway, this is a rotation study. So students, OIST has a system of rotation. So one student, physics student, wanted to do something about neural network. And what we did is, so we wanted to use reservoir to try to learn stochastic movement. So it's called Levy flight. And one example is Levy flight is saccadic eye movement. So actually, there are small saccades and big saccades, and amplitude of saccade is not sampled from Gaussian, but it has long tail, and this kind of movement is called Levy flight. And stochastic, so no one teaches stochastic trajectory to reservoir. And so this is one example of training set. And in that, we used reservoir, but we introduced bursting neuron. As you may know, or you may not know, in many motor control, so neurons sometimes produce single isolated spike, but often produces burst of spike, right? And I was interested in whether burst of spike can contribute to motor learning better. Okay, so we trained this network. So this is a two-dimensional saccade. Therefore, we have a two readout neurons, and we basically trained this network by using Levy flights. Okay, and we found, yeah, actually, so in short, if you use bursting neuron, learning becomes much faster. So this is a network with bursting neuron. This is a network with just isolated spikes, neuron with isolated spike. And if you take many trials, after all, they reach to similar level of performance. However, bursting neuron reaches to the reduces error much faster. Okay. And we studied how this difference occurs. And actually, this was, I think, most interesting. Yes. By the way, we used so-called forced learning, but spiking neuron, spike version forced learning produced by the group of Clopas. But it is, the learning rule is basically rate-based learning. Yeah, you just count number of spike within each burst as a firing rate. So it's not so different from the standard force learning. And here, what we did is we counted the number of bursts, around the onset of this saccadic movement, big jump during saccadic movement. And this before learning. So 0 is the time difference between burst onset and this movement onset is 0. And before learning, there is not much. There is some peak, but not much. However, after learning, so many bursting neuron shows synchrony to the onset of this large change, large jumps. And this is actually very intuitively easy to understand because you have to generate big movements. Then you need more spikes. So that you recruit more bursting neurons around this time. Okay. And but although this results are very simple, but I like these results because so this predict. Actually, the experiment shows that the burst occurs around the onset of burst saccadic eye movement. That is one thing. And another thing is I have been wondering whether we can use synchrony for some computation. And this is one example. So our results suggest synchronization burst across neurons can be used for some type of motor learning. And actually, this motor learning is possible by using rate-based learning rule. So if I combine bursting neurons and rate-based learning rule, forced learning, it is sufficient to use synchrony for computation. Yeah. And yeah, this I think is most interesting result, at least to me. Yeah. Okay, this is what I showed. Yeah.
Very beautiful result indeed. Thank you very much, Professor Fukai. I think we could thank our dear lecturer once again. Yeah, Alexei and have raised their hands. I think if you, it's a clapping. Okay, I see. Okay, let's check if Tom is here. Yay! Tom is here.
Yeah. Hi.
Tom, can we check your presentation if
Yeah, sure.
it's gonna be in right mode?
Yeah. Let's check that. Let me just this button. Okay. Share. Click this button. This one. Can you see?
Looking great. Yes.
Okay. So yeah, just let me know. Maybe I can just keep it in presenter view.
We'll start within 5 minutes. Okay.
Sure.
Okay, dear everyone, I am happy to announce that now we're having a lecture of Tom Burns. He's also a PhD at Okinawa Institute, and Tom is a person who has very wide interests, from philosophy to spiking neural networks and even applications of algebraic topology and geometry in neuroscience. And for everyone, please check Tom's course on that. Very nice lecture course. Recommend. Tom, you're welcome.
Yeah, thank you for the kind introduction, and yeah, thank you for this wonderful opportunity to present. I think it's actually quite a unique opportunity because there's not only quite a lot of high math people, but also computer scientists and neuroscientists and physicists and all sorts of people. So I'm very excited to be in the mix. And because of this unique opportunity, this unique forum, I actually decided to present possibly my most mathematical work. If there's time at the end, we can look to some other things, which include some flavors of topology and geometry. And they are essential components, but I would say that this is much more, at least to me, interesting, because in a lot of work that incorporates, say, algebraic geometry or topology in neuroscience or machine learning, I would say it's, that there are a few of the regular characters that come. And here, I think we're breaching into some new territory, which is quite exciting. So this is all joint work with Robert Tang, who's now in China, and I'll be talking about multi-agent navigation and a social place cell hypothesis. That should be place cell hypothesis. So first of all, do you think, I should change back to the pointer. Do you think that an autonomous vehicle could navigate this intersection? I personally doubt it. And the reason is that the people navigating here are abiding by particular norms which probably aren't in the training set for an AI. Also, too, they have an intuition. They have lots of physical data and experience that would be very difficult for an AI or any sort of artificial system to learn, I suspect, although no doubt it's possible. Here is a beautiful example of birds, a flock of birds, doing something called a murmuration. This is a beautiful thing to watch, and we could sit here and watch it all day. But how does it happen? Of course, similar sorts of dynamics can happen in swarms or indeed in schools of fish and that sort of thing, mostly governed by very simple local laws. But cognitively, how can we explain with the same sort of system the sort of thing that you just saw in a complicated urban environment and this in a natural environment? Okay. So, this work is all already out in a preprint and has also been partly published or talked about at a workshop. So I'm going to be talking today about grid worlds. Grid worlds are these flat two-dimensional arrays of cells made of squares, and each cell or square can be occupied or not. In this case, we've got a 3x3 grid world, and this cell here is occupied by a koala, an agent, and this one here is occupied by a beach ball, an object, and the rest of these are empty. Now you might say, Tom, why did you go from very complicated navigation and urban environments and natural environments to such a simple system? Well, because it's so simple, we can scale it very nicely. It's easy to test new algorithms, natural and artificial. We can also make it more complicated. We can add rewards and punishments and all sorts of things in this discrete environment. We can also, therefore, make it quite similar and applicable to some real world environments like swarm behavior, like emergent behavior, like navigation. And what I'm particularly interested in is that it provides us a formal setting for specifying a whole range of navigation and cognition problems. And in particular, what I'll show you today is an example of a safety problem. Formally, we're going to be using the framework of Abrams, Grice, and Peterson called reconfigurable systems. And it works like this. We have a graph G, and we have a set of labels on that graph. And then we have a set of states, and these are labels of the graph G, on the vertices of G, we're using these labels A. It's a function. And we have a set of generators which locally relabel the states. I'll get to those formally in a moment, but let me just show you an example. This is a state graph, or this is a reconfigurable system in its entirety. Each vertex here, for example, this one here and this one here, is a state of the system. It's a unique labeling of the system of the grid world. In this case, we've got a 1x5 corridor of 1x5 grid world. You can see here the labels here: agent, object, empty, empty, empty. Here, empty, agent, object, empty, empty, and so on. You'll notice too that these are connected by edges with colors. These colored edges refer to these two possible generators, these two possible particular movements or actions which are possible. First, the push or pull is that if you see this subgrid, so if you see agent, object, and then empty, you can swap the labels for empty, agent, and then object. So what is that doing functionally? Well, the koala is pushing the ball, and then here, if we go backwards, the koala is pulling the ball towards it and moving backwards. Here with move, it's just simply that the koala is moving, or the agent is moving across to an empty square. So you can see that if we start on this far left side, we push, 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 pull, pull, pull. And then in the upwards direction, at least the way that I've plotted this, is the koala moving backwards. And you can imagine if we add just an extra square here, this staircase structure will just continue to extend further. So this is a simple example. Let me just clarify and formalize a little bit more of what I mean when I say generator. A generator phi is the following. There's a subgraph, which is support, and this is a subgraph of G. There's a subgraph called the trace. That's a subgraph of the support, but notice that it can also be equal to the support. In this case, our traces are always going to be equal to the support, but that's not necessary. And then we have an unordered pair of local states, U0 and U1, which agree on the support but differ on the trace. In our case, like I said, that the trace is equal to the support, so they differ entirely on the labeling. And we say that this generator is admissible at some state S if we're in one of these two local states. So if this holds, we can apply phi, the generator, at that state to obtain a new state. And you notice that you change the labeling for everything in the trace, but everything else stays the same. Also notice that if you apply phi recursively, then you just get back to the state that you were. This is a more complicated example. Here on the left is the state graph, and this vertex that I'm shading is actually the current state that's plotted on the right. I can't squeeze these 3x3 grids into the single vertices in this picture, so I've done it in this way, in this animated fashion. And this beautiful petal structure isn't purely a coincidence. In fact, if I pause this and rotate, and then draw some circles, ovals around different clumps of states, you'll notice that there's two scales of geometry happening here. These clumps, within them, all the edges are dark red. Those dark red edges represent the move generator. Whereas connecting them, you see orange edges. These orange edges are connecting different clumps of the agent moving around only with the agent pushing or pulling. Another thing you notice, of course, is there's a 3x3 grid here. And what do you know, there's a 3x3 grid here. In fact, these clumps are where the object is static. So I've also rotated this for convenience to show that actually this clump here is where the object is static here. And the rest of this space is where the agent can freely move around while the object is static. And you notice, for example, what must this state here be? Well, that state must be the agent moving down here, because then it would be able to be adjacent to this vertex here, which is a pull action. That edge is a pull action into the center, pulling the object into the center. What must this state be? Well, this state must be, pardon me, the koala moving into the top right, because then it could push the object down into the bottom right corner. So there's already quite a bit of information that we can gain from this perspective of a state graph, but let me go a little bit further, and let me first also just describe a little bit more about these state graphs and reconfigurable systems. So again, these reconfigurable systems on a graph G with a set of labels A consists of a set of generators, which we've seen, together with a set of states which are closed under the action of admissible generators. And the state graph, or the transition graph, which I'll denote like so, is associated to this reconfigurable system and has its vertices as the set of all the states with edges connecting pairs of states by a single generator. Those are edges. This is the state graph. But we can go higher dimensional. Consider state complex. So this is a 2x2 grid world, and here I've got room to plot the states as individual vertices in this picture. And I've got the edges here, those are moves, but then I've added some shading here. These shadings geometrically are squares, even though they don't look like it. That's because they're surrounded by 1, 2, 3, 4 vertices for this left shaded region, and then here 1, 2, 3, 4 vertices for that right shaded region. Let me just explore what does this shading mean here on the left. Well, to do that first, let's follow the 4 cycle. What happens when we go from this state to this one? Well, this koala appears to move up. Okay. Then when we move here, the other koala moves down. Okay. Now let's follow going back the other way. So if we go there, the koala here moved down, and then, okay, this koala moved up. So all that's happening between these 4 states is the koalas are just sort of sliding past each other up and down. And if you follow on the right, they're actually just going horizontally. So in other words, they're not interfering with one another. They're not stepping on each other's toes, so to speak. And therefore we shade in this region to say that they're commuting moves. That is, that they can happen independently, it doesn't matter the order, and they can happen at the same time even. You'll notice though that we don't shade in this center cycle. Well, why is that? Well, let's follow. So we start here, we go down, this koala moved to the right, and then, sorry, to the left, and then this one, okay, the top koala moved to the right, and then the top koala moved down, and the bottom koala moved up. So what happened was there was a half twist. There was a swapping at the locations of the koalas. And if you do this twice, you'll get a full twist. So you've got a half twist for each cycle around this 4 cycle. And therefore it's topologically interesting, and therefore we don't shade it in. You could say that the shaded in regions are topologically trivial or less interesting, at least. So these commuting generators and state complexes are the following. We generate it in the following way. We have a set of admissible generators at some state, and we say that those generators commute when they can all be applied independently of one another, so they sort of don't affect each other, and the resulting states don't depend on what order they're applied in. So therefore the state complex S of a reconfigurable system is constructed by starting with the state graph and then adding in these n cubes whenever you see a set of 2 n states which are related by n admissible commuting generators. One very nice feature that is not immediately obvious, but then quite clear, is that totally different grid worlds can have exactly the state complex. This shaded vertex is the current state of both of these states of different grid worlds, but they share exactly this state complex and exactly this shaded vertex. And I'll give you a moment just to ponder why that might be. And that moment's over. The reason is because you can just clearly invert the labels. If you switch these empty tiles for agents and these agent tiles for empties, you just get exactly this grid world on the left and vice versa. Therefore, topologically, they're exactly the same. Now, these state complexes, you might have noticed, are quite cubical, squarish. Here's a lot of squares, a lot of n cubes. So just what is a cube complex? Well, it's exactly what you've been seeing already. Each face is a point, a line, a square, or an n cube of some elementary cube. And also too, when you have some n cube, you have all the things under it. You have closure, downward closure. And also too, if you have, for example, these two squares, if I draw this edge, if I just highlight this edge here in red, that edge is shared by both squares 1 and 2 in this example. So they both have a common face, and that's how they're glued together. It's a bit like Lego. What's interesting are these state complexes, the way they're defined, are actually non-positively curved. That's what MPC stands for. And this isn't my result. This is due to Abrams, Grice, and Peterson. The state complex of any locally finite reconfigurable system is non-positively curved, is a non-positively curved cube complex. And the significance of this, I'm going to leave the technical details out, but the significance really is that we have very fast local algorithms for calculating geodesics. So consider the situation where you're at this state and you want to get to that state over there. How do you do that in the most efficient way? Well, you just follow the geodesic in the state graph or the state complex. And for example, here, it would be this state moving to this state. And of course, there's an equally shortest path, just slightly different going that way. But these geodesics, you don't have to calculate the whole state complex to know if you're in a non-positively curved cube complex. You can actually just look at local information. Part of the reason is because of the very nice hyperplane structure in these non-positively curved cube complexes. Now, how do you know something is non-positively curved? It's a cube complex. If I give you a cube complex, how do you know? Well, you use Gromov's link condition. And I'm going to give you the intuition and then the technical details. So consider that you have a solid block of cheese and a hollow box. If I cut these corners sort of in a slightly diagonal fashion, then what is their cross section going to look like? Well, the solid block of cheese will have sort of this triangle. If I look at it from the side, that's the cross section that I'll be looking at. It'll be a filled in triangle. Whereas the hollow box, it will be a hollow triangle. And this is essentially what we do when we do Gromov's link condition. We check to see what triangles are filled in and which are not. So more formally, a finite dimensional cube complex is non-positively curved if and only if the link of every vertex denoted like so is a flag simplicial complex. And just a reminder, a flag simplicial complex is one where if you have the boundary of some simplex, you have the simplex that it bounds. So simplicial complexes, if you were unfamiliar, they're analogous to cube complexes, but instead of having squares and n cubes, we just have triangles and tetrahedra and high dimensional analogs. Those high dimensional analogs we call n simplices, n for the dimension. And a flag simplicial complex is basically one that has no missing simplices. So let's do some examples. For example, take this cube complex here, and then let's pick this vertex right in the middle. We're going to calculate its link. The link of this vertex is this simplicial complex. Notice that the vertices of the link are basically the admissible generators, because what we do is we draw a vertex wherever with this sort of small sphere, a small local sphere intersects with different components of the cube complex. And then we draw edges between these vertices when we have squares, and then we would draw a triangle if there's a cube that we're intersecting with, pardon me, basically sets of commuting generators in our case. So in this case, we have a null curvature, so non-positively curved. Also, here's a negative curvature example. Again, looking at this vertex in the center, we can see that there's some negative curvature here. And again, if we construct the link, we don't get any problems. This is a flag simplicial complex. However, if we have positive curvature, like so, then we have the following problem. We have a missing triangle in the middle. And you can see that because you have 1, 2, 3 vertices, which will be the admissible generators, and then you connect them by 1 simplices because they have squares. But because you don't have this opposite corner, and if I just draw that, for example, here, you don't, oops, no, that shouldn't be, ignore this. Because we don't have these and we don't have this. Sorry, my headphones turned off for a moment there. Because we don't have all of these other faces, we don't have this solid cube, we don't have that solid block of cheese, we have a hollow empty box, and therefore we have this hole, this missing simplex. It's not a flag simplex, it's positively curved. So one thing that is problematic, however, in the case of grid worlds and cube complexes or state complexes is the following. Consider a 2x2 grid world where you just have one agent. It's very simple. Maybe the simplest you could think of. Well, simplest would be a single square, but okay, this is pretty simple. The koala can move around in this little 4 cycle, and while it does so, it generates this hole in the middle, right? This is not going to be filled in because, well, you can't move up and right at the same time, right? That doesn't commute. But you know, spiritually, it feels like there shouldn't be a hole here, right? There's this morally speaking, this is problematic. It's saying that there's a non-trivial topological feature here. Everywhere else that we have, it's sort of like an interesting topological feature. Really, when we're dealing with a grid world, we're discretizing a continuous 2D plane. There's nothing really blocking this koala moving from here to here. There's nothing blocking it from moving from here to here or anywhere. Also, too, it doesn't matter about the order, right? If the koala wants to go from the bottom left to the top right, it doesn't matter if it goes up and then right or right and then up. So we say, no, that should be filled in. We add a square and we fill it in, therefore removing this topological quirk. But at the same time, we can no longer guarantee non-positive curvature. We call this dancing with myself. So it's a koala doing a little dance in a 4 cycle. We call it dancing with myself. It's a reference actually to an Australian song from the 80s. My collaborator and I are both Australians. And this dance we denote delta, and it comprises the following data. It's the support is given by that 2x2 subgrid that you saw. The 4 local states are the support, and we have to be in exactly one of those with 3 surrounding floor labels, and we have to have the 4 move generators consisting just like the picture you saw. And we say that this is admissible if we're in one of these 4 local states. And then we introduce the modified state complex. So the modified state complex as prime of a grid world is the cube complex, which you obtain by filling in the state graph as you would normally, but you also commute or moves with dances. And specifically, whenever you get 2 to the L plus 2m states, they'll be related by a commuting set of L generators and m dances. Then we get an n cube, which n is equal to L plus 2m. So what happens to the interesting topology? Well, that stays the same, right? There's no dances here that are going to impinge on anything that was of interest or uninterest before. But we do get situations that can fail. So consider these 2 scenarios. At the top of these grid worlds and the bottom with a particular state, and the bottom are the links for this particular state. So here we've denoted phi 1, 2, 3. Those are different generators, move generators. And then delta 1 here is the dance. And you notice that, well, if you're going to make the link of this state, well, of course, you'll have as vertices these three elements as vertices, and then you'll connect 2 and 3, because 2 and 3 can happen at the same time. They can commute. 1 and 3 can also happen at the same time. And we say that this dance is sort of 1 and 2 happening at the same time. So that's why it connects 1 and 2. But the problem is, of course, that 3 cannot happen at the same time as this dance. That will crash. There will be a collision there. So we don't fill in this triangle and we get positive curvature. In this case, on the right, well, okay, you can see that 3 and 1 and 2 and 3 and all these combinations of moves can happen at the same time. But the interior of this tetrahedron is going to be hollow, because these dances can't happen at the same time. They'll crash on the diagonal. So this is also a case of positive curvature. We prove that these are the only 2 conditions where you can get positive curvature. Also, we prove that if you check just for empty 2 simplices and empty 3 simplices, that you don't have to check anything further. This is all you need to check. Also, I'll just note that rather than constructing the links and doing everything else, you can just check the relative positions of the agents. These agents are separated by a knight's move in chess. These 2 agents are separated by a 2-step bishop move. And we note that here in the proof. If you now follow with me to the world of these modified state complexes, you'll see that there's lots of failure cropping up without even looking at the modified state complex or the state complex. You can see that there will be failure because they are separated by a knight's move. In fact, twice, once this way and once that way, right? And this is the current state that we're in. And these are the commuting moves. So these are sort of red shaded squares are those where there are sort of 2 movements that can happen at the same time. Whereas these blue squares are dances. So there'll be a dance here, there'll be a dance here. And then if they switch positions, then there'll be another dance, another set of 2 dances. So there'll be a total of 4 of these blue squares. And therefore 4 failures actually at the link condition, funnily enough. If we look at other examples, you find even more failure. I'm not going to go through all the failure here, but notice, of course, that there will be a lot. Then we actually did some experiments. So we've got some software that we developed to calculate, in a 3x3 grid world, and you can do it for different sizes, but we just, for expository purposes, did it for 3x3 grid world. And then we varied the number of agents. We counted the number of states which are non-positively curved in those situations after exploring all the possible states. We also calculate the number of dances and the number of commuting moves. And these are the link condition failures as the number of total failures, the mean per vertex, and the maximum number of failures at a given vertex. Because of course you can have a simplicial complex with lots of missing simplices, right? So first of all, the commuting moves are symmetric about 4.5 agents. And you know that already, right? Because of what I showed you before with the case of 3 agents and 6 agents having exactly the same state complex. Why is that? Because you can just invert the labels. What's interesting is that the number of dances also is symmetric about 3.5 agents. And that's curious. We haven't exactly figured out that reason, except to say that probably if you have too many agents, you sort of crowd the dance floor. You can't dance if there are so many people in your way. What's interesting too, the percentage of states which are non-positively curved in any particular cube complex is the worst situation in sort of percentage terms, in a probabilistic sense, is when you have 3 agents. But actually, when you have 4 agents, it's actually a bit worse. Sorry, I should say probabilistically it's worse when you have 4 agents, but the the case of, like that the maximal amount of a failure actually happens when you have 3 agents. Now, what are some applications directly coming from this? Well, first of all, positive curvature can indicate that there are available moves in a state that you cannot safely do at the same time. So automatically, that's something that you can use in, say, operations research, also, say Amazon warehouse robots or whatever you like, train systems, all sorts of things. And our theorem is basically saying you only need a bounded amount of foresight to detect potential collisions. So that's not only useful for machine learning, but also quite appropriate for neuroscience. It shouldn't be the case that I need to look infinitely in space in order to avoid collisions with my conspecifics, my fellow humans, and other things. Also, too, we think that this might help study safety navigation in learning systems more generally, because you can detect collision using purely geometric information here, and you can also use it maybe to provide coordination. And we think that, furthermore, that these sorts of pieces of information, geometric and topological, might actually be useful for learning algorithms and might even be already used by learning algorithms, biological and artificial. Just to give you a flavor of one particular way that we're trying to see if these sort of pieces of information are already in learning algorithms, is that we are training multi-layer perceptron, so an extremely simple artificial neural network, to detect the grid world dynamics. So what we mean is give a state of the grid world, and then ask the network to predict what's the next state. It should be a third in this case, because the only thing that can happen is the koala can move in one of 3 directions, right? So these are the 3 possible states. Now, after doing so, what we can do is we can look at the hidden activity of these neurons. In this case, there are 50 of them. It's a few too many to analyze directly, so we do some dimensionality reduction and put it down into 2 dimensions using PCA. Now, why even do this? Well, geometrically, consider the fact that it's really reasonable that you should find 8 in the center of the hidden space in a multi-layer perceptron that's trained to detect MNIST digits. And let me explain why. So let's do the same thing. So we're putting through different numbers of 8. So when we put a number 8 through after training, we observe that these different activities light up. And here, the 8th neuron will light up as our prediction that this is a number 8 if our system is any good. Now, but we're not interested in the prediction, we're interested in the internal representation of 8 in this hidden layer. Now, if you plot that, you'll find 8 is here in the middle. And I said that was obvious. And that should be obvious because if you think about this combinatorially, for example, like an LCD, 8 is the combination, the union of all possible segments of the digits in a rough sense, right? And then as we move away from the center, we're deleting or taking away different things. So some subset of the union. And it's reasonable that geometrically speaking, that this should be in the center. So we're wondering, is there something like this that we can see in the case of our grid world dynamics? So we just have done some very, very preliminary work in this domain, in this direction. But let me explain just how preliminary, but also still how interesting it can be. Consider this 3x3 grid world with 1 agent only. And if we plot the states after we train the network to predict this extremely simple example, you'll find that there are 9 points where consistently in PCA space, the predictions or internal activities of the predicting network is landing, pardon me. And right now, these are basically lots of points drawn on top of each other, but why don't we consider them as vertices in a graph? And in fact, if we look at this particular state and just call this one the orange state, and this little circle here is the orange state, and I've actually already drawn the edges, that's a mistake, but just go with me here, pretend you don't see these edges. These 3 orange vertices in solid orange, these three here, are the ones that are being predicted next, which we can know already from our network because of its output if we just currently feed that back in. So we're not using some external knowledge. But if we do this for all possible combinations, basically a random walkthrough, sort of recurrently activating our network, we'll find the edges between these vertices. And if it's a graph, we don't have to keep it in this exact geometric situation. We can put these vertices anywhere. And if you do that by first labeling and then redrawing, you'll find that it comes out to be a dual of the grid world, which is also quite reasonable. All of these, the different locations the agent can be, and they're connected to where they can go next. Something that's interesting further is that if you look on the right side here in yellow, all of these vertices clustered on the right, or the high values, positive values of PC1 are on the sides, and then all these on the corners or in the middle. E is a little bit of an anomaly, but if you just ignore the position of E and then look closely at, for example, pairs of vertices here, for example, A and F, you notice that A is above F. That is, in principal component 2, A is larger than F, and also A is above F, in this sort of Cartesian system that I've drawn the graph in. And that's actually true for all pairs here, so D and I and B and C and H and J. Now, I promised something about social place cells. So let me take a huge deviation away from this highly theoretical formal world to social place cells. For those that don't know, social place cells have been found in rats and bats. As far as I know, they might have been also found elsewhere, but those are at least some of the first and most famous. And these are really interesting cells. So you find them in hippocampus. You find them that, for example, in this situation A, this orange mouse observes the blue mouse moving to the right here or to the left. These are different trajectories. And these red dots indicate where, from the perspective of this, we're looking at a neuron activity in this orange mouse. Sorry, I'm just going to have a drink of water. Sorry. We're looking at a neuron in the head of, in the hippocampus of this orange mouse, and it's firing when this blue mouse is moving in this space, right? And so you can see the heat map here. It's a little bit here and a little bit here. But yet, when we look at this same cell, for this orange mouse, it's actually firing down here. So it's somewhere else. Another thing that happens, you can see that in bats, again, we're looking from the perspective of the orange bat, and it's observing this blue bat move around. This cell fires when this blue bat is going here, and yet when the orange bat is flying, its activity is somewhere else. It's quite interesting, and it should kind of give you a hint of where I might be going already. But let me just explain a little bit further. There can be self-dependent and self-independent social place cells. So in the case that you are the orange rat, okay, and you observe the blue rat having some task where it has to go left every time, and you have to go right every time. So these are different cases where you have to go on the same side or the opposite side as this blue rat in order to get some reward. Then actually the representation for the other rat will be different. These are the different representations. And yet, if you see in this case, so this is your own representation, right? So, which is somewhat different like before, but here the representation of blue is changing depending on which side you have to go to. Whereas here, your representation is going to be on the side that you need to go to, but your opposite, sort of your perception of the blue rat is actually consistent, always the same, because it's always going that way. So so this is called independent on the right, and this on the left is self-dependent. And I propose basically that these can form as egocentric vector social place cells as a basis for collision avoidance. What are these situations if not exactly that? They're egocentric in the sense that we care about the relative positions of other agents. They're vector orientated because, well, we care about direction. They're social because, well, we're talking about multi-agent navigation, and they're to do with place. And this exact sort of mathematical feature, topological geometric feature, could be exactly what forms some basis for this computation. And indeed, here's roughly speaking the continuum of these different egocentric vector social boundary cells, they're called, but I propose that there ought to exist something similar for, I'm sorry, my headphones turned off once more. But I propose that so these are for boundaries, basically the boundaries of some box. You can see here on the left, these are hard boundaries. These are sort of like soft boundaries. And these are definitely not boundaries, but they're vector based. And you can look at the paper by Alexander et al. 2020 for more information. There's been subsequent work on this as well. But essentially, the closer you are to some boundary, the more, in this case, and you're heading towards it, the more that these fire. Whereas in these cases, it's less and less. And I propose that basically some continuum like this ought to exist for social place cells as well, in order to help, say, collision avoidance. But you can also think of much more sophisticated navigation that ought to arise from this as well. So the take-home messages are the following. Purely geometric and topological features can capture non-trivial task information. Here I showed collision detection. You could clearly do more. Such features are represented in learning systems. And also social place cells, I think, may form the basis for this sort of sophisticated navigation and cognition, since I actually think of navigation and cognition as not being that different at all. And in such situations, you could use local intrinsic geometric and topological data to really form, like I said, a basis for fairly sophisticated, non-trivial cognition. In the future, we'd like to expand this further, and you're welcome to join us. Basically, the sort of situation with grid worlds is that it's so simple that you can just add more labels, more generators. One question we've had is, should we fill in other cubes or cells? You'll notice, if you remember, in the flower and the petal arrangement example, there's like a big 12 cycle. Those are the petals, and actually those are a very sophisticated dance of the agent moving the object around in a 4 cycle. So that's interesting. Should that be filled in? It wouldn't be a cube complex anymore, but what would it give you if you did fill it in? Also too, you know, how are some of these other properties relating to different aspects of multi-agent grid worlds, for example, congestion, failure braiding, optimization, geodesics? Is the modified state complex with n agents a representation for the n-strand braid group, for example, a classifying space? Also too, you're welcome to do this independently, or I would prefer if you were kind enough to at least chat with me and we could exchange notes and ideas, but you're welcome to do this independently, and you can do so already. You can play with our software. I made this GUI, which you can easily classify a grid world, save it, and there's code for you to instantly create the state complex, calculate the curvature of all the states, explore all the states, and so on in a breadth first search. But warning, there's combinatorial explosion, so you should potentially be wary. Yeah, no koalas were harmed during the production of this research. I thank you for your time, and I'll take questions maybe now, and then if there's further time, I can discuss other things or other projects.
Thank you very much, Tom, for this exciting, fun, and highly conceptual talk.
Thanks.
Okay, I feel like we have some questions.
Sure.
Maybe I can ask several questions or maybe comments. Yeah, I like your talk very much. Yeah, it is kind of, well, it kind of correlates with something that we are doing right now that we've done with Victor Buchstaber.
Okay.
It is actually, I didn't know about this notion of reconfigurable system that you mentioned. But actually we did something similar, and we also okay, so maybe a question. So do you know the notion of Schreier graph?
No. Could you explain it?
Yeah, it's I think you as a mathematician you should know what a Kelly graph is.
Yeah.
Okay, so Kelly graph is that you have a group and you have some generators, and then you connect two elements of a group if they are related with multiplying from the left, for example, by some generator.
Yes.
And Schreier graph is very similar thing, but instead of a group as the vertices of a graph, you take some set on which your group acts. So your group acts on a set, and you choose some generator set for a group, and then you have configurations. Configurations are elements of your set, and some of them are connected with an edge if they are related with generator, with acting the generator. So you just replace a group with some set on which group acts.
Yeah.
And okay, so this is the first thing. And what we did, we just we were not satisfied with graphs, as you told. The graph is kind of not very interesting. We want some higher dimensional cells like attached to this structure. This is what you are doing, and you can like consider some higher dimensional analogs of Schreier graphs or Kelly graphs, and you can build them in a similar manner, but instead of just choosing a set of generators, you choose a set of generators and then some higher dimensional objects. So you choose some partially ordered set of relations, and then you using this structure you can draw some higher dimensional analogs of Schreier graphs or Kelly graphs. And I was just triggered by your pictures with these koalas and balls. So these are, I understand that this is a more restrictive case because this is not like fully like group action on a set, because sometimes some operations are banned in your settings.
Exactly. Yeah.
But I guess that this is kind of further step. So we can like combine our attempts to formalize things like that, and I guess that what we did with Buchstaber. So this was about some graphs with labelings, and you can like relabel something on the graph, and this may be related to your question from some of the last slides, I guess, about this types of higher dimensional cells. Like so not in our setting we got not only cubes but some cells of other types. For example, maybe the most well-known thing from which we started actually is maybe you know the notion of permutahedron. So this is a kind of a polytope. This is a convex thing, but it encodes. So if you have a line graph like simple path graph, and you put labels on this graph like from 1 to n in some order, and then you allow some relabelings, some types of relabelings on this graph, then the structure which encodes your configuration space for this system becomes a sphere. It becomes what is called permutahedron. So you can like if you just allow to relabel neighboring vertices with change their labels, then you will get Kelly graph of the symmetric group with the standard set of generators, and it is a kind of just one graph. But permutahedron is a higher dimensional structure which lives above this. And it has some hexagonal, for example, it has some hexagonal cells, and these are
That's great. That's
Yeah,
great.
these things are very fascinating, and there was also like work on generalization of that in the spirit of what you are doing. Maybe maybe you will be interested in a notion of graphicahedron. So maybe I will write it in chat, and they are doing like yeah, maybe a similar thing. They are putting cells, some higher dimensional cells on, well, calligraph in their case, but you can do this not with calligraphs, but with some other things. So it was very exciting. Yeah, I very liked what you talked, and I think I shall read more papers and maybe books on these reconfigurable systems which you mentioned. So maybe you can advise some sources.
I recommend, if you want, there's a paper by, Abrams, Grice, Peterson. If you just look up this paper, it's our preprint. You will find a citation for the for those. I think it's called Geometry and Topology, or Topology and Geometry of Reconfiguration by Grice and Peterson, which is a fairly good paper, but it should be mentioned also that there was one more author that also worked with Grice, Rob Grice in UPenn, who also contributed to this development. And also just to look at this pretty slide, this is the 12 cycle that I was talking about, by the way. So yeah, this would be interesting. Should we fill that in? If so, what results would be interesting?
Yeah, and also this about this picture. This looks very similar to 15 game, but 15 game like in 15 game you have much more objects, and there is like some very old paper on. Well, I think it is graph theory, but you can like play 15 game on any graph.
Right.
You can guess why, and there is a question on which graph you can reach any state from any other state, for example. And there is a characterization of these graphs. I don't remember exactly the characterization, but it is quite funny, I guess. So maybe it can be solved using some higher dimensional structure. So these are just big thoughts. But
yeah. Thank you.
Yeah, that's very great. Yeah, thank you for your talk.
No worries, thanks.
Dear friends, do we have any more questions? Okay, maybe in YouTube. It's not like not likely, so I wanted to ask a questions on generalizations, but I guess Anton Andreevich just answered it. Yeah.
There are generalizations, top generalizations, I'm sure. And this is wide open now. You can, I don't want to keep this to myself. I hope others work on this if they want. So you've got the tools, and there's also code there to make these pretty animations. And if you prefer, maybe you can replace the koala with I don't know, what's a Russian?
Bear, obviously.
Bear. Okay. So, well, yeah, koala, bear, maybe bear. Yeah. Okay. Yeah.
Okay. Well, if we don't have any further questions, then let's thank Tom once again for his amazing talk.
Thank you. Thanks.
And dear friends, we'll meet within, 2 hours of break. We'll have a very good lunch, I hope. And someone would have a nap.
Bon appetit to everyone.
Yeah. And, well, I hope to see you all and even more after the break. Thank you very much, dear friends.
physically happening, or it's in just Zoom meeting?
Yes, it's physically happens in Moscow at the moment. Well, of course, it's mostly online format,
I see, okay.
obviously. But tomorrow we at least tried to have some offline speakers.
Oh, I see, okay.
Tomorrow I have to say that would be partially
I see.
in Russian
I see.
and partially in
I
English.
see.
So it would be quite challenging, I guess.
Yeah, okay.
Sorry. Okay.
So in Russia, anyone actually working in this topological data analysis?
I actually work on topology data analysis.
Oh, I see.
That's why I decided to invite you.
I
You were
see.
one of the first speakers on my list
I see. I see.
when I decided to make this conference.
So what's your interest? Like, how do you get into this TDA area?
Well, it depends on how far in time I would like to start talking about it. But in short, I've been a physicist,
I see.
firstly acoustics engineer, then plasma physics, even
I see.
fusion physics. And after that, I decided I have to do more pure math. I went
see.
into unstable homotopy theory. So I am algebraic topologist.
I see.
Worked on number theory a bit and algebraic geometry, obviously.
see.
And after that, I met Professor Anokhin, who inspired me to look for some applications of my pure mathematical knowledge and intuitions in neurobiology, and obviously hyper networks, which Cognitome is assumed to be, are very fun to explore mathematically wise. And of course, there is a lot of data you can explore using methods of topological data analysis, like activities of neural groups, for example, many dimensional.
Okay.
Okay, let me check if YouTube live translation is on. Seems like so. Moo, is it 8 o'clock in the morning at your basement?
Yes.
Oh my God. You woke up so early for the science.
Well, it's okay. Well, sometimes I usually get up like 5 a.m. So I think it's okay. I mean, it's give me opportunity to talk to Russian people. Actually, this is first conference I'm actually talking to Russian people. Actually, I had one postdoc from Russia a while ago, but other than that, actually, I don't have any interaction with the Russian people. So it's a great opportunity, I guess.
Yeah, we're also very happy about that. And we hope that it will be productive. I also hope that first three speeches, especially Tom Burns, Tom's would be very interesting for you. You would find them very useful. And of course, my speech was also topologically data analysis oriented. In particular, I'm interested in topological signal processing now applied to neurobiological data, because you can present your data as a sequence of conditions, like the functional system of neurons at every particular morning has some state, and if you represent the state as zeros and one, whether the neuron active or not, you can represent it as a unique integer number, and you can plot a whole signal from that. And you can use some Fourier analysis and topological data analysis to make attractors, for example, and graphs of states.
I see.
Which seems to be very funny when working with spatial navigation, for example. And dear Nachum, I am very, very happy to see you on our conference too.
Okay.
And we've been waiting for your talk too.
Okay.
Yes, so all our speakers are absolutely stunning and amazing. Nachum, can you hear us?
Yes, I can. Hello, everyone.
Yeah, we can see you.
Okay, great.
Are you comfortable starting the presentation within a couple moments?
Yeah, sure.
Thank you very much. Can't wait to hear about your researches once again. It's been a month.
Not much has changed, but okay.
Oh, it's like it's only getting better.
Yeah.
Okay, let's check if your presentation works.
Okay, so let me see just.
And I would ask YouTube participants to write if they can see everything clearly and if they can hear everything. Okay, that beautiful presentation.
Yeah, so it's okay. We can.
I think yes. I can see that the sound and the stream are going. We have picture and we have sound. Okay, dear participants, please welcome Nachum Ulanovsky from Weizmann Institute of Science from Israel. And he's gonna give us his amazing talk about neural codes for natural behaviors in flying bats. As a mathematician and physician in the past, I was stunned by Leonard Jones models you proposed and dozens of place cells activation areas that bats have. That's absolutely unique and life-changing experience. Okay, welcome.
Okay, thank you so much for the invitation and for the introduction. And привет вам всем. Я говорю по-русски, но немножко тяжело мне о науке говорить по-русски, потому что я приехал в Израиль, когда мне было 4 месяца, так что я перейду обратно на английский. So I will talk today about our work on neural codes for natural behaviors in flying bats. So let's see if that yeah, okay. So just before I begin, or as a beginning, I want to make 3 general points, just that we're all on the same page, that my goal, scientific goal, is to understand the neural basis of behavior. So I'm interested in how the brain generates behavior. And of course, you need to focus on something. So we focus on spatial behaviors mostly, and in recent years also on social behaviors. But the interest is more broad in the neural basis of behavior. And I'll try to make the point in this talk that it is crucial to study the brain under more naturalistic behaviors than typically done. So I will tell you several different stories from work in my lab, but you can think of all of these stories as illustrations to the importance of studying natural behaviors, or the power of studying the neural basis of natural behaviors. And okay, we'll go through them one by one, but I just want to start with this will be a key take-home message. And last thing, as you know, and as obvious from the pictures I've put, we study these questions in bats. And it's important for me to emphasize that the motivation is not to understand bats. Bats, of course, are interesting and very unique and animals, and there's a lot of curiosity-driven research here, but of course, the main motivation is to understand the brain. And as I will try to argue, we can use bats to ask questions that are difficult to study in standard animal models like rodents or humans, because they have these interesting behaviors. They move in 2 dimensions, they move fast, so the speed helps us in various aspects. They have this sense of sonar that we can use in interesting ways, as I'll show you. So we try to utilize their interesting behaviors to ask general questions in systems neuroscience. Okay, so this was sort of a general introduction. And so as I said, we are interested in the neural basis of behavior. And I often ask myself, okay, so which behaviors should we study? So suppose you're interested in social behaviors or in group behaviors, so you want to study something like this, perhaps group hunting in lions and communal defense in buffalo are both very interesting group behaviors. But then you might say, well, this is uncontrolled. How am I going to study this? This is too noisy, too variable. So therefore, most people who are interested in group behaviors or social behaviors do something like this. Maybe a tube test in mice or other similar tests. So in a tube test, for example, it's a test for hierarchy. You put two mice in this tube and they're pushing each other, and whoever is winning is considered higher in dominance hierarchy. So you can construct a dominance hierarchy, like you use the ELO rating, like in chess, basically to set a dominance hierarchy. And I think a lot of us have in mind this dichotomy or trade-off between, on one hand, controlled but unnatural experiments in the laboratory, like this one on the left, versus natural but uncontrolled behaviors on the right. And I would like to argue today that it's not exactly like this. It's not a one-dimensional axis, but not so simple, because, for example, the behavior on the right is not entirely uncontrolled, because the lions, for example, are not just, or the lionesses are not just running randomly. They actually have specific strategies or tactic of attack, and they repeat them over and over. So each individual attack has some similarities to the others. So they have a repetition of behavior that is in a way a little bit controlled in the sense that they repeat it over and over. Likewise, what the buffalo do is pretty repeatable. So this is not entirely uncontrolled. It's not noise. And likewise, this on the left is not entirely unnatural, because mice are burrowing animals. They live underground in burrows, at least the wild mice. So for them to encounter another mouse in a tunnel underground is not entirely unnatural. So it's not such a simple separation. In fact, I'd like to argue it's more of a two-dimensional axis, where one axis is artificial versus natural behavior, and another axis is uncontrolled versus controlled. And of course, you can have extreme cases, like, for example, head fake self-stimulation of dopamine centers in the brain that has been used for many decades, is a very controlled behavior. Rats and mice will do lever pressing to deliver, to stimulate their dopamine centers in their brain over and over again. So it's highly controlled, also highly artificial. On the other extreme is 24/7, totally unconstrained behavior of the animal, very natural, but very uncontrolled. And of course, the line connecting the two is basically the axis I've shown you on the previous slide. But then there are behaviors like this one. One of my favorite examples, bird song, at least in zebra finches, is a very natural behavior. They sing behavior naturally, but it's also very controlled in the sense that a male zebra finch will repeat the same song over and over hundreds of times a day with a precision of a few milliseconds, 2 or 3 milliseconds. So it's a highly precise, highly controlled, highly reproducible behavior, but it's also natural. It doesn't require training. It's a natural thing. So, lion hunting is maybe somewhere here. It's also very natural, but maybe a bit less controlled than the bird song. So I would like to argue that there is a sweet spot or a sweet area where we should go, which is behaviors that are relatively natural, but also relatively controlled. And we should try to move away sort of from this diagonal to the upper right corner. These are the behaviors that we should study. And this is a point that I would like to make during the talk today. And in our work on the neural basis of navigation, basically this is where we've tried to go. So this is sort of the general approach of what I'll be telling you about today. And now going to the specifics of what do we know about neural basis of navigation in laboratory environments, which is where it has been studied quite a lot. Of course, I think you know probably most of this. We have several types of ambus and the medial neuronal cortex, place cells, grid cells, and direction cells. So place cells, these are all examples from rodents. If you let a rat run in a 1 by 1 meter box, so this is a top view of the box, The black line is the trajectory of the rat. It's just running around randomly. Why is it running randomly? Because you throw in food pellets randomly. So it's basically a random foraging task. It's not really a navigation task. And then when you measure for one neuron in the hippocampus, John O'Keefe discovered that there are neurons that fire in one particular location. So this neuron fires in a northeast corner of the box. Another neuron may fire in the northwest corner, or in the south, or in the center, etc. So when you look at all these neurons together, they tile the environment and tell where am I in the environment. So we call them place. Then in the medial cortex, there are grid cells. The experiment is the same, random foraging, but the neuron fire is not in one location, but in many locations that create this beautiful hexagonal lattice that is sort of like a distance meter, can be thought of a distance meter. Those are the grid cells that the Moses discovered. And then there are also head direction cells, neurons that don't care so much about where am I in space, but they respond to when the animal's head is in a particular direction, in this case east, or another cell could be north, etc. So you can think of them as place cells are sort of neural maps, grid cells are neural distance meters, and head direction cells are neural compasses. So all the elements that you'd like to build a navigation system in the brain. Now, what we ask in my lab is, how does real-life navigation differ from navigating in small laboratory setups, like the one I just showed you, this 1 by 1 meter box, which is a typical experiment, or some experiments would be maybe a 1 meter linear track or something like that. So you can give several answers. One is that the world is not flat, it is complex and three-dimensional. So how does that affect the system? The world is not 1 meter in size, navigation occurs over kilometers. So how does the scale of navigation affect the system, navigation system in the brain? Often animals navigate in groups, so how do the social aspects or the group aspects affect the system? And lastly, as you navigate, you need to sometimes switch to other things like pay attention, avoid predators, or pay attention to conspecifics. So rapid switches between navigation, other behaviors, how do they affect the system? You can give more answers, but these are things that we have been investigating experimentally in my lab. And this is also the outline of the talk that I will give today. So our study animal is the Egyptian fruit bat. So these are relatively large bats, much bigger than the bats that you know in Russia or in Europe or in the US. They weigh about 150 to 180 grams, so they're pretty big, which is good for us because then we can put all sorts of equipment on them, neural loggers, ultrasonic microphones, etc., etc. So it helps to have a big animal. Also, if you'll just look at their eyes, then you will know that they actually see very well. I'm not sure I'm going to show you any data, but just look at their eyes and you'll believe me that their vision is actually very good. It's much better than rats or mice. But they also have a sense of echolocation where they emit ultrasonic clicks, and from the echoes, they can measure distances and directions to things out there. So they have both vision and echolocation. I'll mention echolocation a little bit later. And so to put you in the mindset of what does it mean to navigate for a bat, this is from a study that we've done already more than a decade ago, where we just track the navigation behavior of bats outdoors. This is purely behavioral study. We put these little GPS data loggers on the bats that record their trajectories. And then we measure trajectories that look like that. This is an example for one animal that flies from its cave. They roost in caves. They live in caves. And this particular animal went to a particular tree, fruit tree. These are fruit bats, so they go to forage in fruit trees. And this animal went to the same tree night after night for about a week. So each line here is a different night, a trajectory from a different night for the same bat. So it's one bat over 7 nights, and you see it forms this nice flyway over, in this case, 15 kilometers, and it can go to the same individual tree night after night, even though there are thousands of other fruit trees in the environment. So these guys can really navigate very precisely. And so let me start with the first point that I made about 3D, the issue that the world is three-dimensional. So 3D spatial codes in the bat brain, 3D place cells, 3D head direction cells, and 3D grid cells. So before I tell you about 3 dimensions, I'll just tell you that in 2 dimensions, we some years ago, we've done studies where we've ratified the bat. We've put bats in boxes just like the rats and let them crawl randomly in search for food. So on purpose, we've done this two-dimensional experiment in bats so we can compare to rats, and we found basically the same thing. We found place cells, grid cells, and directions in bats that were similar to those in rats. So in 2 dimensions, it looks pretty similar to the rodents. But then we went to 3 dimensions. Mikhail Yartsev, when he was a PhD student in my lab, he recorded from place cells in the hippocampus area CA1 using a wireless telemetry system that we developed at the day. And this is an example of one neuron recorded in a big flight room that we have, about 6 by 5 by 3 meters in size. So it's a pretty large room. And what you see here is the firing rate color-coded. So red is high firing rate, blue is low firing rate. And you see this neuron had a nice place field, so a certain region in space where it fired at a high rate. And this region or place field was more or less spherical. And in fact, we found that for most of the neurons, more than 90% of the neurons, the place fields were spherical, meaning that whenever the bat flew through a certain sphere in the middle of the room, the neuron fired. And the fact that the sphere means that there is the same resolution in the X, the Y, and the Z dimension. So this was for place cells. Then a few years later, Arseny Finkelstein did an experiment where he was also a PhD student in the lab. They're both have their own labs now. And Arseny Finkelstein, he asked, are there 3D head direction cells in the bat's brain? And the answer was yes. He recorded in dorsal presubical, and that's the first brain area where head direction cells were discovered in rodents in the 1980s, and basically found 3D head direction cells there, neurons that respond whenever the bat's head points in a particular 3-dimensional direction. So, there or here, something like that. And interestingly, I don't have time to go into that, but Arseny was able to show that the coordinate system in which these 3D head direction cells are operating is not a classical spherical coordinate system, which is the textbook way to represent 3D head directions, but it's a toroidal coordinate system, meaning you have 360 degrees of azimuth by 360 degrees of pitch. The problem with the spherical coordinate system is that you have a discontinuity in the zenith and in the nadir, which is a problem for biological systems. Also, mathematically, it's a problem, but a toroidal coordinate system does not have a discontinuity. So it turns out that the bat's brain, does not have this problem of discontinuity. If you'd like, in the questions, I can give you more detail, or you can have a look at the paper that's been published. And he was able to experimentally show it's not a spherical coordinate system, but a toroidal coordinate system. And just to say that following these studies, other groups have looked for 3D head direction cells and 3D place cells in a bunch of species and found them. So both 3D place cells in rats climbing through a complex 3-dimensional structure from Kay Jeffrey's lab, very similar to ours, 3D head direction cells, both in monkeys and in mice, again, with very similar properties to what we have found. So it looks like it's a general property across mammals. Okay, what about 3D grid cells? So this is a project that Gili Ginosar, another PhD student in the lab, has undertook. And so in 2 dimensions, let me just show you an example of a nice grid cell from the Moser lab. Grid cells are characterized, have 2 main properties. One is that they're famous hexagonality, they're hexagonal lattice, and the other is that nearby activity fields, right here again, the gray is the trajectory, black dots are spikes. The activity fields or firing fields have fixed distances between them. Okay, you'll immediately say, well, if you have an hexagonal lattice, then as the byproduct, you also have a fixed distance between nearby vertices or activity fields, which is true in 2 dimensions, but as I'll show you, it's not true necessarily in 3 dimensions. So that's why we need to look separately at the issue of a global lattice versus local distances when we're talking about 3D. So what should we, or what might we expect for 3D grid cells? So you could think of various ordered structures. For example, maybe when going from 2D to 3D, we'd find columnar structures. So essentially, the firing field will be elongated vertically, but on the XY axis, it'll be still a hexagonal lattice like here, or maybe you'll have a 3D generalization of a hexagon, because of course, hexagonal packing of circles in 2D is an optimal space packing pattern. And likewise, when you go to 3D, and you want to pack spheres optimally in 3D, there are two optimal packings, the FCC, face-centered cubic, and HCP, hexagonal closed pack. And so there were models, theoretical models, that predicted that in 3D we might find something like that in the brain, in grid cells. So we wanted to look at that. Also, maybe there are other lattices, maybe cubic lattices or some other lattices. So that's another possibility. On the other hand, maybe there is disorder, no particular lattices, or maybe something in between. So these are the possibilities that we considered. And to test it experimentally, we let bats fly in this large flight room, as I said, that we have in the lab, about almost 6 by 5 meters by 3 meters, and we put around the perimeter of the room, we put these landing platforms or landing balls on which the bat could land. They were positioned really close to the walls. So the idea was that they will not be in the middle of the room. They will be very close to the walls, but they will be at different heights. Some are low, like here, some are high, like here. And the idea was basically that the bats will be flying between them and will thus cover three-dimensional, the three-dimensional volume of the room, which indeed they did, covered the 3D volume of the room very nicely, and flew long distances, in this case, typically 2 kilometers per session, which is very nice. So we had a good three-dimensional coverage, behavioral coverage of the room. And then here are some examples of neurons that Gili recorded in the medial neuronal cortex, which is where grid cells are found. You see here 8 examples. So you see that in all of them, you have these activity fields or firing fields. First of all, it's not one. Doesn't look like a place cell in the hippocampus. Typically, in this room, we usually had 1 or 2 firing fields, but it had many firing fields, and they're certainly not vertical. Okay, so we can already exclude this possibility. We also did explicit analysis to check whether they're elongated. The answer is no. But I'm not going to show you this analysis. Just by looking at that, you'll believe me that there's no particular elongation in actually any direction. We checked all directions. So then the first thing we looked at is there this local distance that is a characteristics of grid cells. So we defined place field. We had some criteria, statistical criteria for firing fields. For grid fields, I won't go into the detail of those. And then after we define these grid fields, we can ask for each field, what are the distances to its 3 nearest neighbors, and collect those 3 distances. Then the next field, what are the distances to its 3 nearest neighbors, etc. So collect all this set of distances, of course, without counting twice. And then we can construct a histogram like here of the distribution of distances between nearest firing fields. For that particular neuron, for example, and you see that this histogram is narrow. So the fact that it is narrow looks like there is a fixed distance scale. So to ask, is this significant, we compared it to shuffle. So we can do 2 things. First of all, you can ask what is the CV, the coefficient of variation, which is the standard deviation divided by the mean of this histogram. So in this particular neuron, it's 0.17, so which means it's a low number, which means that this histogram is narrow compared to its mean. But also we can compare it to shuffle data, and actually the coefficient of variation is lower than for all the shuffles in 1,000 shuffles that we did. So it means this is statistically significant. So 20 to 30% of the multi-field neurons, the neurons that have multiple fields, had this statistically significant local distances consistent with being grid cells. 20 or 30%, it was depending on which type of shuffle we used. So that's quite a high percentage. Then we asked, is there a global lattice like this or some other lattice? And the short answer is no. I have many things that I want to show you today, so I'll skip the details of the analysis, but basically it's a negative result. We spent almost 2 years trying to look for various global structures using all sorts of techniques. And the short answer is that we could not find any hint of an FCC or HCP lattice using any of the methods. Literally 0 neurons showed a significant FCC or HCP pattern Or for that matter, we also look for other local angles. Is there a hint for cubic lattice or any other lattice? Again, the answer is no. So again, I won't go into the details here, but again, it was already published last year, so you can see the detailed analysis there. But we basically could not find any global lattices. So we can rule out this. I already showed you that. We rule out the FCC or HCP analysis, also lattices, also other global lattices. On the other hand, it is not a disorder, a complete disorder, because we see these local distances in a lot of cells. So it looks like a locally ordered system. So this was the conclusion for the experimental analysis of experimental data. And then we turned to a theoretical work together with Yonatan Al-Khadef, Yoram Burak, and Chaim Sopolinsky, three theoreticians. They undertook a theoretical analysis where the basic idea was the following: to treat, so just like in many physical and chemical systems, you can model the behavior of the collective behavior of the system via just local pairwise interactions between particles, like molecules, for example, or any other particles. Just you have pairwise interactions, that's the only thing that matters. And then depending on the parameters of the system, it can give you anything between complete order and complete disorder and anything in between. So we thought maybe if we treat the firing fields as particles and have pairwise interactions between them, this can explain what's going on in 2D versus 3D, and can explain whether you have a lattice or not, depending on the parameters of the interaction. So specifically, we use the Lennard-Jones potential that is widely used in physics and chemistry. So it's essentially potential energy versus the distance between the two particles. In our case, it's between the two fields. It looks like that. It has repulsion at short distances, with repulsion between the particles. It has attraction at longer distances. And then, in general, the particles would like to be in the minimum energy, except that there is noise or a temperature term that can cause the particles to deviate from the precise minimum where they're supposed to be. And I want to emphasize that such a potential or a similar potential can be achieved by a self-organizing neural network with competitive plasticity. People have already done in the past, not in the context of grid cells in 3D, not in the context of what we are doing, but people have already in the past done explicit neural network modeling where a network self-organizes based on competitive plasticity, and then you can get effective interactions between activity fields that look like a little bit like Lennard-Jones potential. So this is something that I think is a work to be done theoretically more explicitly, but it's actually quite possible that something like Lennard-Jones potential could be achieved in a realistic neural network. And in fact, it doesn't have to be a Lennard-Jones potential. I'm just showing you the examples for the Lennard-Jones potential, but we looked at many other potentials, and it works for all of them almost equally well. All you need is really some local minimum. It doesn't really matter the exact shape. Okay, so what we basically find is that the pairwise interaction model, this Lennard-Jones model, works really well. If you look at the y-axis here is a distance metric between the data and the model. So the lower the better, means the model expresses data better, and it's much, much lower than for random Poisson and for fields arranged randomly in space. So that's another way to say it's not arranged randomly in space. It actually has some local structure, local order. We also compared to other arrangement. For example, you could say maybe it is an FCC or an HCP, but that is you jitter the individual fields, but it's an underlying global lattice. So we compare to that possibility as well, with matching the amount of noise. And the answer is no. The pairwise interaction model, again, explains the data much better than this alternative model. So it seems to be doing really a very good job. Again, the fitted temperature is relatively high. So there's a relatively high effective temperature on the system to yield this local structure. If the temperature is very low, it will start yielding things that look like grid cells, like perfect grid cells, perfect lattices. Okay, so to compare explicitly between 2 dimensions and 3 dimensions, we use the radial density function, which is the classical way to look at arrangements of arrangements of particle in statistical mechanics. And it's basically just looking at all possible distances between all particles. And so, for example, if you look at this lattice, then if you look at the distance between this particle and its nearest neighbors, you'll get a peak. You have a lot of particles at this distance, distance 1, and then you have a lot of particles at distance 2, double the radius, etc. So you should expect to see multiple peaks. But when we look at the 3D simulations, or simulations of the model in 3D room, we don't see these multiple peaks. But on the other hand, when we squish the room to a height of 10 centimeters, so we did a quasi 2-dimensional simulation, we actually do see repeats at 1, 2, 3, 4 meters. So in 2D, in fact, we do get individual, these are just individual examples that look very much like grid cells, whereas in 3D, it looks really very much like the data where we have local, more or less fixed distances, but not a global lattice. We also did a full phase transition. It's in the paper. I don't have it here, where we actually parametrically change the height of the room, and there's really a phase transition there between having, for when you have a certain height, you have a disorder when you squish it enough to a height that's more or less the size of the distance between the fields. When you can only fit one layer, there's really a phase transition to an ordered system like here. Okay, I don't have the graph here, so I sort of described it in words. And just to give you an intuition at this point, some of you might be confused. Why is there this fundamental difference between 2D and 3D? So in fact, it turns out that it's already well known in physics there is a fundamental difference. The intuition is that if you try to take spheres now and put it on a 2-dimensional table and try to cram them as much as you can, then they will always invariably produce a perfect hexagon like you see here. And anybody here who played billiard or pool knows what I'm talking about, right? You took billiard balls, put them as close as possible, you get hexagons. That's because you have only one possibility. You have a sphere here, you have only one place to put the next one. So there's 0 degrees of freedom here. But if you do the same in 3 dimensions, if you take now a lot of spheres, balls, and throw them into a big 2-dimensional box and shake them, you'll never achieve a perfect FCC or HCP lattice, these perfect sphere packing lattices, but you'll always get something like this that is slightly less efficient by a few percent. But if you look at the local distances, like you ask yourself, what is the distance between this sphere and its nearest neighbor of the sphere, because the spheres are jammed, they're all touching each other, or most of them are touching each other. So in these jammed packing arrangements, you have a local distance that is preserved, but not a global lattice. That's because in 3 dimensions, you have 2 possibilities to put the next sphere. So you have this very fast divergence of the combinatorics of the arrangements. And that's what you'll never get the perfect lattice, but you'll get something like this. Okay, so but this gives an intuition. Something similar to this happens in the Leonard-Jones model. There's a soft model. It's not the hard spheres, but the effects are similar to what I'm showing you here. Okay, so to summarize this part of 3D grid cells, I showed you that there are cells in the medial thoracic cortex of flying bats with multiple 3-dimensional fields. 20 or 30% of them were grid cells, have a local distance scale, but no global lattice. And we suggest that the fundamental property of grid cells is not the hexagonality per se, but actually having multiple fields with a characteristic distance between them. And then in 2 dimensions, this model yields, as an emergent property, it yields a global hexagonal lattice that emerges from these pairwise interactions that are confined to a 2-dimensional surface. But if you go to 3 dimensions, that the exact same model will give you only a local structure that is a local distance metric or local metric for space, but not a global lattice, exactly as in our data. So the same model explains both grid cells in 2 dimensions in bats and rodents, and grid cells in 3 dimensions in bats. And just to tell you that in parallel to our paper, there was a paper published from K. Jeffrey's lab in rats climbing in two-dimensional structures that also did not find perfect 3D lattices in grid cells in rats. So it's not just in bats, also in rats, it seems to be rather similar. And the implications that I think of this are really important because because we have a local metric of space in 3 dimensions, this argues against models of grid cells that rely on precisely periodic firing, which is most of the models of grid cells. So basically, at least for 3 dimensions, these results argue against most models, most functional models of grid cells. And so I think this really is an important challenge for the field to understand how this works. This means also that grid cells may underlie short-range 3D navigation, but probably not long-distance navigation, at least not based on the models that people have in mind for grid cells. Okay, I don't know. Should we stop now for questions about this part and after this move to the next part, or should I continue straight? How do you prefer to do that?
Let's check if we have questions in YouTube chat, or if someone wants to.
Yeah, if someone wants to ask a question, maybe it's a good time, after each part to ask questions, or what, whichever way you want is fine by me.
Maybe one quick question.
Yeah.
So in the standard, the classical stuff with internal cortex, like people posit that, okay, the activity lives on the torus, right?
Right.
So and this is of course because of the hexagonal structure. So what is it? It is hypothesized what happens in your case of 3D but place fields. So is there any maybe a 3-dimensional space that people can put it into? Or
that's a great question. I don't know the answer to this. It could be. So I think there is, in general, there are these two sets. There is this set of results that shows that grid cells live on a low-dimensional manifold, a torus, now this recent paper, but even before that, just the fact that pairwise correlations between grid cells are preserved between behavior and sleep, etc. All of these types of results from the past suggest that there's some low-dimensional manifold on which the grid cells live. So this is sort of one thing, but this is sort of consistent with a rigid grid in an open field environment. But then there are our results, which suggest that in 3 dimensions, it's totally different.
Yeah.
Also, even in 2 dimensions, in rats, there's been a series of studies in recent years that showed that grid cells can be deformed, near reward zones, near if you do a non-square geometry like this trapezoidal experiments from O'Keefe, then grids get distorted. So all of these local distortions are also seem, at least on the face of it, to be a bit inconsistent with the idea of a low-dimensional manifold, because it's like the hexagon is broken. So I think trying to understand how this kind of continuous attractor type dynamics on a torus on one hand versus this pairwise interaction or local idea, how they live together, I think it's perhaps the biggest challenge theoretically for the field today. And I think it could live together. For example, it could be that, so there were several options. Either the activity lives always on a torus and the space of multi-neuronal recordings, but then, for example, the translation of that to the activity in space in these models is done via velocity signal. So if that velocity signal changes, for example, in 3 dimensions, or it changes in non-square geometries, or it changes near reward zones, etc., this could explain deformations that you see in grid cells while still being consistent with this torus idea. This is one possibility. Another possibility is that actually the torus itself gets distorted, right? That you have activity bumps, for example, that are not equally spaced in, for example, 3D or in these other conditions. So I think it's an empirical question, both experimentally, we're trying to look at that a little bit, but also theoretically, how do you make these two ideas live together? So that's a great question. I think it's a very important open question for the field.
Oh, actually, I have a question. The bats actually move in 3 dimensions, right?
Yeah.
So this is a hypothetical question. suppose the bats move only in 2 dimensions.
Yeah.
Then the geometry and topology of the movement might be quite different between 2D and 3D.
Yeah.
So how
this is a great question, which it's a prediction. Basically, let me rephrase your question. I think what you're saying is, suppose we let the bats move in a 2-dimensional surface, yes, while the room is still 3-dimensional, right? Of course, I can also restrict them. I can put in a low ceiling such that the environment is 2-dimensional, but I may not even need to do that. In fact, I suspect, and we've never done it. It will take a few years. So these experiments are pretty challenging, and it might take a few years. So I don't know the answer, but I think if these landing platforms around the room were not the same altitude, but will all be, not at different altitudes, but will all be at the same height, right? A string of landing platforms like on a rectangle, right? All at the same height. The bats will generally fly between them more or less at the same height. They'll not have a big reason to move much higher or lower. It might be a bit of a concave 2-dimensional surface because they sort of fall and they fly like that, right? But it'll be a quasi 2-dimensional movement, let's say, I suspect. And then we can ask, what will the grid cells do? Will they have, even though the room is 3-dimensional, but the movement is 2-dimensional, will you have equal spacing or not? I think my prediction, it'll be hexagonal because the movement surface that determines that's important, but yeah, but it's a prediction. I don't know the answer.
Okay, thanks.
I have a question. One more question. So it's maybe a kind of very straightforward question, but you got in your experiments, you got some point clouds in 3D, and I think you can check its Voronoi cells. Do you know what kind of polytopes appear as Voronoi cells in 3D?
No, we haven't. It's one of the things that was on our list to do, but we've never actually done it. So I don't know. We have looked at also local lattices, so which is something I didn't have time to go into, because you can have fractured lattices that are like locally FCC, but they keep breaking. So like local zones. We use some algorithms that can compute that, and again, we found no evidence that something like this happens. But we didn't do the Voronoi around each of these vertices, or what's the shape of that. So I don't know.
Yeah. Okay. Thank you.
Okay, maybe I'll continue on. And
we have one YouTube question.
Okay. We have one more question, then I go on.
Yeah, about grid cells also. As the spheres being packed, are all of them of the same size?
More or less, yes. We analyzed that, and the size, it's biology, so it's noisy. It's not exactly the same size, but they're more or less of the same size. I mean, we didn't see any systematic changes. Sorry, within, maybe I didn't understand the question, but I mean within the same neuron. So one neuron has many firing fields, and each of these firing fields is more or less the same size. If that was the question, that's the answer. If the question was what happens across different neurons, so here we actually did record across the dorsoventral axis of the medial entorhinal cortex, and just like in rodents, we found that when you go more ventrally anatomically, you find grid cells that have fewer fields. The fields are bigger, and the spacing between them, or the lambda, or the distance between them is bigger. So between different neurons there are differences, but within a neuron the different firing fields are the same size. I hope I answered the question. Okay, so let me move on and talk about the next point that I made about the representation of very large spaces in the brain, right? That real life navigation occurs in very large space scales, not 1 meter. So how would that affect the system? This is the work by PhD student Tamir Eliav, who just graduated, and together with Shir Maimon. So basically, the motivation for this is bats navigate hundreds of meters underground, like in these caves in Israel, and also outdoors. I already showed you an example of this bat, and this can serve to illustrate the problem. So if you look at this particular example, it's just an example, but it can illustrate the problem. The volume that the bat moves through is huge. So in this particular case, it's 15 kilometers long by 2 kilometers wide. The flyway is 15 kilometers long by 2 kilometers wide by about half a kilometer high. How do I know it's half a kilometer? Because we've measured with the GPS altitudes from about 100 meters to about 600 meters. So it's about half a kilometer high. Okay, so we're talking about 15 kilometers by 2 kilometers by half a kilometer. So about 15 cubic kilometers volume. If you want to tile a volume of 15 cubic kilometers using a laboratory-sized place fields measured in small boxes that typically are about 10 centimeters in size. So you want to take 10 centimeter spheres and use them to tile a volume of 15 cubic kilometers. You can all do the calculation in your head or on the back of the envelope. And depending on how much overlap you would like to get, you need something between 10 to the 13 to 10 to the 15 spheres, or 10 to the 13 to 10 to the 15 neurons, if we're talking now about place cells in the hippocampus that have single place field typically, you would need 10 to the 13 to 10 to the 15 neurons. Now, the entire dorsal hippocampal area CO1, this where the place cells are located, is about 10 to the 5 neurons. That's the order of magnitude. Okay, maybe 200,000. Not 100,000, but it's an order of magnitude of 10 to the 5, not 10 to the 15. So we have 10 orders of magnitude of a problem here. It's not a small gap, it's an enormous gap. This means there's no way that what we find in small laboratory environments translates one-to-one to what's going on outdoors. It has to be different. Either a place field scale up inside or something else must happen. Otherwise, there's no way it's going to work, because we have a 10 orders of magnitude of a problem here. So this was sort of the motivation, and to address this experimentally, what is the representation of very large spaces? We had to solve 3 technical problems. One is we want to be able to record neural activity from single neurons in the brains of bats, in this case at unlimited distances, very large distances. So for this, we developed this 64-channel neural logger. So it's unlike this telemetry device that I told you about in the beginning for the place cells. This one is storing the data. It's not transmitting the data, it's storing the data on board. So now it's really unlimited in distance, at least in principle. And you can use it to record really nice spikes in flight. These 4 traces are the 4 channels of one tetrode, and these are spikes. So for those of you who have seen electrophysiological recordings, this is supposed to impress you, that you can get really nice signal-to-noise ratio. This is in flight from the hippocampus of a flying bat. So it works really well. And in addition to the 64 neural channels, we have also behavioral sensors, so ultrasonic microphone and 9-axis motion sensors, so a gyroscope, accelerometer, and magnetometer. We can add even a camera. We can add a lot of different things. So unlike the typical experimental setup where all the behavioral sensors are external to the animal, here we put all the behavioral sensors on the animal, and they're flying with the animal. So you can think of it as an entire onboard flying electrophysiology lab. It's very small, really tiny. And this has been really required some technology development that we've done with some engineers here in Israel. And over the years, we've miniaturized this wireless technology quite a lot. I mean, some of the miniaturization is because just generally electronics has improved over the years, kind of unrelated to us. But you see that today, the 64-channel system that we are using today is smaller than the 16-channel system that we used when we started some years ago. This is the 16-channel system. What we have today is even smaller than this and 4 times more channels. And we are moving now to higher channel counts. It's pretty easy to use to move to 128 or to 56 or to make a neural logger for neuropixels or things like that. It's relatively straightforward nowadays. Okay, so this is one thing we had to solve. The second problem we had to solve is to, and of course, these kinds of systems allow you, we develop them for the issue of scale, move over large scale, but having a wireless system is very useful for many other things. For example, if you want animals to go under something, rats or mice to go inside tunnels, wires will not work there. Or if you want multiple animals interacting, which actually I'll show you later on, try to record from 10 animals with 10 cables, you'll get a big mess very quickly and no neural recording. So again, wireless is very helpful when you want to go to more natural behaviors, which I remind you is the general theme of the philosophy of the work in the lab. Second problem that we had to solve is to measure the positioning of the animal very precisely. So we could use GPS. The problem with GPS is that it's not very precise. It gives a precision of a few meters or maybe 1 or 2 meters at best. And we want to be able to dissociate between 10-centimeter place fields, as I said, and something else. So we need to be able to measure more precisely the GPS. So we found this solution from a French company that is like GPS. So it relies, how does the GPS works? It measures differential time of arrival of signals from satellites in outer space. But then, of course, there's atmospheric aberration, so that introduces inaccuracies. Here, we can deploy, instead of satellites in outer space, we deploy an array of ground-based antennas on the ground. The distances are shorter, so it all becomes much more precise. And then we can reach, originally we reached a precision of 10 centimeters. And we have recently added more antennas and reached a precision of even 5 centimeters. And over long distances. And also we want to build a large behavioral setup. So we built this tunnel. It was originally 200 meters long tunnel like this with multiple landmarks that can unequivocally tell the bat where it is. And so the bats were flying in there. This is now shows an example of a behavior from one bat. This is the position, 200 meters. And this is the time within the session in minutes, and the bat was flying back and forth and back and forth and back and forth. We put 2 food platforms at both ends. Some of the bats took the food, some didn't even care so much about the food. They just literally enjoy flying. So we let them this enormous space, and they just would fly many, many, many times, eating 5 or 10 times during the entire session, because food is not necessarily their main motivation. It's really the flying itself, which they love. And some of the bats did up to 100 or even 120 flights within one session, which is 20 or 25 kilometers in one recording session, which are crazy distances. You'd never get this in a small environment. And they flew really in the center of the tunnel. This is the cross section of the tunnel. They flew really in the center. So we turned this into a one-dimensional problem on purpose, because here we really want to ask a question about the scale of the environment. So we want this to be simple and one-dimensional in this case. And they flew at a very fast speed, 7 or 8 meters per second. This speed was very fixed along the entire tunnel. This will be important in a second. So in this case, we're utilizing the bat's flight speed to measure the representation of large spaces, right? If we'll use an animal that moves much slower, like a rat, it can do a couple of traversals of this tunnel, but then that's it. It can't do anymore. And here, because it moves so fast, we can get 100 repetitions of this enormous space and get really nice rasters. So this is an example of one neuron that Tamir recorded. And what you see here at the top are firing rate maps, firing rate versus position in the tunnel. And these are, the bottom are rasters. So each line here is one flight, dots are spikes, so it's a different flights. And we separated the flight to the east in blue and to the west in red. So what you see from here are several things. First of all, that the red map is different than the blue map. That's actually well known in the past in the laboratory environments as well, that place cells fire differently when flying, when moving in 2 directions. We've actually shown that in bats as well in the past in small environments. What's more interesting and new is that, first of all, some of the fields are really large. You see this field, for example, it's 20 meters or so, and we've even seen bigger fields. So certainly there is scaling up. You increase environment size, place fields scale up. Secondly, you see multiple fields, right? The neuron is active here and here and here in many locations. And third, and perhaps most surprisingly for us, the size of the place field was different in different locations. So there's a big field here, a small field here, a small field here. Here's another neuron, has a big field here, a small field here, a small field here, a medium field here. So neurons have multiple fields, and the different fields are different in size from each other. So we have here, some large fields, also small ones. We have multiple fields, and this what we call the multi-scale property, where the same neuron can have different scales or different sizes of place fields in different locations. Which all of this looks very different from place cells recorded in small laboratory setup. We've also recorded in a small portion of the tunnel. We blocked off 6 meters of the tunnel, and then we see single place field. Everything is normal, right? So basically, it's an issue of scale, really. And that just summarizes the population results, where the number, if this is the number of fields per direction, we get sometimes 10 or even 20 place fields in one direction. It's not your single Gaussian as you typically think of in the hippocampus. And this is the multi-scale property. This is the field size ratio between the largest and the smallest place field, right? So the size of the largest place field divided by the size of the smallest place field for the same neuron, right? And this could be 5, 10, or even 20. 20 means that the same neuron has a 1 meter place field here, but the 20 meter place field in a different location in the tunnel, right? So it's really a multi-scale property or multi-resolution. So this is very, very different than typical neural codes in the brain. Think of V1, right? It's like having a V1 neuron, a visual neuron, that has 20 different or 10 different receptive fields that vary in size 20-fold between each other. This is pretty crazy. So, okay, maybe I'll skip some of the controls. The fields did not cluster nearly stable along the. Really, this is a genuine result. And so, again, we have this very different type of neural code for very large environments. So to understand that, we teamed with Yonatan Al-Khadef and Misha Tzodik's to try to understand how that, what's the advantage of, functional advantage of having this weird type of a neural code. And it's really, I have one slide here that's summarizing a big work here. So I'll try to make it justice, but it's really just give you the gist of what we tried to do, or what Yonatan and Misha did here. So basically the idea was to take a decoding approach and compare various encoding schemes. So on the left, so we compared in this case, 6 different encoding schemes. In fact, we had 8 different coding schemes, but here I'm showing just 6. So look at the left here. This is sort of shows the firing of 10 different neurons. Each row here is a neuron over 200 meters. So each neuron here fires in one location. The blue is the place field of the neuron. So this neuron has a place field here. This neuron has a place field here. So different neurons have one place field per neuron. They're small and they're in random locations in the tunnel between different neurons. Okay, so this is the classical place code. On the other extreme is scheme 6 here, which is basically trying to capture what we find in the, and it's actually fitted to what we find in the bat, which is different neurons have many fields, and the sizes of these fields vary across neurons. By the way, the distribution of sizes is log normal. It's amazingly well fitted by a log normal function. Okay, so, and then we looked at other coding schemes. I won't go into the details here, but the bottom line is that what I'm showing you here is what we call catastrophic probability of catastrophic error. Catastrophic error means a decoding error that is bigger than 5% of the environment size. So it's a bit arbitrary, but it just means a big decoding error. And we would like to look at the probability of that versus environment size. So this is simulation. So we can now simulate anything between 20 meters and up to 1 kilometer. So the result was that in a small environment, 20 meters, all of these schemes did equally well, but in a large environment, there was a big difference. 200 meters, which is our environment size, the experimental one, the purple scheme 6, which captures the experimental data, had a much lower probability of catastrophic error than scheme number 1 in red, which is the classical place code. In fact, this is logarithmic scale, so it's more than 2 orders of magnitude better than the classical place coding. Okay, so our answer is that the reason we need this multi-field, multi-scale code is that it is much better in terms of coding, specifically for large environments. For small environments, anything goes. But for large environments, you can encode environments up to 1 kilometer with only 50 neurons pretty precisely, and we show this in the paper. Okay, so to summarize this part, on spatial coding of large spaces. It's very different from lab setups. We have many fields per neuron, often more than 10 fields in each direction. We have some very large fields, up to 30 meters or even more, but also very small fields. Some of these fields were 1 meter or even 50 or 60 centimeters. So it's not that everything is blurry. Some of the place fields can be extremely precise, but then the same neuron that has a 50 or centimeter or 1 meter place field will have a 20-meter place field elsewhere, which is this multi-scale spatial coding. We did also, Maimon did experiments that showed that this multi-field, multi-scale coding is independent of experience. This is a whole different set of several sets of experiments. She recorded from day one and showed that already from day one in the tunnel, from the first day the bats are put into the tunnel, we already see this multi-field, multi-scale code, and it's stable across, from day one onward. And very surprisingly, she also looked at laboratory-born bats versus wild-born bats. So everything I showed you so far is for for wild-born bats. So we actually go outdoors and I catch myself the bats that are wild bats in Israel outdoors. We catch them as adults. So these are bats that have seen large spaces, 3 dimensions, everything outdoors. And then we bring them to the lab and do the experiment. But we also have a colony in the lab, so we can take labs that were born in the lab. They have never seen 200 meters; they're only seen small rooms, and we can ask what will be in their brains. We expected there will be a big difference, but the result was striking. That there was actually no difference. We've seen exactly the same multi-field, multi-scale coding. The histograms were completely overlapping between the wild-born and lab-born bats. So this is a very striking negative result, but it suggests that there's something really very basic and fundamental about this multi-field, multi-scale code that you see it, even in lab-born bats. And we have a theoretical analysis of decoding errors that explains why multi-scale coding is better for large environments. So this was all, looking at what individual neurons do. We've also recently, Tamir Eliav started to look at neuronal populations in freely flying bats. So we've used a decoding algorithm, a cluster decoding algorithm developed by Lauren Frank's lab. I won't go into the details, but it works really amazingly well. We can decode. So now, based on all the neurons that we have, we can decode the position of the bat versus time extremely precisely. So this shows examples of 2 flights of 2 bats. The black is the real trajectory. The red, I don't know if you can see it, is the decoded position. They completely overlap. So that means the decoding works really well. This is the confusion matrix of predicted position or decoded position versus real position. The color is the probability of the decoded having a particular decoded position. So it all falls very precisely on the diagonal, and we show that in many of the recording days, we have a median decoding error of 50 centimeter, a median 50 centimeter for an environment of 200 meters. So that's 0.25% of precision. That's very, very precise. So during behavior, we can decode the position of the bat very precisely, which is nice. But why is that interesting? Because then we can apply the same decoder while the animal is in rest, on the balls, resting, or during sleep. And why is that interesting? Because as some of you know, during rest or sleep, people have observed in the hippocampus of rodents what's called replay sequences. So place cells will replay sequentially one after the other in the same order as they were activated during the behavior itself. And this is thought, for example, this replay during sleep is thought to be the basis of memory consolidation. It's literally as if the hippocampus is replaying what happened during behavior, and then we know that these replay occur during sharp wave ripples, and this is being transmitted to the prefrontal cortex or to the neocortex generally for long-term memory storage. This is sort of the idea or the gist of the theory that's pretty dominant in the field. And so this is why it's interesting to look at that and look at how the replay looks in very large environments. And so, yes, we can have replay. We do see replay in this very large environment. So this is now the decoded position versus time. This is the decoded probability. So just like in rodents, we see that these decoded kind of these replay events are short in time, a few hundred milliseconds, and you already see that they don't. These 3 examples don't span the entire environment, which in our cases was 130 meters. a segment of 130 meters of the tunnel, but they replay a piece or a segment of the tunnel. In fact, this was generally, they occur during sharp wave ripples, just like in rodents. And so this is sort of summarizes the result that we have. The duration of these replays is short, is a few hundred milliseconds. This is the distribution of the duration of the replays during sleep and during wake. The compression ratio, which is the ratio between how fast the replay unfolds, because you can define speed here. This is position versus time. So the slope of this is speed of replay. How much of the speed of replay divided by the speed of flight during real behavior, this is this is called the compression ratio. This is the distribution of that. It's about 10 on average, just like in rodents, okay? So that means the replay occurs at a speed that's 10 times faster than the actual real world behavior. But the result of that is that the distance that's being replayed is short, is on average about 10 meters. We never see 200 meters or 120 meters. It's always, so the 20-meter replay example that I showed you here are actually quite extreme, right? This is about 20 meters here. If I look at the y-axis, this is actually extreme examples. Typically, it's more like 10 meters. So it means it's really being replaying short segments. So this is very different than what you see in rodents, where typically the entire environment is being replayed. So, so it's sort of depicting short snippets of long experience. So you can think of two different interpretations. One is that the piecewise replay of information in this large environment might be like using small information packets for transmitting large messages in artificial communication systems, right? Think of the internet, right? If you want to transmit a small file, you can, or small message, you can transmit all of it at once. But if you have a big file to transmit, it's being broken into pieces or packets and being transmitted in packets. This is how the TCP/IP protocol works, right? And so maybe something like this is happening here. The information is being broken into packets. Another idea, we've been talking to people who train deep neural networks, where one network is training another network, and they told me that they use similar training strategies, where you feed short, small snippets of the information. That's better for training deep neural networks than feeding all information at once. There's some interesting theoretical reasons for that, but empirically, that's what they all do. And they said it looks like what we are seeing here, these short snippets, is essentially the hippocampal network is training the neocortical network. This will be another interpretation of what we are seeing here. Okay, so this is another work in progress. And okay, maybe I'll just skip that. And I'll just tell you that we are now building or actually not building, but finished building a 700-meter tunnel plus a very large maze that's 60 by 35 meters in size. And here in the tunnel, we're asking questions about coding of large spaces across different hippocampal subregions. We're already starting to see interesting difference between CA1 subregion of the hippocampus and other subregions. And we can ask questions about representation of multi-scale environments. For example, this little tunnel versus the big tunnel, is it represented differently? Think of this little tunnel as my room here. I certainly have a high resolution representation of that, but when I go outdoors, it's a bigger environment, probably the resolution is not as good. So how is the brain representing these situations where I go from my office to outdoors, or from this little tunnel to the big tunnel? Are the same neurons participating in representing both sub-environments or different neurons? And if it's the same neuron, do they rescale, etc.? So many questions like that. And again, we are recording large ensembles of neurons in flight while doing that. And in the maze, which we're now starting experiments in, we're asking questions in general. How does the brain represent complex environments? Because this is much more complex than ever tested before with electrophysiology. What happens in the brain when we ask about representation of multiple goals, because we put here multiple navigational goals, 4 navigational goals in this case. So we can ask, how does the brain represent multiple navigational goals? What happens when the animal needs to reorient ourselves? So what does it mean, reorientation or recomputing the path? What does it mean in the brain? Decision-making, planning. There's a lot of aspects that we are starting to look at in this very large maze. Okay, so I think this is for this part. Maybe we can ask 1 or 2 questions about this, and then I'll move to other stories. How much time do I have, by the way? I should finish at 5:30, including questions or without the questions? Like, where do I stand on time? I'm not exactly sure.
Well, actually, we expect to finish with question by 5:30, but I think if we will have a slight delay, of course, we will give Moo a little bit more time if he doesn't mind giving shifting for another 5 minutes.
Okay. Yeah, so maybe if people have 1 or 2 questions, I can answer now and then move on to the next part.
I have a question about this multi-scale and multi-field phenomenon.
Yeah.
But if you take some regular, for example, 2-dimensional lattice and put some circles of fixed radius in each node, and then choose some random line, straight lines through these circles, that it seems to me that the behavior of intersections will be similar to what you showed us on your line. Can't it be the explanation of this behavior? So maybe there is some 2-dimensional structure that you just don't observe in your experiment with line.
We actually tested exactly that. So you mean if we take circles and arrange them randomly, or if we arrange them in a hexagonal lattice?
Yeah, these are 2 questions.
In hexagonal lattice, okay.
Question, or according to some of other models, some regular models.
Okay, so we haven't tried what you're saying. If I arrange circles not hexagonally, but just put them somehow randomly in space and then put a 1-dimensional line through them, maybe this can explain the data. Here, the answer is maybe yes, I don't know. What we did test is if you take like grid cells, basically circles on hexagonal lattice and go in some random angle, this can also give fields of different sizes, right? Which is not necessarily going to look like a grid because, you here you go to the center, here you go to just the edge of the circle. This we tested, actually. I didn't have time to show you. It's in the paper. It's in one of the supplementaries. We tested that, and this definitely cannot explain the data, because if that was the case, we could show that if you look at the spectrum, the spatial spectrum of the firing activity, you'd expect to see periodicity in the activity, and this was completely like 1/f, no spectral peaks. So this actually, we completely ruled out this possibility. But the other possibility of random circles that you cut with the line, yeah, maybe. And I don't think it'll be easy to test with our data, right? Because I'm not sure what will be the prediction that will be.
Yeah, but maybe in your large maze and this large tunnel,
perhaps, yeah,
you can observe something like that.
Mm-hmm. Yes.
Okay, thank you.
Is there another question? Maybe a last question, then I move on.
Well, I have a little question.
Sure.
First of all, it's very cool that you have noticed that replace acquire only on short snippets. I guess what you mean, an analogy in machine learning is called a batch size.
Mm-hmm.
And I have a question about also temporal structure of replace and how place cells ensembles are being restructured. It's very well known that the neural ensembles, in particular of place cells, tend to migrate over different cells within some time. And have you tested if it happens? Well, of course it does. It happens in bats, mouse brains too, but does it preserve this sparse structure of place fields?
So by migration, you mean the slow change, like drift over many days?
Yes.
Okay.
That.
Okay, so we looked at that. Okay, we use electrophysiology, not imaging, so we can't really record the same neurons over a month. But we did see, even within a given day, that some place fields appear and disappear. So we do see some dynamics. Actually, it's nice because we have multiple fields, so we have an internal control, right? Because let's say if a neuron has, in this case, 4 fields, right? and 3 of them you see over the entire recording, but 1 disappears, actually the fact that these stay there, it's sort of a control for stability of recording, etc. So we have an advantage here in a way. And we do see such cases of fields appearing and disappearing. It's a low rate. It doesn't happen very often. We did actually look at what happens in the first 1 or 2 days versus later. In the first 1 or 2 days in the environment, from day 1, when the first day that is introduced to this environment, and the first and second days, more or less, it happens at a higher rate. So this appearance and disappearance of fields happens more often, and then the rate decays, but it does not go to 0. Even after a month of recordings, we do see fields appearing and disappearing from time to time, but the statistics maintain exactly the same. So first of all, the rate of appearance and disappearance is exactly the same. So the number of place fields is fixed over the month or 2. And but also the general statistics, this log normal distribution, etc., is very stable. But it's hard to compare the rate of this to mice. I think it seems to happen at a lower rate than in mice, but it's electrophysiology versus imaging, small environments versus large environments. It's hard to make these quantitative comparisons, but generally we do see some dynamics.
Thank you.
Okay, maybe I should move on just in the interest of time, and then at the end we can also have additional questions on this if people want, of course. Okay, so the third point that I made is about the social aspect, social coding in the hippocampus. So David Omer, who was a postdoc in the lab, now he has his own lab in the Hebrew University. He asked a very, seemingly very simple question. So, because bats are very social mammals, maybe you should expect that in the hippocampus of bats, where you find place cells, the neurons that represent where I am in space, maybe you would find also neurons that represent where you are in space. So this was the question, and to answer this, we developed this task where we had 2 bats, a demonstrator in blue and an observer in red. So the demonstrator was flying from a start ball to ball A, let's say in this case, and the demonstrator was supposed to, and the observer was supposed to mimic it. Okay, if the demonstrator goes to ball B, then the observer also should mimic it. So it's a mimicry task, and we recorded in the hippocampal area CA1 in this task, and what we found, this is an example of all of this is 1 neuron, that when the observer bat, the implanted bat, was flying itself, here the gray trajectories of the flight of the bat itself, here's an example of a neuron that fired in a particular location, so it's a place cell basically. But when the bat was not flying anywhere, it was stationary on this rest ball and was observing the other bat, the neuron still fired in a spatially confined area. So it's essentially encoded the position of the other bat. So we call these neurons social place cells. And there's many details here. We could rule out trajectory planning, because you could say, maybe I'm not really representing the other bat. Maybe while the other bat is flying, I am already planning my own trajectory. So really what you see here is a representation of my own planning. So we had 3 different analyses to rule out trajectory planning. I won't go into the details. You can ask, is this really social, right? Maybe anything that moves through space will elicit this type of activity. So we compare the responses to a live bat, to the conspecific, versus responses of 2 plastic objects that were moved through space. And we found that some of the neurons also responded to the plastic objects, but always differently than to the live bat. So it seems that there is something special about the social aspect. And so we suggest that the social place cells may be important for social spatial cognition, so representing a specific, but also maybe other things like predators and prey. So we as humans are very good at knowing where our friends, think of a party, where are my friends, etc. Or in football or in basketball, the really good players, they're very good at knowing where are their friends or the people in the other team. So this ability to know the locations of other individuals is very important for humans and for animals, for any social animal, you might argue. And so these neurons may underlie these abilities. So this was already published, and in recent last few years, we tried to push this further. Saikat Ray, a postdoc in the lab, is recording from this bat colonies. Essentially set up a colony of bats that have between 5 and 10 bats that live in this room 24/7 and interact, and we develop these methods to track the identities and positions of many bats simultaneously in this three-dimensional room. So it's actually not so trivial to do that, but we develop methods to do that. And then we see, first of all, that as the bats, when the bats are flying between this location and that location, so when you look at this flight trajectories, there's a neuron that fires in a particular location in the air. So you'd say, okay, this is a regular place field in the middle of the air. But it turns out that this neuron cares about whether I'm flying to a location where there are other bats or when I'm flying to the same location when there are no bats. So you see that the neuron fires very differently in this condition. So essentially, if you're going to a party or you're going to an empty party, this neuron cares a lot. So this is a social modulation of place cell firing, if you will. We also showed that these neurons can represent the identity of the bat. I won't go into the details, so it matters like which other bat is there or not. And we also looked at, on these nets in the ceiling, what happens in terms of the representation of other bats. Now, this is a very multidimensional problem because if you think about it, I have, let's say, 5 other bats. So there's my position, my head direction, but there are also the positions of 5 other bats. There's distances of 5 other bats. There are angles of 5 other bats. Very quickly, this becomes, a 20-dimensional behavioral problem. It's like computing a tuning curve in 20 behavioral dimensions. Very difficult. So that's not what we did. To do this, we took another approach. We used a GAM. GAM is a generalized additive model. It's a little bit like GLM. GLM is, I'm sure we all know about this, is a linear. It's a linear model. It's very powerful. So GAM is a non-linear model, so it's more powerful than GLM. But the advantage is that, like GLM, we can assign the contribution of each of the components. We can ask how much does the position of bat 1 versus bat 2 versus bat 3 contribute to the model, etc. So using this, I'm now showing you just one neuron. And you see, first of all, that if you look at the firing rate in blue of the neuron versus, over 1 hour, the model in orange captures this firing rate really nice, tracks the actual firing rate very nice. So the correlation between the blue and the orange here is very high, 0.6. But then, as I said, we can ask how much does this component contribute, and it turns out there are 3 main components that this neuron cares about. One is my own head direction. So this is the real empirical head direction. This is what the model says. Very nice match. This is the place tuning for my own position. Again, nice match with the model. But the model says also that this neuron cares about the position of bat number 5. Not bat number 4, not number 3, not number 2, only bat number 5. So there is this means that there's identity coding here. There is coding of a location of a particular animal, but not other animals. And again, different neurons encode different animals, and we're now trying to analyze whether there's different coding for, for male versus female, as a function of a hierarchy, social hierarchy, etc. So there's many social questions that were never really looked at in any species, and we are now analyzing this in the bats. And lastly, I'll already switch to the last story, and then I'll open to general questions. It's about the last story is about natural switches in behavior that rapidly modulate hippocampal coding. This work by PhD student Alexei and Shakil Palgi and Dan Bloom, all of them PhD students. And basically the rationale for this study was that natural behavior is very dynamic. So for example, when I walk from my home to work, I live a few minutes walk from the lab. So as I walk, initially I navigate, then I get to a junction. I need to decide, do I turn right or left? Because there's 2 ways to get to the lab. So there's decision making. Then I might meet a colleague or a friend on the street and talk to them. So there's social interaction. So within these few minutes that I walk from home to work, I switch between navigation, decision-making, and social interaction. That's how real-life behavior looks like, right? But that's not how neuroscience research is done. Typically, in neuroscience research, we do one thing at a time. You do now navigation research. And then another lab in another brain area would study decision-making. Then the third lab in a third brain area in the third country will study social neuroscience, right? So it's all, one thing at a time. But real life behavior is very dynamic. So we wonder, how do neurons respond to this very dynamic situation? And in particular, navigation is also very dynamic. You navigate, but you need to pay attention to conspecifics, predators, prey, etc. So we try to create a situation that mimics this dynamism. So in particular, we did this experiment with 2 bats in the tunnel. So the same tunnel as I showed you before, but now we record from 2 bats. And either one bat is flying alone. This is, we call a solo situation, or the 2 bats fly versus each other and cross over each other. We call this a crossover. This is not in different blocks. This is completely intermingled, right? Because basically the 2 bats are always there. Just, when one bat is flying and this one is resting, we call this solo. When they're both flying, we call it crossover. So it's completely intermingled. And basically these crossovers happen in all possible locations in the environment. And we can have 2 extreme hypotheses. One, for example, for a pure place cell, if it's a pure place cell, and you plot the firing rate, the hypothetical firing rate of a hypothetical classical place cell as a function of position in the tunnel and distance between 2 bats, you would get something horizontal like that. Okay, I showed you before, this is one place field. I showed you before that we have multiple place fields, so maybe you should have 3 or 4 or 5 bands like this, but they're all horizontal. No modulation is a function of distance. This is for a classical place cell. For pure distance coding cells, you'd expect to see something like that, right? Doesn't care about position, but is tuned to distance, in this case, distance 0. Okay, in fact, we did find some neurons that looked like these extreme cases. So here's a neuron that's more or less a classical place cell, is not very much modulated by distance. Here's a neuron that's more or less a pure distance cell, doesn't care so much about position, and here it's distance tuning. But in fact, most neurons showed more complex combinations. So here's a neuron that responds to a combination. Again, the color here is firing rate, and it responds to a combination of a particular position and a particular distance. Here's a neuron that's actually inhibited at a distance 0, etc. So we saw a whole zoo of responses there. And again, we have a lot of control. This was just published last week, so you can already look up this paper. And we had control. These sort of responses could not be explained by lateral deviations or changes in speed, all of these other things. We have very extensive controls for that. And across the population of neurons, if we stack all the distance tuning, so these are the distance tuning of a neuron, if we stack all of them, color code them, and we stack them for all neurons, then we see tuning for all possible distance, but with some overrepresentation of distance 0. Now, the nice thing is that we can ask, for example, what is the effect of attention? Because one of the advantages of the bat is, as I said, we can record on board the sonar signal. And we know for many years that bats, as they attend to something, they increase the rate of their sonar calls. Okay, this is known way before us, but we can utilize that. So here you see an example of that. Here, 52 trials mean 52 crossovers, and this is a raster, not of spikes, but of echolocation clicks. Each dot here is an echolocation click as function of inter-bat distance, and you see that the bats increase the rate of their echolocation click before they meet each other. So this is an index of attention, if you will, and this is nice because we have here, this is how it looks when you sort of collapse it, and we have here an independent behavioral index of attention, which is a great advantage in bats. In a freely moving rodent, how exactly are you going to measure when is it attending to something? It's hard. In bats, you put a microphone, you record echolocation, and the animal tells you I'm attending to something. So that's a great advantage. And then we can ask, does the level of attention affect the firing of the neurons? And the answer is yes. This is what Shaket Palgi looked at. So if we divide the flight to those with high click rate or high attention versus ones with low click rate or low attention, during high attention flights in purple, the firing rate of these neurons was higher than during the low attention flights, and it was the same across the population. And we could decode the distance between the bats better during the high attention flights than during the low attention. Again, it's all in the paper. I don't have time to show that. And we could rule out, it's not because these neurons are auditory and there's like responses to auditory responses to individual clicks. In fact, most of these neurons did not respond to the individual echolocation clicks. It's more of a state of high attention versus low attention. And lastly, we asked we found that there was a very complex combination. So if you look now at this neuron on the left, you see that at this position in the tunnel, it's not tuned at all to distance, so it's flat like here. But at this position, it actually shows distance tuning, so it's tuned like that. So the tuning to distance is different in different locations in the tunnel. For this neuron, it's even more extreme. It's inhibited in one location as a function of distance, but it's excited as a function of distance in another location in the tunnel. So it looks like there's a complete non-separability. So if you treat this as a two-dimensional function of x and y, of distance and position, the two-dimensional function of x and y is not equal to the multiplication of the marginals of a function of x versus multiplied by a function of y. And again, the data supports that. So really, it looks like the firing with the distance tuning within different place fields is completely independent of each other. And so there is a really non-separable representation of position by distance. And again, we teamed with Yonatan Khadef, and he did a theoretical decoding analysis that showed that such a non-separable representation actually improves coding. All the details are in the paper. Unfortunately, I'm running out of time, already ran out of time. So let me just summarize this. That during crossovers, many place cells in hippocampal area C1 transiently encoded the distance from the other bat. This neuronal representation of the other bat was very brief, lasting for only a few hundred milliseconds. And then it switched back to the normal place tuning of the bat, right? So basically, there's a switch from coding of my own position. Then when the bats approach each other, it switches to representing the position, but also the distance of the other bat. And then it switches back to representing my own position. And we have evidence that it's really a switching rather than a multiplexing. Again, in the questions I can explain why, and it's all in the paper. But it looks like a really fast switching, and this switching occurs within as fast as 100 milliseconds. So that suggests that the neural coding is very dynamic. The neuron really literally switches its basic processing or basic function or its neural code, if you will, from one thing, position, to completely different thing, position and distance, very rapidly when the behavior is very rapid and dynamic. And here again, we capitalize on the fact that the bat's flying very fast. Because they fly so fast, it's 7 meters per second for this bat, 7 meters per second for the other bat. So it's 14 meters per second combined speed. That's 50 kilometers per hour. That's really fast. So everything here happens extremely fast behaviorally. That's why we saw this very fast neuronal switch. So let me summarize. These are my last two summary slides. That, as I started in the beginning, I talked about the importance of going to more natural behaviors. And today I've showed you that if we go, none of what I showed you was completely natural, right? But there were more natural aspects of behavior than typical. So for example, when we went to movement in 3 dimensions, we found 3-dimensional place cells, we found 3-dimensional head direction cells that had these toroidal coordinate systems. Grid cells looks completely different, which rules out most of the theories of grid cells. So we found something really novel and surprising. When we went to natural spatial scales, all hell breaks loose. We see this multi-field, multi-scale coding that looks completely different than in small environments, and we actually have an understanding why is this useful, because coding is better. When we looked at social interaction, we found representations of other individuals, of the identities of other individuals, etc. And when we allowed the animals to switch its behavior in a rapid manner, we found very rapid dynamism in the neural code. And so the take-home message, as I said in the beginning, is about the importance of studying natural behaviors. Of course, science in the last 400 years have been very reductionist. And I come from a physics background, so of course, I don't deny the importance of a highly controlled and a reductionist approach. On the other hand, I always worry that if we're interested in the neural basis of behavior, if we control too much and we reduce too much, we might miss the very thing that we came to understand, which is behavior, because behavior is rich and complex and context dependent. And therefore, I think we need more diversity in the approaches. It's okay to have the reductionist approach, it's important, but we also need to go into more natural behaviors. And as I told you, every time we do it, we find something surprising and interesting about the brain. And in fact, I'm writing now a book for MIT Press called Natural Neuroscience, where I try to make the argument that for any brain system, it's important to look at more natural behaviors. And in fact, there's already hard data out there in many of these systems to show that neurons care about whether the behavior is natural or not natural. And so I hope to convince the neuroscience community in the importance of going towards more natural or naturalistic behaviors. And with this, I would like to end and thank lab members, especially Liora Lass, who is a fantastic staff scientist who has been working with me, involved in everything I showed you today. She's really amazing. And I think I mentioned most, or actually all, the students and postdoc who did the work, and alumni. Most of them have their own labs now, and funding sources. And thank you for listening.
Professor Ulanovsky, thank you very much for your beautiful and exciting speech. Let's thank our speaker.
Thank you.
Yeah,
yes,
thank you very much.
Questions, I'll be happy to take.
Dear friends, do you have any questions? Also, YouTube chat, let's check it. Okay, it seems like all the questions have been answered during your great speech. Or probably Vladimir has some question? No? Okay. Then let's thank Professor Ulanovsky once again.
Thank you so much.
And we're welcoming our next speaker, Professor Chung from Wisconsin-Madison, who has an amazing study on dynamical TDA, which I'm just eagering to repeat on the cell level, of course. Okay, Moo, can you share your presentation?
Okay, let's start.
Thank you for your waiting. Okay, we can see it. Thank you.
Okay.
You're welcome.
I'm going to talk about physical clustering, topological clustering methodology for estimating the space. Usually, space estimation is done by the usual ordinary differential equation model, but we will do it without model, purely data driven. So this is a bunch of people who helped us on this project. So before we started, actually, this paper is actually, you may want to read this paper. This talk is based on this particular paper. Sorry. Before we start, actually, we are organizing the workshop, topology data analysis workshop. This is a second year. We actually did it last year as well, and this time it would be hybrid. We will have a Zoom session. I mean, we basically have a Zoom, but at the same time, there will be actual workshop in South Korea. Postdoc is one of the top science university there, and so I'm actually going there. I am applying there. So I think we may actually have it. Actually, this is it's September, the last week of September, and I hope that we can actually host another workshop next year. So let me give you some motivation for this talk. So I want to basically design some sort of distance measure. I want to measure there's circle A and circle B. I want to measure. Obviously, they are topologically equivalent. Circle is circle. Right? There's no topological differences there. So I want to design a distance that basically give you distance 0. There are no topological differences. Now, then I can actually measure I have 2 circles here. The circle A is basically translated like this. Okay? Then again, the distance will be 0. Now think about this problem. can you come up with some sort of distance, geometric distance that has this property? I don't think you can find such distance. It may be difficult actually to design it. By this, you can actually train the deep learning to recognize this kind of topological variance invariance, but that may take a while. That may take. I don't think it's an easy problem even for the deep learning. However, we can design such distance quite easily through the Gaussian diagram. And another example. So in this case, the circle has been deformed to a triangle. Again, then since there are no topological differences, the distance between AB should be equal to the distance between the C and the B. Right? Again, such distance, I don't think you can find the geometric distance doing this. As far as you know, only topological distance can handle this kind of problem. And this is an example where you actually need a topology. Now, I'm going to cut the circle in this arc here like this. Okay. Obviously, they are different objects. Since I'm cutting here, the distance between A and B and the distance between C and B should be different. How different? Now this is 0. Okay. So distance can, and they are different. So that means distance between the C and B should be bigger than 0, right? Are you following our logic here? It should be bigger than 0. Now this is that. This means that this is the topology distance is not additive in a sense that if you look at the actual geometric distance, because in a usual geometric distance between A and B should be bigger than distance between C and B. Why? Because there are more set here, so that means it should be actually bigger. So our topological distance, it actually have a totally different characteristic or property compared to geometric distance. We can actually at least come up with this kind of conclusion purely based on this simple example. Now, so let's formulate this problem differently, more mathematically. We want to build a topology distance that's basically matter in some space. So this Gaussian homology is essentially solving this problem. Okay. And so the actual application. So we want to analyze the brain networks. Okay. This is a brain network we obtained from MRI scanner. Basically it's scanned at 1 mm resolution. So this particular example, in 1 mm resolution, that should be about 300,000 nodes. Okay? But the brain network with 300,000 nodes is too big. So we actually subsample it, and only displaying about 50,000 nodes. So it's a brain network with about 15,000 nodes. I want to this is actually these are two brain networks coming from two different people. I want to compare them, okay? And quantify if they are different or if they are similar, and how they are similar, how they are different, and if there is differences, I want to localize the brain regions. That's different as well. Now, then how do people analyze brain network? They usually use edge weight here. Edge weight basically is what we call connectivity. Okay? So brain region A, brain region B, we compute some sort of connectivity measure. Usually obtained, basically we mainly use a correlation, Pearson correlation. And then at each given node, given two nodes, we compare A and B and see whether these numbers are bigger than this number. That's the usual way of how brain networks has been analyzed these days. Now, as I said, there are 300,000 nodes. It's too big. So what the people usually do is that we mainly parcellate the brain into bigger regions, and usually the number of the parcellation is usually 100 to 300. Okay? So for example, hippocampus will be analyzed as a single hippocampus. So hippocampus will be a node, one node. Amygdala will be another node. For example. So by the way, if you have any question, just interrupt me and ask me question. And how we are trying to analyze brain network. This is what the brain network has been analyzed. Usually people analyze at the connection to connection. They simply compare at the edge level. But that's basically pairwise interaction. Basically. Given two nodes, I want to measure some sort of pairwise interaction between the two brain regions through some sort of the correlation measure. But such approach is very limited. However, for example, in a brain network, you can have some sort of the feedback loop, something like this. For example, they pass through a couple of the brain regions. Then how you want to quantify such kind of the loop or cycle structures? So we trying to analyze brain network not at the edge level, but trying to analyze it at the cycle level, basically. Okay? So how I view the brain network? I view it as a cell body of the onion. It's a microscope image of the onion. So this is actually a network or graph as well, where you have a bunch of cycles like this. Another example would be the honeycomb, actually. So you have a cycle here, cycle, a bunch of cycles. Okay? So this is how we view brain network, as a collection of cycles, right? Rather than collection of the connection, pairwise connections. Okay? So let me explain about the actual brain network data. So it's actually the scanner in our school. It's coming from the Weizmann Brain Imaging Laboratory in our school. So if you go, so the person will go here, head will be here, and the person will lie down like this. Okay? And go inside the scanner. It's a 3 Tesla magnet, very strong magnet actually. And then we do scan it over time, and then we see what happened inside our brain, and we can actually measure this at the 1 millimeter resolution these days. For example, the voxel position 1 and voxel position 2, you can have this kind of the fluctuation of the brain signal actually. It's very, very noisy actually. In this data, I would say there are like a 99% noise and only 1% signal. That's the usual characteristic of the brain network, brain signal actually. There are more noise than the actual signal. And, but this 1% signal can be consistently observed if you do a lot of image processing, signal processing, and then perform a static analysis. So in this talk, I'm going to show you 2 data sets. One is actually coming from our school, and it has 29 about the 300 time point. The other one is it's a human connectome project. It's a public domain data set managed by the NIH. It has 1200 time point. I think local data is actually better because HSP data, it's actually a good data set, but the problem is that it's collected from the more than 10 different schools. They try to use the same image occlusion parameters and protocols. Depending on if you use a different scanner, the image may be slightly off. I don't think that so I would say smaller sample size data from the single scanner might usually is actually far better data than the bigger sample size data from 10 different sites actually. So how big is the brain network data is actually? I'm going to construct the brain network by, for example, correlating this time series of the signal. So if you are using 1.5 Tesla magnet, you usually get the 2 millimeter to 3 millimeter resolution. For 3 Tesla, you can get 1 millimeter resolution. And in 3 millimeter resolution, you have like 25,000 voxels approximately. And that usually give basically each voxel will give you no. So basically, you have about 0.67 billion connection. That's a lot of the memory. It's about 5 gigabytes of RAM memory per subject actually. So this is example of those. And actually, this actual the connective matrix obtained from the actual human brain. Now, so from using that, you can actually do this kind of the visualization. Now, if you do 1 millimeter resolution, you would have about 300,000 voxels. How big? It's gigantic. You will have about 90 billion connections, and that requires about 700 gigabytes of RAM memory per subject. So if you try to analyze 100 subjects together, there's no way you can actually even know the single subject data into computer memory. If you have like a laptop. Anyone has a 700 gigabytes RAM memory in US desktop? Most people don't actually. So it's very challenging data. So how we do it? Well, for example, so in this kind of example, suppose if you have a 300,000 node, and then how many cycles will be approximately total number of cycles in this brain network is about this approximately. So I mean the data is so big. So what we're gonna do is that we're gonna actually do some sort of the data reduction, more like it's what we call biological data reduction. And why? I explain why it makes sense. This is actual fMRI signal, rescue fMRI signal, and we can scan it at millisecond level. But for this talk, I'm going to show it only every 30 seconds, and to show how stable the pattern, spatial pattern is. Okay? 30 seconds passed, another 30 seconds passed, another 30 seconds passed. Very stable, right? Okay. So that means even though if you look at this kind of pattern temporarily, it's highly fluctuating. That doesn't mean that it's also fluctuating over space. Over space, it's more stable. So brain's activity here will be very similar in spatial domain. So that means we can actually postulate the brain into similar functional part like this. So that's what the people do in brain imaging area. We simply postulate the brain into some functional clusters. In this case, we use the 116 brain regions, and then we simply average the time series here and get a single time series. So this is what the time series averaged within brain regions. So we have 116 time series, and per subject. So that's what we analyze. Then using that, I'm gonna basically compute the correlation between these. That will give you 116 by 116 correlation matrix. That's changing over time. And this is an example. As you can see, this correlation matrix or connectivity matrix is fairly stable. I mean, it's still fluctuating over time. Still, high correlation regions are still high correlation region. And so this gives you very stable data. By the way, this is actually the 1 meter fiber strategy. It's obtained from the diffusion tensor imaging. And we are basically projecting this connectivity matrix into this 1 meter fibers to see which region is which region. So I can immediately see this region is actually corresponding to this one. Split red cluster. So you see that how stable they are spatial. Okay. So it's what we call dynamically changing correlation matrix. Now this in the neuroimaging, we basically analyze this dynamically changing brain connectivity matrix, and it has a correlation is basically correlation matrix itself. You can think about that as a non-negative definite matrix. But if you assume the non-negative definite matrix, what happens is that you can actually introduce artificial dependency purely from the algebraic dependency. So that doesn't make much sense. So usually correlation matrix is traditionally modeled as positive definite. Okay? So we're gonna assume that the brain networks will be following some sort of positive definite correlation matrix, and then you can work out some mathematics actually. Because if you think about the collection of positive definite matrix, it's basically it forms like a manifold, which is basically a convex manifold. So you can actually, it's not a vector space actually. So positive definite is, you don't have a negative part, so you cannot form a vector space there, but at least it will form some sort of like a complex cell like this. So using this kind of the extreme values, you can work out some nice brother. I mean, that's a problem. So now our main tool to analyzing the dynamically changing correlation matrix is Gaussian homology. So if you want that, we have a MATLAB code. You can play with it. And so let me start with the introducing the filtration, what the filtration is. Filtration is like a sequence of the object or vector space that's nested. That's called the filtration in math. And well, the question is how to build how this filtration structure. So the main question is, I have this kind of data. I need to build some sort of filtration structure. that's nasty. That's really needed for the building this Gaussian homology. So people has been developing a whole bunch of the filtration, Leib's filtration, Moo's filtration. In our group's case, we call it the graph filtration. So everybody has their own favorite filtration structures, but if you use some build, I mean, you can build any kind of the nested object. It's not that hard building a nested object in this kind of the structure. The problem is how to build it in such a way that the final outcome is very consistent and robust. That's a difficult question. Okay? So you have to build the filtration structure in such a very peculiar way to come up with the consistent result. Okay? So our approach is we're going to use a filtration in such a way that it will give a monotonic feature function. Why monotone feature function? Monotone function is something like this. If you have this monotone function, data quantification is so easy. Let's think about it this way. If you have two functions, one is block-children like this. Another function is always monotonically increasing. Okay? Now, if you compare two functions, which one do you think is much easier to analyze? This function is. Okay? You can actually build a lot of mathematical structure there. So this is the approach we take it. It's purely statistical point of view because analyzing monotonic function statically is essentially a triviality. It's essentially solve the problem. And that's why we are trying to build a filtration that give you monotone feature functions. By the way, this is petty numbers. If you're using, for example, Leib's filtration will not give you monotone petty numbers. So this example, Leib's filtration is basically building by connecting all the points within some epsilon radius. So this epsilon radius, so we simply connect it. If the radius is bigger, we essentially build the triangle as well. So you have a triangle, and if the radius becomes bigger and bigger and contain the more than four nodes, 1, 2, 3, 4, then you will form a tetrahedron, solid tetrahedron basically. So as epsilon goes increases, you have a much higher dimensional simplified complexes. Okay? That's why Leib's filtration is computationally very demanding. It's not necessarily efficient filtration, even though it's a baseline methodology people widely use. It's not a good method in terms of the data as a form of data representation. It's not very efficient. It's very time slow, very time consuming. So these days, if you are working on a filtration structure, there are a lot of ways to come up with a scalable version of the filtration structures. We don't use it. We never use it. I don't like it. I don't like ellipse filtration. So why? So it's called the Gaussian diagram. What the Gaussian diagram basically measures is that whenever you increase this kind of here, the filtration value is called epsilon. When we increase the epsilon values, we take a look at how something changes. Okay? For example, this triangle is born here. Okay? Eventually, it will die at certain point. So, both value, it's a both value here, that value here. Then, this triangle, I can represent it horizontally at the both and that at this particular point. And this diagram is called Gaussian diagram. Persistent diagram. And this is what we sometimes, I mean, you can think of this as some sort of embedding. topological embedding in two-dimensional space, where the horizontal axis is the characterizing the both of the component or both of the some features, and vertically it's the depth of the features. Okay? So if you do it, the connected component is easy. You can, in a leaf filtration, you will have this kind of pattern. It's just one-dimensional pattern. However, the problem is that the high-dimensional topological features, it's a cycle. 2D would be H2 would be, you call it cycle void or something. It's like a high-dimensional portion of the cycle. That's what the H2 is. You keep going this. All these features beyond the H0, everything is just scatter point in 2 dimensions, okay? Which don't seem to have any pattern there. So it's analyzing this is very hard. That has been the biggest challenge in this area. Now, if you use our approaches, I can show you that our Gaussian diagram is always one-dimensional. That's a way to build a one-dimensional Gaussian diagram like this. And the way is simply ignoring this building triangle. Okay? Just do not actually build the triangle or do not build the tetrahedron. Simply leave it as empty space. Then we should call that as a graph filtration, because it's basically you're building a graph actually. Okay? You're not building the high dimensional simple complex. You're basically building a graph. Okay? And then with that, you can show that 0D topology is basically given by here. 1D topology is exactly here. So let me explain how to define a graph filtration. So graph filtration is basically you need to basically have a rigid complete graph. Just starting point. So you have edge weight. Now I'm going to make this as a binary graph by thresholding and assigning edge weight to be 1 or 0. And you threshold at this epsilon. So epsilon is our filtration value. Then you can actually show that this basically forms this kind of nested structure with increased edge weight. Okay? So this is comparison. So if you do leaf filtration, usually, I mean, the original definition of leaf filtration is actually usually built on top of the metric space, but it's not actually needed. You can actually still build a filtration structure, even it's not exactly a metric space. So, and you basically build like this. The difference with the graph filtration is that the graph filtration, we are only using node edges without any triangle or high dimensional simplexes. So you have this. So in the case of graph filtration, as the filtration value increases, you are actually increasing the size of this simplexes. However, in our case, we are reducing, we are deleting. So think about it. In this case, in the leaf filtration, you are actually connecting the node and making it bigger and bigger. While in our graph filtration, we always start from the biggest set, a complete weighted graph. Okay? That's the biggest set. Then you are deleting one edge at a time, sequentially. Okay? So it's kind of opposite. And if you do that simple structure, you can do a lot of things actually. So this idea of graph filtration is actually presented. I think this is actually a journal paper using topology data analysis for analysis brain network. On poster. So in this case, we actually use Betty 0. a lot of people ask me why we never look at the persistence. Persistence usually means that the persistence usually is defined by that value subtracted by the both value. That's what the persistence is in position homology. But we don't look at it. Why? Because in our case, if you're using graph filtration, it's exactly like this. Yeah, you have exactly one dimension. So either you have both value alone or that value alone. Okay? So you don't need to worry about it. You don't need to worry about the that value and both value simultaneously. You only need to worry about the both value or that value. So that zero is more than enough. In our Epiogen graph filtration, Betty numbers is the persistence. So things are very simple, and with that you can characterize a lot of things actually. So with that, for example, this is autism data set. It's a pediatric control, normal subject. ADHD is attention deficit something here. So that's showing that the pattern difference, topology pattern difference of the graph filtration. So let me give you more detailed explanation of the persistence actually. So it's basically a lifetime of the feature. So that's whenever we say zero cycle, that usually means a connected component. Now, in the graph, connected component is something that's connected. So in this case, number of connected components is 1 here, 1, 1, 2, because here, those are two disconnected components. Yeah. In this case, 3. And those are Betty numbers, Betty zero numbers. So each time we delete let's start from this graph. I'm gonna delete one edge at a time. So I'm gonna delete here sequentially. I'm gonna delete this one is deleted. Next, I'm gonna delete the 0.3. Next, I'm gonna delete this. Next, this one. Next one. Keep sequentially deleted. And each time you look at what happened, and the zero number will be always increasing. You can show it. So, and now at this point, at the point 4, this component will born here, right? But once this component is born, it will not die of the filtration. So it stay there. So both value is that value is infinity. It will die at the infinity. Okay? But you don't want to worry about the infinity. So you don't need to worry. So in the case of the zero-dimensional person, it's just you just need to worry about the both value. Okay? It's basically an infinity subject by the both value. So that's one. Now in the case of the that's right. So you only need to encode the both values when the both occurs for the connected component. So that's what our Partian diagram. So our Partian diagram is looks like this. Now I'm going to do this for the cycle. It's a loop basically. And for this loop, look at it. We're going to start from here. I'm going to delete this edge. If I delete this, currently there are 2 cycles here. But if I delete it, these 2 cycles will merge and form a 1 cycle. So there are 2 cycles here, 2. But if I delete it, it will be just 1 cycle. Okay? You keep doing it. You are basically destroying cycles. So you can show that the Betty one, which basically count the number of the loop or cycle, is actually decreasing. It's monotone. And if you ask this question, how many cycles are there to graph theory people or the computer science people, they may say that there are one cycle here, this one, and bigger cycle. However, they may say there are 3 cycles. Well, the problem is that the bigger cycle is a linear combination of these 2 smaller cycles. So A is actually cycle here. Make it more clear. So this bigger cycle will be this cycle. I mean, this is going here. So I'm going to cancel out. Then AB, if you add AB, you get this cycle C. Right? So that means one cycle is dependent. Right? So that's exactly why. That's why we have only 2 cycles, and that's the number of the better one. Okay? It's clear. It's clear. So then, now for the cycle, look at this graph. When is the both of this smaller cycle here? This one. Why? It's already there. So both value of this cycle must be minus infinity because it's already there. Right? So you stop on minus infinity. Only worry about when this cycle will disappear. For example, in this case, when this cycle will disappear, when you disconnect this part. So you only need to worry about the depth values of the cycle. So in the 1D percent, it's basically you don't need to worry about the both value, only the depth value. So our percent diagram is exactly 1D like this. Okay? Now then with that we can do, so this is example. BJ, yeah, this is BJ's visualization. So if you look at you can actually tabulate when the cycle is born. When the cycle is destroyed. So it's the timestamp of when the cycle is destroyed. It's just 4 cycle that has been destroyed here. So you can actually keep track of this in sequential manner. So you can actually index each cycle through that values. You really have to understand, if you don't have this kind of system, how are you going to index all this cycle in the graph? How are you going to do it? Systematically. I think this is the most efficient way of indexing every partial cycle in the graph. Actually. So this is actually with the BJ, basically this is what we have been working on for the last 1 year. That's what we did actually. So this example, given a graph coming from the correlation matrix of the brain network, you can actually compute the bet0 and bet1. It's actually come from real data. So that's why it's kind of 직접 pattern. It's not going to be that smooth. But bet1 looks very smooth. Can you see the difference here? Because there are 116 nodes, the bet0 is somewhere here, but total number of cycle here, bet1 is approximately, so approximately this number. And can you guess why it's so smooth? Why it's not that smooth? Well, look at the number. It's a big number. Let's keep changing. So each time cycle is destroyed, but when you delete one edges, how many cycles do you think you can destroy? Maximum 1 cycle. If you delete one edge, you may not destroy cycle, but there's no way you can destroy 2 cycles if you delete one edge. It's always maximum 1 cycle. It's only changing 1 cycle at a time. So filtration value, they're like So if you go through this all the way, there should be about 16,000 steps to delete. On the other hand, number of connected components is basically 116 here. So that means there are more sparsely changing. If you delete one edges, the total number of the component that's increasing will be maximum 1. So that's why it's very zigzag pattern, while the petty one is always. You can actually look at the actual genetic differences in that actually. For example, it's identical twin, non-identical twin, to see how they are similar. If you look at it, the zero cycle is a connective component, one cycle is a loop. You can actually get this kind of very nice pattern. Now, you're not going to get this kind of nice pattern if you're using linear filtration. It's going to be like a zigzag pattern. Crazily fluctuating zigzag pattern. That's why we're using the described filtration over leaf filtration to make it things more interpretable. So based on this, we're going to show you something. We're going to decompose graph into edges that destroy cycle and edges that actually create a component. Okay? Now we can show that the edge set, total edge set, is basically partition this way. We show that where H1 is null actually. It's basically a partition. So total number of the edges that actually destroy cycle is this. Total number of the node is the total number of edges that actually create a component is this. But this is a petty. I would say it's a worker. Maximum petty one. Maximum petty zero. So if you add this, you get the total number of edges. That's your total number of edges. That basically, I'm going to now, if you combine all the edges that create a component, what you get? Actually, you get a maximum span tree. It looks. Then, any edges that doesn't form a maximum span tree, what does that? It's basically this part, H1. Those are the edges left out of maximum spanning tree. So, given a graph, maximum spanning tree is related to the zero-dimensional homology, while non-maximum trees will be related to the one-dimensional homology. So that's what we call it the broad entity composition. We actually published it. This is actually impressive. Actually, so I think it should be published in a month or so. So now this computation identify all and that and all this decomposition is actually n node n time. Why? What it takes to construct a maximum spanning to a minimum spanning tree? Well, that's you only need to sort the edge weight. Okay? Sorting is n log n time. So that's why. Then this is quite interesting. So with this, in the case of the connected component, you can actually basically build some sort of vector space representation of connected component by taking each node as the basis. So if you build the total number of nodes, you can build a vector, something like this, where you're going to assign value 1 at the ith node. Then using this vector representation, you can do something like spectral representation using like graph Laplacian. Now then, how you do that with the cycle? Well, for a cycle, why don't we actually build the pieces? One cycle or two. The way you're gonna do it, you're gonna use this maximum spanning tree. So you start with the maximum spanning tree. Okay. Then on this maximum spanning tree, you have an edges that actually disrupt the cycle. You can, as I just said, we can actually index it, this cycle, to this depth values. So sequentially add one depth values to the maximum spanning tree. Okay? Sequentially. Then this is basis, one base, second basis. Okay? So there are only two bases because there are like Betty 2, the Betty 1, the maximum Betty 1 is here. In this case, is 2. So you should get only two bases. Okay? So this is one base. This is our base. This is one thing is our base. So this is our base. That's it. That's how you build vector space representation collection cycle. Okay? Then you can do something called, you can connect this whole story into Hose Laplacian, and well, you can read the paper how we actually connected this to Hose Laplacian. And now we're gonna use your Wasserstein distance here. Now, Wasserstein distance is widely used. So in the usual Lipschitz filtration, you have a bunch of the scalar point, you have a bunch of scalar point. Okay? As I said, this is topology embedding, topology embedding. Now, I want to compare the distance between these two. And most people these days use Wasserstein distance. There are a few other distances like a Bartlett distance and Komorov-Hausdorff distance, but I would say our group mainly use Wasserstein distance. I think also other groups also use Wasserstein is more widely used. Actually, what we call optimal transport. When the measure, the whole set is just one, you got a probability measure. So that's in that case, this special case of optimal transport is called Wasserstein distance. Now, I would say if you look at the literature, especially in data science, I don't think most people are not going to use a more general measure. They're going to restrict the measure to be one because they think so much simpler and simply use Wasserstein distance. So use of Wasserstein distance is more widely used, especially in the deep learning, all this machine learning literature compared to the optimal transport. And if you want to do Wasserstein distance between graph representation we are using, you can just download the code there. And this is the usual definition of Wasserstein distance. So given a two random variable, you basically define distance to be infimum of the expectation of this. This is two Wasserstein. Obviously, you can build the R Wasserstein with a different norm here, but I prefer two Wasserstein distance because I think it's much easier. Why? It's a quadratic term. You can actually split it into h, x. If this is a scalar value, you can just do xy plus y square. Take expectation. That should give you expectation of x square minus 2 expectation of xy plus expectation of y square, right? And then you only need to know the moment, the second moment of the random variable. That's all you need to compute the Wasserstein distance. So two Wasserstein distance is most widely used distance compared to other distances. So I like the simple things. Yeah. When I got older, I prefer much simpler things. So in our case, Gaussian diagram is just sorry. This is general case. In the general case, Gaussian diagram is a scalar point in the two-dimensional space. Okay? And then using that, you can build the empirical distribution where this is Dirac delta. Okay? Dirac delta is like a spike like this. everywhere is 0. Just 1 point, it's infinity. So this is d of delta x. Yeah, sorry, x minus. Then, if you integrate this function, you get value 1. So you can think about this as like a property. Okay? I mean, there's no such thing as Dirac delta in reality, but it's more like abstraction, algebraic, and you can actually write this way. So from scalar point, this scalar point is translated into this kind of property measure. Then with that, you can work out the detail. Then you can get this simple expression where this is bijection. It's basically permutation. The basically permutation of this label, how this point is mapped to which point, and it should be a bijection. And so you can work out a lot of mathematics here. So there's so many. I mean, there are crazy number of paper out there on Wasserstein distance doing this stuff, and this bijection or permutation. Now, this is basically you have to compute the permutation of this point to see and compute this distance. See which one is smaller. Okay? Which permutation is small. Now, how many permutations are there? It's Q, so it's Q factorial. Okay? Now, it's basically minimizing in the space of the Q factorial. How big this Q factorial is, you can work out some math, Stirling's formula. You can show that it's exponentially big number. It's a gigantic number. If you have more than 100 factorial, you cannot compute it because digits are so many. There are so many digits and 100 factorial. So if you have more than 100 scalar points, it's usually computation in all this permutation spaces beyond our computing capacity. Unless I don't know, there's the probably in the future, computing power is just so much. You may be able to compute every possible permutation, but not now. Right? You cannot compute every possible permutation. Right? So then, how are you gonna do the optimization in this astronomical big number? Right? What's the quickest way to do the permutation in the space of the permutation? Well, the quickest way is usually gradient descent. Yeah? You such gradient descent, basically do the differentiation and such gradient descent in a big space. Probably the gradient descent is that you don't know whether you are hitting the global minimum or the local minimum. Right? That's the usual way. Okay? Now, in the case of this, we have no solution. The Hungarian that does computation cubic runtime. You don't have to compute every possible Q factorial. You can do it in a cubic runtime. Obviously, that's amazing time saving. However, that's not good enough. If we have like a million data point, try to match them. You cannot compute it. It's too slow. How slow this algorithm is? And cubic runtime is not a scalable. Any algorithm that's cubic runtime is not scalable. Why? The cubic runtime is equivalent to the matrix inversion. If you try to invert the 10,000 by 10,000 matrix in computer, you will know the pain, how slow the matrix inversion is. It's that slow, right? So it's not scalable algorithm. So there are people, for this one, so the coupled algorithm, most popular approach is the sliced Wasserstein distance. So sliced So what they do is that this problem they project it into one dimension and try to come up with Wasserstein. try to compute the Wasserstein distance, not in 2 dimensions, but in 1 dimension. In 1 dimensional case, there's no mathematical solution, analytic solution. So you can compute it by hand. That's the usual approaches. It's the most powerful approaches. We're gonna take the same approaches. When we worked out this problem for last 2 years, we actually didn't know what Wasserstein distance is actually. Someone actually pointed out that our approach seems to be very similar to the Wasserstein. That's how we got to know it. Kinda, I would say, we kinda reinvented the wheel, I guess. That's what we did actually. So our approach is Q, log Q. Slice the Wasserstein distance basically does projection into 1 dimension. They do it K times. So their approach is actually slow. So actually, it's the same algorithm, but you have to do K number projection sequentially. So it's, they need this K. And so let's compute the Wasserstein distance. In the case, I said that our scatter point is just the Gaussian diagram. I'm going to compute the Wasserstein between the 2 Gaussian diagrams. But in our case, our Gaussian diagram is 1D or 0D. Okay? So this case. So it's basically 1 dimension. So if you work out the detail, you can show that the Wasserstein distance, 1-dimensional homology. This case is just matching both values in that's basically MST, maximum spanning tree. Okay? And so whenever you have a maximum spanning tree like this, with another maximum spanning tree, you basically figure out how to match the edges to which edges in the maximum spanning tree. Okay? That's what we do here. Then this part, you basically match non-maximum spanning tree part to non-maximum spanning tree part. Now this has a exact mathematical solution. It's the solution is you simply sort the edge weight, simply match smallest number to smallest number. Right? That's the solution. Yeah, it's kind of triviality. So if you work out, and, so what you do is that you simply match them sequentially. Okay? Sequentially match them, and that's how we speed up the computation. Comparing Wasserstein distance. And using this, we can do something more than that because we exactly know which edges are matching to which edges using Wasserstein distance. In our case, we know. If you're using other distance, the traditional Wasserstein distance, Gaussian diagram to Gaussian diagram, you don't know which edges are connected to which edges. But our Wasserstein distance, because it's a 1-dimensional projection, we exactly know which edges will be matched to which edges. So we can use it to the graph match. Okay? So I want to match this graph to this graph. Okay? So this is my graph here, and this graph. Obviously, topology is very different. I can still do the graph match. And this kind of how are we going to match? Well, I'm going to do this both that decomposition. So these are the MST, red is MST. So I'm going to match this part separately because they're basically it's a partition. So you don't need to worry about each other. Okay? So I'm going to match the MST to MST and the non-MST to non-MST. Okay? And you simply sort them and match it. So here, the large to large. Okay? Why you want to match a large data value to the large data value first? Well, that's a longer distance usually considered signal Gaussian homology. Okay? Because it stays longer with the filtration. So longer positions you should match always first. So match it. Then what about this? This one is unmatched. Okay? Unmatched value should be projected and match that way. Because that's the only thing. That's the only way. Now, obviously, it doesn't give you the bijection. Eh? This bijection issue, you see from this formula, basically assume that you are matching equal number of edge weight to the equal number of edge weight. Yeah? So this is, in this case, we have to do a data augmentation. Yeah? So this is augmentation. Then do the same thing. And that's right. Now, using this, we can do the graph matching. And we can do the averaging. So I want to compute the average of these 5 graphs. I want to average them and divide it by 5. Okay? My claim is that you get this. Okay? Same thing. If you average this, you get this. Because we know exactly which edge will be matched which edge. I know the correspondence to this correspondence. I can do the average. So that's how we do. Using this tool, we can also do the regression. Now, in this case, I'm going to match the rest of the fMRI to DTI, it's a different imaging modality. And so the rest of the fMRI is captured by the very dense cycle, the narrow cycle. In the structural brain network, it's basically 1 meter fibers. 5 meter fiber, if you look at 5 meter fibers, there's almost no cycle. And so, I mean, there are some cycles that are brain region that actually kind of the brain to the back of brain. You don't want to do that. So you need some sort of geometric constraint. So this basically from geometry. It's a previous neuron. I basically use a previous neuron, cell2 neuron basically. So with some amount of its parameter where we basically control amount of topology, and this is Wasserstein. As a topological signal. So then you can actually match graph two different networks coming from the different scanners, and optimization is slightly more complex. You have to do the gradient design in this particular case. So this is estimated network. So I'm gonna load this. Okay, this can be viewed as some sort of superimposition of these two parameters. So what you do is you do the gradient. You basically take the gradient on the edge weight, and you can actually show that the edge weight, the gradient distance is basically given by us also. What's going on. And you can work out the detail and show that this kind of topology. So gradient descent in this example, you can basically do the sorting the both value and that value and match them. So you don't actually need to compute the gradient. You just sort them and match them. That's gradient in this case. So again, the runtime for the gradient descent algorithm is n log n. It's very fast. This is done by Tan, and he reported that the five iteration model, almost all the cases, most of brain network 5 iteration will actually. So you do a gradient descent 5 times, and that will basically give you convergent result. Using that, we can actually compute what we call heritability index. It's a measure of the amount of the genetic heritability. And so if you're using traditional correlation measures, you get this kind of very sparse signal. But if you do the topology learning, you can actually boost the signal, get the more brain regions. That seems to be hereditary. So now we can actually do something more than this. What you do is just a visualization, nothing more, but that's not good enough. We can actually put some sort of statistical significance on our claim or hypothesis. So we can do the what we call topology inference. And what the topology inference usually does is that given some specific hypothesis, we are putting some strength of this claim, our claim, on a scale of the probability from 0 and 1. And so in the case, we have a bunch of the brain networks. We have a bunch of brain network, M, and another brain network. Why don't we use something like this? Why don't we just partition it somewhere? It's a group 1, group 2. So you have a collection, one network, collection one network, I have a collection with a bunch of network, collection of another bunch of network. I want to know if they are different. This is traditional string inference problem. And the way we approach this problem is that we actually get the idea from the clustering. In the usual clustering, you actually need to compute the between group distances or within group distances. And you need to maximize one of them. So this is what we call the between group distances, and this is what we call the within group distances. Then you can form this statistic, ratio stat, and to see how, and so then you basically compute this number. Well, this is our observation. We already know the partition. For example, this can be a male and this can be a female. I want to know, given a male brain network and female brain network, I want to know how far they are different. Then you compute this, then that's our observation, observed statistic. Now, we need to compare this observed statistic against what we call null distribution. Null distribution here in the case would be under the null assumption, there's no difference. That means we're going to swap them, male and female, all mixed up. Then obviously, if you mix them and randomly split, you're not going to see that. You should not detect any signal. So that could be the what we call reference distribution or null distribution. So our observation, this is our observation. We're going to compare the distribution of the distance under the permutation of every possible male and female. That will give you like this. Okay. So that's how we do the inferences to see whether how far this our observed test statistic is from the null distribution. More away from the null distribution, we have a much stronger signal. The problem with this approach is it's called the permutation test. I just explain what the permutation test is. The problem with this traditional permutation test is, I just said, permutation test is n factorial. It's too many. There are too many, and so how we speed up this permutation test? Well, again, one idea is quite simple. Instead of permuting all the group labels, male and female, randomly permuted, we're not going to do it. We only permit one subject, one female or male, and swap it. If this size is female, male, I'm going to move one female to the right hand side. I'm going to move one male to the left hand side. Swapping. It's called transposition. Under this transposition, it's basically we are doing a random walk. We are doing a random walk in the space of all possible permutation and trying to figure out how this test is changing over the one random walk. then using that idea, we can do the computation. And this one is really best traditional computation test. And then you can do can compute this statistical quite easily. For example, in our data set, if you can do the permutation, about 50 million permutation, and compute all the test statistics and do the significance in about 40 minutes. So well, this I'm going to skip it. It's too tedious. It's just validation to see if the method works or not. So we also did some validation based on the ground truth. So we used the modified version of the Erdos-Laney random graph model. It's a well-known model where it's basically you're putting a Bernoulli distribution on the connection of the edges. Now that's too simplistic. Original Erdos-Laney model is too simplistic. So you can split your graph into two forms like this. You want to assign the property P within a module and 1 minus P between the module in such a way that you can have this kind of modular structure. So in this case, it's 2 modules, and in this case, we have 3 modules here. Okay. Then, obviously, and they are topologically different. Because it's a graph structure with 2 modules, this graph structure with 3 modules. Obviously, they should be viewed as a topologically different. And we tested whether our method can detect the recognize these 2 different graphs as different or not. And we compare this with the traditional graph matching. And the bunch of the method are there. Most of the graph matching algorithm actually because Hungarian runtime. So most of graph matching algorithm, they're best ones actually, but they're still cubing runtime. Why? This is our method. Our method is essentially flat. It's you have to watch it carefully. This logarithmic scale. Still, it's essentially flat because it's n log n actually. So, and computation, I don't think any existing graph matching algorithm computer science can match our method in terms of the runtime. Nothing beats our method in terms of runtime. Then you may ask, well, okay, you guys method are best, but what about the performance? Well, I claim performance is also very good. So we actually compared those kind of the random graph model of project to see whether method can detect signal or not. And in this case, it's a small, it's called the p-value. P-value is the strength of our claim. Smaller p-value usually means better. It's called p-value. And in this, if you look at it, it's actually, we are coincidentally beating almost all the methods. So it's quite good. Cool. So I don't think, I haven't seen anyone, I give a lot of presentation on this topic. I haven't seen anyone, I haven't seen, no one actually came to me saying that our method beat your method. No one yet. I've been presenting this lecture for the last 2 years, or especially on this slide. No one came to me saying that their method beat our method. So I think our method might be the ultimate weapon in the graph matching. We are not even using deep learning. We are not even doing deep learning. It's still doing quite well. So using this tool, we're going to show you how to do the clustering. Well, that's quite how you do the clustering. Well, it's a Wasserstein distance, zero D homology, and this is Wasserstein distance, 1D homology. Okay? So what the graph graph clustering does. I want to cluster this graph into this form. Okay. So that where cluster CI is the collection of this graph. Okay. Something like this. Okay. So, and something like that. So sorry, I think I realize that the notation is off here. This equal notation. Given this, sorry. N graph, I want to partition this graph into K, such that there's no overlap between two clusters. That's what the clustering is. Actually. It's basically clustering is basically partitioning of the graph. That's what the cluster does. Then again, we're gonna use the topology distance here. It's basically this topology. Okay, so there's also type. I mean, you can use only this part or this part. If you're only using this part, that means you are clustering the brain network purely based on a cycle. While if you're using only this part, you're clustering your network purely based on connected component. Okay? We want to combine that together to have some more common measure of the topology similarity or dissimilarity. Then if you use that, and you can form some sort of the algorithm is essentially K-means clustering. You can actually use K-means clustering with different you don't want to read it through, it's too tedious, but you can. I can guarantee that it converges quite quickly, and I mean, this is more interesting stuff. We actually tested. You can see that this simulation, there are two cycles, and this cycle is just rotation. Okay? So obviously, each group, all these groups are topology equivalent. It's just a rotation. Okay? So they should be topology equivalent. So if you test for the topology difference between all these groups, you should not detect any topology difference. That means if I combine this data and try to cluster, your clustering should not perform well. I'm serious here. This is a simulation setting designed to treat existing clustering. All the clustering algorithm will fail this task. Okay? If you're using some deep learning based approach, you have to train the deep learning such that this one should be treated as identical. But unless you do that, I don't think using the unsupervised learning cases, I don't think unsupervised, no, there's no known cluster algorithm that can handle this problem. Yeah, other than this topological distance project. So this is setting to So we actually did a clustering. K-means clustering always is perfect, almost perfect clustering. Hierarchical clustering also does perfect. Okay? Why? They view this as a different. So they partition this differently. So you get a perfect 100% clustering performance. Okay? But if you do Wasserstein based clustering, performance is miserable. It's only 50%. They are not clustering well, but that's the actual answer. You should not cluster well because all these are identical, topology identical. So you should not cluster them at all. So this is the answer. And I will say this is a world first person. If you compare this number purely, you said that there are 50% more burst types in the existing methodology compared to our topology cluster methodology. So you should not use a traditional K-means clustering with a Euclidean distance to do this kind of topology clustering task. You will learn more. And so now this actually come up with the issue. In this case, there's no signal because all the groups are identical, topology identical. Then you may ask whether topology identical signal can be viewed as signal. Yes, it is signal. Signal is you have to understand what is signal and what is noise. It's how you define what to be signal and what to be noise. My signal can be noise to you. Your noise can be signal to me. So how you define it? So in this paper, actually, that's how approach we actually took it. And so this is epilepsy signal, epilepsy EEG, and this is epilepsy seizure here. Seizure study right here. Right? So we are given this data. We basically need to figure out the identify channel where the seizure happened. Oh, this is a hard problem, really hard problem. Purely based on signal without any prior information. And this, by the way, this is diffusion. Diffusion smoke. You basically do the diffusion and the smoke to it, which is very equivalent to Gaussian corner smoking, and you get this kind of the base smoke pattern here. Now, if you use existing method for the signal detection in the EEG, every channel will show the differences between. They will say that these two signals here and these two signals are different because that's what the seizure happened. And so most methods cannot identify which channel is the source of the seizure. And we actually, when we apply our topology approaches, what we observe is that there's a topology. Basically, what we observe is that in this particular C3, we actually able to detect the topology differences. We are able to detect topology differences, except the 3 channels. And 3 channels here, it looks like there are signal differences. However, if you actually do the topology here would be identical to topology here. Topology here would be identical to topology here. So we thought, oh, it may be possible that some topology signal is actually carrying over before seizure to all the way to the after the seizure. So we identify this as the T3, and obviously there's symmetry. T4, T5 here would be the original seizure. Seizure actually starts somewhere. So I think this makes some sense actually. So that's why. So if you see no topological differences, that also can be viewed as signal. Now, this is a case where we actually design it in such a way that it will detect the topology differences. I simply cut just small part here. This is kind of test the existing method will fail quite miserably. So in this case, chemistry clustering doing okay, but it's only 83%. Hierarchy clustering usually do very well. Actually, hierarchy clustering usually very well, 100%. But the Wasserstein distance actually quite well. Then you may say, oh, well, hierarchy clustering seems to be doing better than the Wasserstein graph clustering. So why we need a topological clustering? Well, we still need a topological clustering to protect against all these false positives. Hierarchy clustering will not do well if there's no signal. It still detects the signal, it's as a signal. So that means in overall, this topology clustering actually perform best. So that's what. Now using that, since we had this understanding of what the topology clustering does, we apply this to this dynamically changing collision. And if you do chemistry clustering, you get this is output. You can actually get 3 clusters. Eventually, if you apply this, you get 3 clusters, 3 states. Just state here, and the difference between chemistry clustering result and Wasserstein clustering is that Wasserstein clustering seems to give you less fluctuation, more stable pattern over time. And but again, K-means clustering, you don't want to rely on the K-means clustering because it will it will produce some more burst particles. So those I would say are most like the burst particles. Then we actually did how do we know actually we have 3 clusters? And see whether this ratio is suddenly changing or not. And it's what we call elbow methodology. And this is identified as the most rapid changes. And that's usually considers the number of clusters. And in both cases, K-means clustering and Wasserstein clustering identified the number 3 as the number of cluster or stage data. However, if you look at the ratio, that's within versus the between. It's basically measures the variability, how stable our result is. It's 6 times smaller. That means our method is performing 6 times better than the K-means clustering. And so this is our estimated state. And this is based on a K-means clustering traditional methodology. Now, if you look at the K-means clustering approach, the output seems to be somewhat random. State 1, state 2, state 3, the pattern seems to be very random. But if you look at the Wasserstein clustering, you can see a symmetric pattern, very symmetric. Everything seems to be symmetrically left and right. You're not going to see observed here. Now, if you do this kind of clustering, I didn't put any model there. I didn't say that this kind of I never put any kind of the left and right symmetric constraint in the actual algorithm, but still purely data-driven approach, it will detect this kind of the lateral symmetry. I don't think that can possibly happen random chance or wrong. So I think Wasserstein distance is really identifying true state spaces in the brain network. You can only get this kind of the symmetry if you plus actual symmetric constraint in your state space model. But we didn't put it. It's purely based on clustering based data-driven clustering state space model. Now, if you do it, we also compute the heredity index. Now, if you look at heredity index, it's more. You can see that this state 1 heredity index is basically it's an amount of heredity you actually inherit from your parents. This is a left and right, right. It's a love connection here. So the state 1 actually characterizes the heredity of the left to right hemisphere, while the state 2 is basically the front and back. And quite nice result, right? And state 3 would be west. So RST, I think that this is the I mean, this is still under review. I don't know whether I'll publish this, but I'm thinking about withdrawing a paper to submit to better journal actually. After doing some more detailed research. Anyway, so I'm going to end my lecture here. By the way, you can actually put some sort of Markov chain on this space model and see what happened. Anyway, that's something else. So this is our school actually. It was Wisconsin-Madison and United States. Our office is actually located somewhere here. I don't think you can see this, but if you want to work with us, actually, well, you can come as a postdoc or graduate student, whatever. Anyway, thank you for listening to my whining tones.
Dear Professor Wang, thank you for your detailed, long, and interesting speech. Let's all thank Professor. And yeah, I'm sure someone would be interested in postdoc or graduate research participation. Okay, dear friends, do you have any questions? If no one has an immediate one, I have two left. Actually, Moo, you have been answering my questions during the talk within the process, but I have a couple left. And the first one is, I'm sorry if I missed that. You've been discussing the benefits of graph filtration over RIPS filtration, and I was a bit suspicious if we have some information loss with graph filtration.
For this one, we don't have it. This graph filtration will not introduce any loss. So if you have graph filtration, get something, you can actually back project your data to the edge level, because we already know pattern that decomposition, exactly know how to match edge to edge. So we can actually back project our topology signal to the original the network space. I think that's the additional advantage compared to RIPS filtration, which you cannot back project the signal to the original space.
And while we are on this slide, you said you've noticed kind of symmetrical things. Have you tried some machine learning algorithms looking for hidden Markov models?
That's actually widely used. Actually, there's one group actually use a hidden Markov model. That's actually that's something I didn't look at. We only look at the K-means clustering because there's a lot of space modeling on this kind of the data set. About 70% of all the publication use K-means clustering. So it's considered a baseline in brain imaging area. There are a few papers, one or two, that use hidden Markov model, but I actually didn't possibly feature.
Okay, probably someone would be interested to experiment that.
But I don't think a hidden Markov model may able to. I don't know. My guess is I don't think you can detect this pattern.
It might depend. I think you should check it, but who knows?
I mean, if you put some inner model, inner model concept, you may have this kind of pattern, but our approach doesn't use any kind of model assumption, purely using the distance measure. That's what we did. So I would say it's essentially minimal assumption, data-driven approach that detect this kind of pattern.
Yeah,
I think that might be the selling point.
Yeah, sure. I was very impressed by the effectiveness of your algorithms. That's just state of art. And well, thank you very much for your very beautiful, inspiring talk. I was very impressed, and I have something to think of.
Okay.
Thank you, Professor.
Okay.
So, last but not least, we have Alexei Ossadtchi today within 5 minutes. Alexei, great to see you.
Hi, Konstantin. Yeah, thanks for inviting. Yeah, hi.
Yeah, thank you for participating. Let's check if your presentation works.
Yeah, let me try and share the screen. Can you see it?
Yes, and I see that on YouTube everything's all right, I guess.
Okay, cool.
So
we'll
we
start
have
in
a couple
5 minutes.
minutes left.
Couple of minutes.
Yeah, about something about that.
Yeah. Okay, I'll grab a cup of water and come back.
Yeah, me too. дорогие друзья. Так приятно видеть, что у вас еще столько осталось с нами на трансляции. Это очень-очень приятно. Так, just a second. I think I have, yeah, now I have my headphones working. Alexei, are you here?
Yes, I'm here. Yeah, can you hear me?
Yeah, everything's perfect.
Okay, great.
So let's get it started.
All right. So yeah, thank you very much for inviting me. And yeah, I will be talking about some relatively simple methods which are basically rooted in linear algebra and some signal processing for analysis of functional networks from magnetoencephalographic data. And I will show you how it can be done and why it should be done in health and in disease, for disease patients, primarily for the epilepsy patients. Okay, so this is a joint work, of course, with my PhD students who are listed here after my name, and I highlighted myself because I'm a presenter, not because of any other reason. And this work has been supported by several institutions where I have laboratories. And also, in particular, I would like to acknowledge Ideas Center that is supporting Daria Kleeva, who is one of the co-authors of this talk and who is working on the global optimization methods for finding the networks. Okay, so I will talk about this. All right, so I direct a center at High School of Economics. I direct a center for bioelectric interfaces, and we do quite a bit of work there. And one of the topic is actually related to analysis of magnetoencephalographic data, and that's what I'm going to be talking today about. Okay, so we are up to quite a long journey, and I hope you'll stay with me, but I also want you to ask me questions, interrupt any time. This will make the presentation more lively and more I don't know, more easier to understand, I'd say. Okay, so we will first talk about phase-like invariant estimation of functional networks from non-invasively observed data. Then we'll look at how we can look at the networks for patients with epilepsy. It will be a different method based on the hidden Markov model approach, and then that is going to be primarily about distant propagation or over long distances, and then we will talk about local propagation and cortical traveling waves, again an application to analysis of epilepsy patient data, okay, and localizing the primary seizure onset zone with the aim to subsequently remove it during the surgery. Okay, so I will go a little bit about the physiological aspects of the signals that we are registering. So we primarily with MEG, magnetoencephalography, or electroencephalography, and with any other invasive methods that we measure the electrical activity, well, maybe it's not for a single spike or single neuron measurements, but if we talk about local field potentials, then that's primarily the disturbance of the field outside the neuron, the pyramidal neurons, or a big number of such neurons who receive input to the dendrite, and they have so-called postsynaptic potential and postsynaptic currents propagating down these dendrite. Okay, and then if we have many of such neurons, like we have it, for example, in layer 5 of cortex, if you have many such neurons with long dendrites and they receive more or less synchronous input, then this bunch of dendrites can be approximated like a current tube, right? It can be approximated by what we call current dipole, and this current dipole we will call a primary source. Okay, this is a primary source. You can think of it as a battery, for instance, which you submerged into a bucket of water, right? And then the battery starting to discharge and starting to produce so-called secondary currents. Okay, the secondary currents, they reach the skull from inside, and then they go through the skull, and skull is very poorly conducting substance, and then they make it to the scalp, and on the scalp we have EEG electrodes located, and just around the scalp we have we have magnetoencephalographic sensors of magnetic fields located, and these magnetic field sensors are more sensitive I will not go into the details, but I can answer questions if you have specific questions about these issues. But the magnetic field outside the head is not so much sensitive, a radial component of this field, not so much sensitive to the secondary currents, but to primary currents, which makes it easier to use for solving the inverse problem to find the sources back on the brain based on the analysis of the magnetic field just around the head, okay, as compared to EEG, which depends on the secondary currents, and the secondary currents, they flow, make very complicated pattern, and this pattern depends very much on the conductivity properties of different brain tissues, and nobody knows them, okay? So, and they change, like you drink a lot of water, you get more conductive tissues, you dry out, you get less conductive tissues, and etc. You have some medication, whatever. So therefore, MEG is a better tool for measuring brain activity, especially if you are interested in solving the inverse problem and finding the sources back on the cortex, okay, back in the head. Okay, so, and with MEG or with most of the other methods, actually, except for the single neuron activity recordings, we register usually the activity of many neurons. Okay, here each line corresponds to a neuron, each dot corresponds to, let's say, some action potential of this neuron that it propagates towards the dendrite of that population that I mentioned to you, right? And then you see these bursts of activity, and if you look at them from far away, you basically have some periodic pattern, which we will call oscillations, and we will be primarily looking at these oscillations when we are working with non-invasive data, okay? So, and these oscillations, they can be nicely classified into different bands. Most ubiquitous and most well-known is alpha band, okay, oscillation, which occurs, well, it occurs at different locations in the brain, and it hallmarks, and if it occurs in the visual system, it hallmarks the switching off this visual system from the external input, okay, if you close your eyes, you'll get your alpha rhythm increasing, like here in alpha burst, right? And you may even have some interesting coupling which occurs during these periods, okay? Or the coupling which occurs when this rhythm disappears because the sources that were generating this rhythm decided to couple to some other sources and to exchange the information, okay? And of course, there are other rhythms like more slower rhythms, delta and theta rhythm, and beta rhythm, which is associated with a lot of motor activity, and the increased amount of this rhythm actually slows you down. The desynchronization, when you have the amplitude of the rhythm diminished, then you get a better reaction time, and etc. So it controls your excitation in the motor system. Again, it's one of the functional, of course. Okay, so but the brain, as we all know, right, that it contains it consists of many brain regions, right, which are anatomically specific, have very anatomically specific types of neurons, right, and you can even you can parcellate the brain based on simply anatomical properties, based on how the tissue looks, right. But it turned out that these brain regions also assume different low-level functions, which requires integration of their processing results in order to support our behavior, okay? And this integration happens through so-called functional coupling, right? And it's mediated by the Cognitome, by the structural Cognitome, by those white matter fibers that connect different brain regions, and by relatively complicated rules of how neurons couple to each other, how they exchange information, how they signal that this information is intended for this population, but not for that population, and how they synchronize in order to establish this exchange of information, okay? So there is this hypothesis that has been pushed forward quite a long time ago by Max Price, by Richard Price, sorry, where the communication via synchronization. So the idea here is that you have, let's say you have three populations, and they go through excitation and inhibition stages, okay? So when it goes up, it's an excitation stage. When it goes down, it's an inhibition stage, okay? And so now these two populations do exchange information because the spikes in this population, they appear during the excitation stage of that green, or whatever, how you call it, greenish population, right? And at the same time, this greenish population is not communicating to the black one because these spikes occur during the negative phase of, well, this is all very cartoon-like, right? But during the negative phase, during the absence of excitation, during the inhibitory period, and therefore this population is not perceiving this information communicated from this greenish population to the orange to the red one, right? So, and of course, this situation can change, and it actually changes transiently. And then at some point, this population may start talking, the green one may start talking to the black one. But this will all be hallmarked if we look at, you know, there are different types of synchrony, right? We can look at, for instance, if low frequency in one oscillate in one region, phase of this low frequency, right, is magnitude of activity in the high frequency band, for instance, right? Or we can look at the correlation of envelopes of this activity, right? Or the type of synchrony I will be talking today about is so-called within frequency coupling, is when we are talking and looking at the activity of population within some specific frequency band, okay? And the communication, the fact that two brain regions communicate to each other is hallmarked by the presence of constant phase lag. Here it's depicted the two population activity, the red one and the blue one. And this is the period where we observe more or less the same phase lag. And in this picture, they show the nearly zero phase lag between the activity, but it doesn't have to be zero, right? It can be some non-negative, whatever phase lag, but it has to be constant over some period of time. And this period of time has to be greater than something that you could observe by chance when you're looking at this kind of measurements or kind of signals, right? So, and then once you have this constancy of phase difference, then you can declare that these two populations are talking to each other, okay? And based on this, you can then declare that those are the nodes of the specific network, okay? All right, so to measure the coupling, if we talk about within frequency coupling, we imagine now, for now, we imagine that we have direct access to the activity of populations. Okay, so we are not using MEG, or we are directly measuring the activity of some population. Okay, some population, the green, the blue one, and the red one. Okay, then let's say we are interested in activity in coupling in the alpha band. So we perform bandpass filtering of our signals. So removing all frequency components except for the narrow band around 10 Hertz from 8 to 12, for instance. And then we can look at these two signals and compute the phase of each of this signal and plot this kind of diagram that you have already seen in the previous plot, right? And then they can do statistical analysis and establish the fact that this duration of such low variance of the phase difference is unlikely to happen under the null hypothesis when there were no communication between the brain regions, right? And then you declare that this is the statistically significant event, and because it's phase synchronicity and based on this hypothesis about communication through coherence, you declare that these two populations talk to each other. Okay, so well, a little bit like expressions for coherence. When we talk about coherence, we usually talk about the analytic signal, right? An analytic signal had the envelope, the magnitude, how it changes, and the phase and the oscillatory component. And you can think that you can represent the signal as a vector rotating in the 2D complex plane. And then another oscillation is just simply another vector. And then at some point, if at some point these vectors go along without crossing each other and stay with the same distance one with respect to another, then that hallmarks the period of phase synchrony. And you can measure it, for instance, if you simply take the product of the two, right? This complex conjugate, right? And then it has to be S here, S1 here, and S2 here, of course. And then that basically the magnitude product, and then the exponent of the phase difference. And if this phase difference is constant over some long period, over some extended period of time, then you get these vectors that point more or less in the same direction, and then you average them, or take the operation of expectation, then you take the magnitude of which you get relatively large vector, as opposed to the situation where the phase difference is always different, and these vectors point, the difference vectors point in different directions, that if you average them, you get a vector here, and this vector length is small, speaking of a lack of synchrony in this kind of scenario. Okay, so this is so-called phase locking value, PLV statistics, which is which will be PLV if you didn't take the amplitude into account, right? But in fact, if you look at, if we can show that if we are talking about the circular Gaussian processes, then the PLV and coherence, which is the expression, the expression of coherence given here, are basically informationally identical, and you can recalculate one from another. Okay, so now. Most, like the majority of the methods for measuring within frequency synchrony is based on exploiting different versions of coherence. Okay, so you can either ignore, as I said, magnitude. You can look at the imaginary part of the coherence, and I will explain why. But this is all centered about the so-called cross-spectral coefficient and its normalized version, which we call coherence. Okay, so the important thing about the coherence that it depends not only on it's a complex number, right? And its magnitude depends on basically how much amplitude and how much the phase difference is stable for the two signals, right? But the balance between the real and imaginary part depends on the phase angle between the two oscillations. Okay, if you have small phase angle, then you have dominance of the real part, and the imaginary part is small. When you have angle close to the phase difference close to pi/2, then you have dominance of the imaginary part, and the real part goes down. Okay, this will be very important in the subsequent portion of the talk. So, but this is very basic, right? So I assume you're familiar with this. Okay, so this is one thing, and the other thing that we have to keep in mind before we start talking about the methods, that these networks that we are talking about, they are transient, right? So we are interested not only in measuring average tendency over time of population A talking to population B, but we are also interested in how these networks organize and, you know, kick in and start talking to each other during specific behavior, right? So like this is the cortex of the cat, with go, no go task. Basically based on the stimulus, she has to decide whether she will move her paw or she will keep her paw still, okay? And then we can see that during the go period, we can see that coherence between the visual cortex and the motor cortex grows because there is an information relay about the visual stimuli that gets transferred to the motor cortex in order to initiate the movement, okay? And then we see this green line where we observe this coherence, okay? The increased coherence, okay? And in no-go case, this doesn't happen, okay? So the networks are transient, this is important, right? So they have also time courses. The strength of coupling depends on the state of on behavior, okay? And which way it depends, it's very difficult to say because we all have closed loops. So behaviors determines the networks, and the network determines the behavior. So, but of course, they're related. Okay, now we considered the situation where we had access, like hypothetical situation where we have direct access to the activity of two specific populations, and how we can then simply compute the measures of the synchrony, and then, you know, make conclusion about they've been talking to each other or not, right? But usually when we experiment with humans, and when we want to collect a lot of data, and when we want to exercise freedom in collecting this kind of data and generating different experimental conditions, we are more interested, of course, in using some non-invasive tools, right? And because these networks, they actually, like if you look at the previous slide, they evolve over very short interval of time. This is milliseconds, right? 200, 300, 400 milliseconds. So this is like hundreds of milliseconds. This time scale is only accessible to the electrophysiological measurements, okay? It's not accessible to fMRI, not accessible to positron emission tomography that is sensitive to metabolism and very slow. And fMRI is sensitive to the blood oxygenation level, and it changes, and it depends on the blood blood flow, which is also changing slowly, and actually lags EEG and MEG by about 5 seconds. Okay, so so therefore we are interested in EEG and MEG kind of measurements, and these measurements are usually performed with an array of sensors, as I already mentioned. This EEG sensors are located on the scalp, and MEG sensors are located inside this dewar, inside this helium-containing dewar, because the sensors need to be in a superconductive state in order to be able to measure femtotesla level signals. Okay, so this is how the sensors look with the old system. Well, not all. This is like the commercially available system now, with 306 sensors total. 102 magnetometers, 102 gradiometers in 2 orthogonal directions. This is the older system, but also available now with 275 sensors, which are radial gradiometers, which are measuring the difference of the field right close to the head and 5 centimeters away from it. And this is the very modern sensors, which are very compact, which do not require liquid helium and coolant. They actually have to get heated by about 100 degrees, but they're insulated, thermal insulated, and can be placed right on the scalp. And they're based on the optical magnetometry principle. And we are actually, though the American ones, we have several of them. They cost about $10,000. We also now developing our own sensor with physicists from Iowa Institute. Yeah, but these guys are pretty cool because you can place them very close to the brain, basically, right? Much closer than you can put these guys, because the thickness of this wall is about 3 centimeters. Plus, everybody's head is different, and therefore you can never have like uniform zero distance between the inner wall of the dewar and the head, and therefore the distance to the center is about like 4 centimeters. Here you have 0.5 centimeter to the sensitive element, and therefore the field, because it decays like 1 over r squared, 1 over r cubed, depending. And we sort of pioneered so-called one-step approach, where we take the data, look at the sufficient statistics of the data, which is cross spectrum in this case, and then we are trying to invert the cross spectrum with our direct goal of finding the network. So when we are inverting the cross spectrum, when we solve an inverse problem, we are not looking for individual sources, but we are looking for the networks. Okay, so the network is our unknown. Well, of course, in this case, it's 2-node network only, but you can solve, of course, for all nodes, and then you can recover all networks, much more with more than 2 nodes. But you are looking for not for the time series of a single source, but you are looking for the 2-node network, and the time series is basically the extent to which this network is active. Okay, to which the elements of this network, the nodes of this network are coupled to each other. Okay, so. All right, so we usually, when we look at this data, this is how they look, and we have multiple channels here. This is activity and time. Usually, when we work with healthy subjects and study some cognitive processes, we present them some stimuli. We call them events here, and we present it many times, and then we average the activity. Okay, so when we talk about the network analysis, we average the outer product of the segments of data around each event, and then we get so-called covariance matrices, or if we do it in the frequency domain, we have coherence or cross-spectral matrices. Okay, but this average operation is important. But when we are averaging, we are retaining the temporal dimension as well, right? Because this temporal dimension is counted from the task onset moment, from the stimulus onset moment, when we present this picture, for instance, or some sound or some tactile stimulation or whatnot to the subject. Okay, so now in order to be able to do some inference here, we need to write the observation equation, basically. And the observation equation of the MEG and EEG data, and actually electrocardiographic data, if you do invasive stuff as well, can be described by this kind of linear model, where your data on the sensors at each time moment, t, is simply a linear combination of activity of these so-called vectors G sub I, which are called topography vectors, with coefficients that are proportional to activity of each of the source on the cortex, okay, on the cortical mantle, okay. So basically, this topography vector, it basically is telling you what kind of activity you will see on the sensors, in this case, if source 1 is active, okay, with unit amplitude, okay. Then the second column would tell you if what you will get if the source 2 is active. The last column will tell you what will happen if what you will see on the sensors if source index n is active, right? And you can basically look at this. You can then write this linear combination of these guys, because all sources are assumed to be active with different extent, in different time instances, but overall, this is this linear combination that you observe on the sensors. Very simple. Plus, you always usually have some measurement noise, which accounts for something you didn't account for in your forward matrix or in your paradigm, etc. Okay, so this is very simple. This is a very simple forward model, and the forward matrix, actually, these coefficients of G, right? They're actually available. So you can take the MRI of the subject, you can extract the cortex, the cortical mantle of the subject very easily. Well, now easily, with available free tools, that were developed over a range of 30 years or something. Then you can have this cortical mantle, and this cortical mantle is represented by this mesh. And each node of this mesh, you put this current dipole as your source, right? And then you model it with Maxwell equations of how this current dipole, what kind of magnetic field will it produce on each of the sensors. And this gives you the column of this forward matrix. Then you take another source and you do the same thing, okay? So matrix G is available, so it's no problem. And it's actually available with pretty high accuracy in MEG for sure, okay? So now let's consider a toy system, okay? So we are interested in the networks. We are not so much interested in sources, but we are interested in the networks, okay? Let's look what happens if you have, let's say, only two sensors. It can be either EEG or MEG. These matrices are called G1, G2 vectors will be different, but the conceptually will stay the same, okay? So let's say we have two sources, two dipoles, and we have two sensors, okay? Then our observation equation is this, right? So it's topography of the first sensor scaled with the activity of the first source, with activity of the first source, and topography of the second source scaled with activity of the second source plus noise, right? Then if we take the Fourier transform or time frequency transform, whatever, or short-term Fourier transform, it doesn't matter, it's all linear transforms, then you get this X capital depending on time and frequency, but the structure remains the same, the spatial structure remains the same, but the time series are replaced by time frequency images of your activity, right? And then for the noise, the same thing, right? And then let's look at the covariance matrix of time frequency coefficients, which in this case will be called coherence, right? Or not coherent, cross spectral matrix, right? Let's take the outer product of X and X transpose, or X at time and frequency by itself, and let's substitute this guy into here. If you expand this, we get this expression here, which is compact notation for mathematical expectation of S1 times S1 conjugate. This is simply the variance, right, of this coefficient, right, and this will be called C11, and this guy is the variance of the second coefficient. Basically tells you the intensity, the variance of the source, right, at this frequency, right? And then you have very interesting cross term, so that this is G1 G2 transpose plus C1 times C12 plus G2 G1 transpose to C21, okay? The coefficients C11, C22, C12, and C21 are the coefficients of the source space cross spectrum, and that's what you are interested in. Primarily, you are interested in C12 because then you will be able to say how much these two sources are coupled to each other, right? Because this is their cross spectral coefficient, and then if you normalize it by the estimates of their power, then you will be able to get this coherence, right? So now if you group this, and you, of course, this is complex, and this is complex, right? This is real, this is real. If you group real and imaginary parts, you'll get something like this, right? Because C12 is complex conjugate of C21 and vice versa. Then you can write this, okay? So this is very important. Okay, this is basically the main explanation of why you get complications when you're trying to study connectivity using non-invasive methods, basically where you do not have direct access to activity of each of the population, but rather have access to the mixture of these activities, right? The real part of the cross spectrum here is comprises basically the so-called spatial leakage term that depends only on the power of sources, and the true real part of coherence, you know, and you remember, right, if the sources are active with small phase lag between them, then this guy will be quite large, right? And this guy is communicated, let's say, to your cross-spectral matrix through this kind of rank 2 matrix, which is basically G1, G2 transpose, rank 1, plus G2, G1 transpose, another rank 1, right? Then this matrix will be carrying the information, this frame basically, right? Will be carrying the information about the presence of coupling between the two sources, okay? But it will be contaminated, this real part will be hugely contaminated by this volume conduction term, okay? And that's the problem, okay? So we have this mixture and we really do not have access to the to this term alone. We always have access to this real part of coherence mixed with volume conduction EG or spatial leakage in MEG term, okay? And therefore, many methods that have been developed, they decided to ignore the real part of the cross spectrum, okay? They just simply ignore it. They just say, well, we just don't use it, okay? Gideon Alter developed a plethora of methods that are basically taking into account on the imaginary part of the cross spectrum, saying that this imaginary part is spatial leakage free, and therefore you don't get spurious sources, okay? Well, that's also arguable, but at least on the zeros level of approximation, this is true, okay? So our goal was to basically get access to this. This just describes several measures that have been developed, but they all are based on the imaginary part of the cross spectrum. The problem is that when you're measuring trying to, let's say, find the networks, you simulate the networks and simulate them with different phases, and this phase differences on the x-axis, and you plot the ROC-AUC, you know, of the task detecting this network in the realistically simulated data. If you work with the imaginary part of the coherence, you can see that you get very poor performance when you are for the low phase lags, and this performance becomes much better when you're talking about the already pi/2 phase lags. If you take the real part, but real part after the processing that I will talk to you about next, then you basically have a reciprocal picture, right? Because real part is indeed not so much sensitive to pi/2 coupled networks, but much more sensitive to 0 phase lag coupled networks. But this is after you clean it from the volume conduction, because otherwise it's mixed with the volume conduction and it just doesn't work at all, therefore people don't use it, okay? And then if you combine the two, then you get more or less uniform performance in detecting networks with different phase lags, okay? Actually, it turned out that there is a lot of coupling that is observed with 0 phase, okay? Like two populations can be talking to each other in a reciprocal manner, okay? They can be talking one, like the red and greenish population. Remember, we have arrows in both directions. And in this case, it was shown by Michael Rosenblum. It was shown that they tend to have small phase lag, close to 0 phase lag, right? Then the networks may be driven by some third source, right? Two brain regions can be driven by some third source, which you simply can't see, for instance, right? Or it can be coupled to some cognitive phenomenon, to some external thing, right? And then you will have close to 0 phase coupling. Then you can also have so-called frequency tuning. If there are two populations which with slightly different properties, then when they start to tune the frequency to the common mode, then they appear to be involved into the zero phase coupled interaction. Therefore, basically, when we observe it invasively with invasive recordings, dominantly we see zero, close to zero phase like coupling, but non-invasively we can't see it. Okay, it's very difficult to see it. Okay, so let's see what we proposed. Okay, so now we've been in that two sources, two sensors paradigm. Now we just extended where we have L sense sources, right? And then we have the volume conduction or spatial leakage term now becomes this, right? So this real thing here is basically, you know, it's real without taking real, right? It's always real. So you have to remove it. Yeah, but anyway, so it doesn't hurt. Then we have the component that is carrying information about the real part of the source space cross spectrum, right? Of the extent to which I's and J's source are talking to each other, right? Which is, we don't know, but we want to learn from the data. And then we have this imaginary part as well, that is mediated by a very similar guy, but in fact orthogonal guy, right? These two guys are orthogonal if you think about them as vectors, right? And we will start thinking about them as vectors in the next slide. But this is slightly different frame, how it manifests itself in the sensor space cross spectrum, right? In the spectrum, cross spectrums we observe from the sensors, okay? So now let's vectorize them, okay? Let's just take this matrix, this cross spectrum matrix, and fix T and fix F, okay, for some fixed T and F values, and just, you know, stretch into a vector, okay? Just by rows or by columns, whatever, just say identically every time you do it, okay? So it's equivalent to just computing the Kronecker product of, you know, if you vectorize GI, GJ transpose, it's equivalent to computing the Kronecker product of GI, Kronecker, GJ, right? Because you just have scales of GJ for each value of GI, right? And then copies, right? So then do this, and you end up with simply linear equation, but this was linear before, but still, it's more convenient to look at it because those are now vectors and very similar to our time series observation model, you know, just activity of our sources, right? But now you have time series or time frequency series of the cross spectrum, which is a linear combination of, you can call them topographies, we call them two topographies because they depend on the two sources, right? On topographies of two sources, on some combination of these two topographies. And this is this linear combination of these guys mediated or scaled by the coupling strengths of the two populations. And this is for the real part, and this is for the imaginary part. And this guy. Well, if you have a linear mixture in our simple business, we usually do projections. Okay, we say, oh, well, in order to get rid of some component that you don't like, you just simply orthogonally project our data away from this component. Okay, so well, the same idea is here. You just take these guys, you collect them for, let me first say this. Okay, hold on. So again, this equation, you have, of course, imaginary part, which is orthogonal to the real part. You can see right here. But then you have spatial leakage component, which is not orthogonal at all by the real by this guy to this guy, and in fact they completely overlap, but they overlap only partly if you limit the rank of the subspace. I will tell you. Just hold on a second. So basically, you have this cross spectral vector CXX, and it's basically it's comprised of the real part. It comprises the real part and the imaginary part, okay? And they are orthogonal, right? But the real part itself is comprises the volume conduction term, spatial leakage or volume conduction term, and the true coupling that we are interested in, okay? So the idea is to apply projection to this guy and to remove this component from this vector, okay? But in order to do this efficiently, in order not to kill all the data, you need to somehow estimate this subspace, okay? The spatial leakage subspace. And you know that this is the subspace spanned by this QII, which are simply GI, GJ transpose vectorized. Okay, so that I will not go into this, but okay. So the idea, very simple. You have this cortex, right? You have dipoles in each node. You have topography in each node, right? You take this topography, you multiply by its transpose, you vectorize it, boom, you stack it into this matrix. Then you get the second one, the third one, and etc. You take all the sources on your cortex, all the locations, potential locations. And then you do SVD on this, because SVD will allow you to concentrate the variance in the column space into a smaller number of dimensions, right? And make an efficient projector, okay? Then you select certain rank, you take the left single vectors of this matrix F, and you create the orthogonal projector that you then apply to the cross-spectrum vector. And this guy applied to this vector will orthogonal projector dominant to these guys will kill them, will diminish their contribution to the cross-spectrum. Will not affect the imaginary part at all because the subspaces are orthogonal, and will slightly kill this guy, but only slightly. You will see that. Okay, so here again, so this is the original expression, and this is the projection operation applied. Here I plotted the norms of the topographies of this QII topographies. Here, two topographies with yellow one, okay, sorry. The QIJ minus QGI norm with red one, with red dots for all locations in the brain, okay? And for the real part, I plotted the blue one, okay, here, this guy, the norms, okay? So this is the norm, okay? So the idea is that if I apply the projection, and okay, X is simply the correlation coefficient between the GI and GJ, between the topographies, okay? Maybe simpler for you to look at the distance between the nodes, okay? The same picture, but ordered by the distance between the I's and J's location on the cortex, okay? So now, you apply the projection and you get this picture. You get volume conduction power decreased by a factor of 20, okay? And of course, you get unaffected imaginary part as expected, and you get affected only the two topographies of the networks which have nodes close to each other. But well, this is something you can't fool physics here, and of course you got to pay the price. But this is a relatively small number of networks, and if you are interested in imaging some distant networks with non-invasive methods, then this actually works quite well. Okay, so now how you select the rank? Well, you balance between between how much you banish these yellow guys and how much you keep these blue ones intact. Okay, so and you can plot these curves of attenuation coefficients. You can take the derivatives of them, and then you can find the optimal projection ranks. Okay, so now this is how the original cross spectrum looks, the magnitude of it. Okay, this is, you see the diagonal which is there, right? And this is how the projected cross spectrum. Of course, projected cross spectrum is no longer a norm. You can basically do regression-based modeling and find the coefficients of these topographies that comprise the kind of matrix. Okay, so your equation is this now: the projected cross spectrum, this orthogonal sign is about the projection. The projection cross spectrum is basically this summation, well, double summation, but who cares, of these guys. These are just vectors. And you can do any kind of imaging here, which we know a lot how to do in our neuroimaging thing, where we have a lot. But yes, this is still imposed, of course, but you usually expect relatively few networks, like not thousands of them, right? Therefore, you can impose some sparsity priors. You can do some simple scans, so simply looking at how much of these guys with specific I and J are present in the data. You can do subspace-based methods, whatever. You can do full-blown inverse And I will then skip this. So this is some simulations here. So we simulated 3 networks with their time range activity changes like this. There's strengths of couple, and the third network overlaps the two, right? And then and then we try to do it with different phase shifts. And if you do this method, then with small phase shift, the psicos works, of course. 2 other methods don't work apparently because phase shift pi/20 is small. Therefore, pi/2 psicos still works. These guys, some of them work, of course, because it's favorable condition for them. And then yes, we get this uniform yellow thin if you use psicos, and if you use imaginary part, as I already mentioned, you get this. You get real part, cleaned real part, you get this kind of performance. Okay, so this is another precision recall. I will not go into this. This is how this whole thing is sensitive to the forward modeling errors, actually sensitive, but not so badly. Okay, and then this is application to the real data where the person was looking at the hand, at the drawing of the hand, and this hand was rotated every time differently. And he or she had to decide whether or not this was a left hand or it was the left hand or right hand. Okay, so you need to do some mental rotations in your head. So you expect some communication between the visual area, right, like maybe ventral visual pathway, and the motor cortex, right? You expect some communication with the temporal pole, where you have all the 3D to 2D mapping happening to the motor cortex, this kind of things you expect. We ran the analysis. We looked at the networks in different subbands. We looked separately at the real and cleaned real and imaginary part. We assessed the reproducibility of these networks, okay? And then we show some of them here. There's a beta band. We have very nice coupling from the ventral visual path to the motor cortex. Then we also have coupling between the 2 motor cortices. Okay, or maybe this is sensory cortex and this is motor cortex, which is also very interesting. But because it's all related to the movement, we have time courses of this coupling. And in the gamma band, we also have very nice and interesting cross hemisphere coupling, because these fibers are very thick and it's very cheap to communicate between the functionally homologous brain regions much faster than within one hemisphere. Okay, so that shows very plausible results, and they applied it already to also to several other things. But because we're talking here primarily on methodological aspects, I will try to tell you a little bit further how we approach this. So again, we have this equation, and we are interested in inverting it, but now we are interested in finding some global solution, not locally scanning on the presence of specific QIJs in the data, right, in your cross spectrum, but interested in some finding global solution, right? And to do this, we formulate this is your measurement, the same, this is cross spectrum, this is this forward matrix, the different abbreviations, but different names, but variables, and this is the activities across spectrum of your sources, and that's the regularization term, which is the mixed norm, because this mixed norm allows you to do the sparse spatially and smooth temporally profiles. Okay, why sparse spatially? Well, because our spatial resolution is actually relatively poor, right? And we expect to see not very many networks, and they will be, they will be really sparse, right? But why in time we want something smooth? Well, because our temporal resolution is very nice, it's very high. Therefore, we have very smooth profiles with respect to our temporal resolution, right? So it's not that the network happened and died. Usually it happens, and gradually dies because we sample very densely in time, right? So then this mixed norm thing seems to work quite nicely. This is though the simulations, we are still in the process of finessing this. That's the networks that it finds. We still haven't played with the scenario where the nodes are closed, but for the original psicos we did play, and there are some Monte Carlo simulations, and we show that outperforms the 2-stage methods to two-step approaches. Okay, and then of course it's interesting to use a structural Cognitome to form the prior, and that will be this kind of thing where this describes how well our solution fits the data. I'm sorry, our solution fits the data. I'm sorry for every time different notation, but and that's the prior term, right? Which is L1 norm, but scaled by the presence of the anatomical connection. Here, we just started basically like several weeks ago, in fact, and we have some prior results which so far not very good. So we simulated two connections. Which was corresponding to weak anatomical connection and to the strong anatomical connection. And then when we do not have priors and solve the global inverse, we kind of recover both. And then when we have only this prior, we recover only the strong connection, which is expected, but we need to do something about it, okay? But this potentially very interesting thing, because you can apply in a very nice and principled Bayesian manner those priors, and you can also model the network's dynamics also with this prior. So it doesn't have to be just the numbers, it can be complex numbers which measure and also you can have priors on the delay of propagation depending on the thickness on the cortical fiber that is connecting two regions. There are many things that you can model here. Okay, this is a long summary which I'm not going to read, okay, because I want to tell you a little bit about epilepsy, and I'm kind of close to the time when I have to finish my talk. So when we talk about epilepsy, in epilepsy, we are looking at the patient data where we record so-called the activity, again, with multi-sensor arrays, and we look for so-called interictal spikes. Those are some very distinct events like bursts of activity, very sharp bursts of activity, which characterize so-called irritative zones, okay? There may be several irritative zones there, okay? And these irritative zones, one of them is actually epileptogenic, okay? Usually, not always, of course, but usually. One of them is epileptogenic, and it's primary zone, which you are interested in removing, and then the seizures will stop propagating, this abnormal activity will stop propagating, and the person will be seizure-free, okay? So first we look at the large-scale propagation dynamics, okay, analysis. So, well, after you have looked at these spikes, you found them in time, where in time, this channel by time matrix, you spotted the time instances where this interictal events happen, right? And then you solve the inverse problem, just simple inverse problem. You look for the source, and you can have usually you have several clusters, and these clusters, as I mentioned, are called irritative zones, okay? And now your question is, who is the primary one? Okay, is it the first one in here that it propagates to the second and then to the third again, or maybe this is the first one and then it propagates to the third, or maybe this is actually the second one and the activity propagates to the first and then to the third. Okay, so we propose to do it with some hidden Markov model because it allows you not only to measure this ping pong between different brain regions, right, between the spikes, because the process is quite complicated, but also to simulate the inaccuracy of your spike detection method, okay? Because nobody can detect spikes ideally, because doctors look at very high amplitude spikes. We look at both, but low and high, but we can make mistakes. Our algorithm can make mistakes. Every algorithm is characterized by certain specificity and sensitivity, every detection algorithm, okay? And so the idea is to say that we're measuring something here in the external world, which is covered from us by this observation probability vector, right? That if we are in cluster 0, then we observe that we detect that we're in cluster 0 with certain probability and we miss it with certain probability, and the same above cluster 1, right? And then we introduce this kind of architecture, this hidden Markov model that has, in this case, it's extendable to 3 and more nodes, of course, but this simple slide about the 2 nodes only. And these 2 nodes, the activity in the first node, the spike in the first node here, the network, this hidden Markov model propagates with probability 1 to so-called, switches to transitions to state 1, P1, so-called propagation state. And this propagation state may end up either in the idle state, nothing happens, no spike occurred in any other region, or it can trigger a spike in the second region or in the third region if we expand it, okay? So some transitions are 0 in this hidden Markov model. Some of them are set to 1. And then we take these events, we basically identify through hidden Markov model tools, so solve and identify the transition probability matrices here, which shows you the transition probability from event in the first stage, from the between E1 and E2, right? You can measure, you can look at this, right? And then you can derive some probabilities that event in cluster 1 propagated into cluster 2 within specific time interval, okay, within specific time window, right? And then you can look at these matrices, right? So this is very briefly, I'm showing how the taking into account sensitivity and specificity improves since, but I will not go into these details. But this is the result of analysis. Basically, for the 3-node network here, you see we show it this way, this matrix Q, but somehow it's raw here. Anyway, it shows you that in this case, you have reliable propagation from cluster 1 to cluster 3, okay? But this is how we agreed to depict our results, okay? And that's for some patient data. We have this patient who had surgery on the frontal lobe, but it was unsuccessful. And then he came into Pasadena Hospital when I was back and working there. And they mapped it. And then they looked at this method with automatic spike detection and then automatic propagation detection. We found that cluster 1 is the primary cluster and it's the temporal lobe, which appeared to be true because after he was resected in this region, he became seizure-free. Then this is how we look at the consistency. We record the patient in different days, and then we apply the same analysis to different days data, and then we look at the consistency, and then we see it, we believe that, yes, this is true. Then it's another patient, again, 3 records of this patient, 3 clusters. Again, we see here the propagation from cluster 3 to cluster 2 here, okay? This patient was not operated because he had this in the very critical sensory motor area and he can be removed. Okay, so basically hidden Markov model here served as a very simple counting tool, right? So that you just simply counted the events, but you were able to take into account the average sensitivity and specificity of the method and get quite reliable results, right? So that was the global propagation, right? The local distal propagations. But it turns out, and the goal of that analysis was actually to pinpoint the primary zone, right? It turned out, we recently discovered this, that if you look at the local properties of the activity within each rotative zones, and if you look at the local propagation patterns, very locally, like 5 millimeters around, maybe 1 centimeter, you can actually find also the signatures of biomarkers of the seizure onset zone. Okay, I will very briefly. So here the same picture, but now we look at the local spike dynamic, local empirical spike dynamics, local in space, I mean. Okay, so the traveling waves, they ubiquitous in the brain, they now very popular topic, and people have already explored whether this activity is a traveling wave or that activity is a traveling wave, and they find these traveling waves everywhere. And in particular, they also found that interictal spikes are actually traveling waves, and when actually what they observed, we didn't know when we started this project, we didn't know that, okay? But when they observed it invasively, they found that when they see traveling wave-like behavior and they remove this thing, this portion of the cortex from the patient head, brain, then the patient becomes seizure-free, okay? And if they don't see the wave and they remove it, the patient doesn't become seizure-free, okay? So that was invasive observation, which we repeated basically non-invasively with MEG. So how we did it very simply. So we have our observed data that we say, okay, we are interested in waves, right? In cortical propagation. So we say, okay, let's data around the spike here of the observed time series. Those are few time series are shown on the sensors, right? Let's say that these networks are, this activity is produced by the activity on the cortex that is propagating down these processes of this little spider, right, which is created on the cortex for each location. And we look at different speeds of propagation, and we for each source location here, we project this elementary directional wave onto the sensors, right? The signature of this guy on the sensors, and then try to create the linear combination of them to describe the data. And then when we identify this model, we are doing it with lasso, with L1 punished coefficient. So we get sparse solution. We want to have as few waves as possible. And we also restrict the sign because, well, you know, the different reasons for that. Okay. And also we scan over different speed values, and we scan over the time window where this wave would occur. Okay. So this is the scan. This is the starting time of this wave within the spike, right? This moment basically. And then this is the speed. And then we look at the R squared, basically where our model fits best. Okay. And if this model fits best, then fits good, then we say, well, yes, this event is behaving like a traveling wave over this time interval. Okay. Then we take patient data. Those are the intricate spikes. We take them, we localize them, and then each guy we analyze with this model, we feed the model. And then for each cluster, spatial cluster, we plot the goodness of fit histogram, the R squared histogram. And then we look at the percentage of spikes that were well described by the traveling wave model versus those that were not. Okay. And it turns out, by looking at nine patients so far, then these guys who have these brain regions who have a large proportion of spikes with that fit traveling wave model well, they actually appear to be epileptogenic based on the results of the surgery, and for one patient based on the independent neurological diagnosis. Okay, let's say take this patient, for instance. This patient is the same that I showed to you. We have this cluster 4, which have very distinct picture. And again, the number of spikes is not a predictor, but this thing is predictor. You know, the fact that the amount of spikes that behave like traveling waves, which is completely consistent with invasive measurements, but this is done completely non-invasively. This is another patient, okay, that we have a lot of guys here in the same region, more or less, but then we have these guys which are not at all behaving like traveling waves, but this guy is. And this portion was removed, and the guy is seizure free. Oh no, this one no, this patient too. This was not operated, but he was diagnosed with this region independently. The rest of the patients were operated. Okay, this patient again we have occipital cluster, and this is an occipital cluster that we see a large proportion of spikes which fit well the traveling wave model and not in the other clusters. Okay, and so far for 9 patients. Except for one patient where we found nothing for some reason. Well, this one, this patient. So we have very nice agreement between the resection or diagnosed location and the region where we observed a large proportion of spikes. Okay, which described by the traveling wave model. And actually, it now that this view has been emerging in the literature, and that the following meta and large-scale propagation dynamics of intellectual activity can be stated. Okay, so we have seizure onset zone. Okay, we have spikes that are generated by the cortical tissue in the vicinity of this seizure onset zone. Okay, these spikes, based on the recent paper by Smith et al. In eLife, these spikes tend to propagate towards seizure onset zone, okay, tend to behave like traveling waves with propagation direction toward the seizure onset zone, and they occur on both, let's say in the simplified world, on both sides, on the left and on the right, but more on the right because seizure tends to propagate when the seizure actually starts, right? It tends to propagate to the right in this case, right? And this is the ictal activity propagation direction, okay? Also, this seizure onset zone seems to trigger the spikes. These spikes trigger the spikes in distal zones, okay, like here. And those are irritative zones, irritative zone 1 and irritative zone 2. And these zones, they have everything okay in terms of the suppression of activity, you know, the GABAergic mechanisms and stuff like that. And therefore, they do not propagate. They die immediately, okay? They don't propagate. They don't show any propagation patterns. And that's why we do not see them. We do not see traveling waves in the irritative zones, but only close to the seizure onset zone, okay? And that's something that this view is emerging. I just summarized it. This view is emerging in the literature, but this literature is all based on the invasive recordings. And all these kind of propagation and this local propagation and distal propagation, they all can be seen non-invasively with the methods that I just described to you, okay? So therefore, I believe that we are close to defining some quite reliable biomarkers for seizure onset zone detection, which I believe very important for the field and for our patients, okay? So I will not do this conclusion again, so I just say that, the traveling wave model fitted to the intractable spikes seems to be able to identify the seizure onset zone by as regions in the brain that produce intractable activities that is described well by the traveling wave model. Sorry for the circle argument. Okay, so yeah, and that's it. I'd like to thank you, my team, Sasha Kuznetsova, who worked primarily on this traveling wave model, Nikita Fedosov helped her quite a bit over recent year. Daria Kleeva works on this connectivity analysis on healthy, on PSICOS thing, and Dima Altukhov, who started PSICOS thing and worked on this and then did this global inverse with mixed norm. Okay, and again, the funding here from Ideas Center to Dasha. Nikita is, although he's at HSE, but he's also funded from AIRI, Artificial Intelligence Research Institute, and me too. And Dmitry is also there. Okay, yeah, thank you very much for listening, and I'm open for the questions.
Okay, dear friends, let's thank our speaker. Yeah, Alexei, thank you very much for such a detailed and great speech. Dear friends, do you have any questions? Also, are there any questions in our YouTube translation?
Maybe in the chat, there's also maybe nothing in the chat here.
Nothing yet. Okay, Maxim, he was clapping. Okay. Well, seems like everyone is
everything is clear, absolutely.
And everyone is very deeply impressed.
Yeah, yeah. Yeah, okay, but well, anyway, it's very often that well, maybe it's a lot of material. It's really indeed a lot of material that has been covered during the talk, and yeah, it's sometimes very hard to make, and also this very how to say, the field is kind of narrow, very narrow field, and only primarily the person who is working in it can ask questions, but yeah. But anyway, my email, you can send an email, you can come.
Yeah, I'm sure we'll have lots of more viewers on YouTube, and after a short probably period of time, you'll have several questions on your email address.
Yeah,
I would say that many machine learners would enjoy your talk.
Actually, yeah, I can tell you that for machine learners, there has been this tendency, because, well, this recently, we just today, right, Facebook announced that they did MEG-based decoding of speech, or not speech, of something the person has been listening to, almost 3-second interval, and this is actually not a trick at all, I would tell you, really. I mean, no, it is cool that they did it, okay, and they did it in a kind of a smart way, okay, but invasively with electrodes located in the superior temporal gyrus here in this primary auditory cortex, you have a so-called gamma band envelope, which tracks very nicely the speech, very nicely. And it's a paper by Kubanik, I believe, John Kubanik in 2013, with David Popel and all these guys. Okay, so it's not a trick at all. And you can really see this gamma activity with MEG. Okay, yes. So yeah, it's an interesting paper, but it's not that, it's not that sexy when you look at the source activity. Sexiest things are if you look at networks, okay? Sure. And in order to be network-centered, you have to apply PSICOS projection to the cross-spectral matrix of your sensor data. You don't have to go into the source space. No, you just take the PSICOS projection, you apply it, and then you killed the volume conduction effects and power effect, but you emphasize the coupling thing. And then you can feed it to your network and do the training, and you can also do time result stuff. You know, that's just an immediate application of the machine learning to this. But also there are methods which are using machine learning for solving the inverse problem, just the classical inverse problem. There have been recently several papers appeared on this. It still doesn't work very well, I mean, I would say, but it started, and I think at some point we will have it working quite well, because we have this ambiguity, of course, when we're solving the inverse problem, because it's ill-posed by definition, at least physically ill-posed, and mathematically, if you don't have sensors so over the head, not only at some location or small portion of it. So yeah, so there is a lot of place to apply, but also we are kind of developing the interpretable AI for analyzing the EEG data, and I was speaking about it at several places already. And so the idea is that we find a solution that we can then look at the weights, and we can pinpoint the population of neurons, where they are located and which the dynamical properties they have. So I think that, you know, yeah, these guys from Facebook, if they interpreted the weights and showed what the weights, what the network has latched to, right? That will be very, very interesting if they could have done that. If they publish it in a journal, and they have archive paper, if they publish it in a neuroscience journal, they will be most likely asked to do that, and then we will take a look. Because that can be just eyes, actually, it can be eye activity, even whatever. Really, it's not that simple. But anyway, yeah, thank you.
I didn't know they were making a MEG apparatus at Fistech. I'm really glad to hear that.
Okay, so about Fistech is something new to me. I know that we are working on this with even Fistech is
Yofa. I mean,
Yofa, yes. Yeah, we are working there with Anton Vershovsky, and we've been working already for, let's say, 5 years on this, but we don't have specific funding for this, so we just do it based on leftovers. And we already now have our, non-zero field sensor working basically, and we have a magnetically shielded room that we purchased just before this thing started, but we don't have it installed yet because of the same thing. And so we are waiting when it's assembled at HSC, and we'll be able to do experiments with humans. But we also have access to the MEG center in Moscow, and we have the room there, but we want our own, so we have freedom of experimenting with it.
Yeah, sure.
Yeah, so
I'm glad to hear that there is one in St. Petersburg too. I used to work at HSE Institute.
Oh, really?
Back in the day. Yes.
I see. That's actually a pretty cool place. I mean, no, really. I mean, in terms of not so much in terms of how much they pay people, but in terms of the work they do, they're really pretty cool. No, seriously.
Yeah. I worked on fusion reactions.
Ah, I see. Okay, interesting. Yeah,
that's where Globus is.
I see. Yeah. So yeah, but technologically, yeah, we are facing problems technologically. Like these sensors that I mentioned to you, the sensor is this, right? Like love is chewing gum, and our sensor is like a box of the smartphone. Okay, and but still, it's because we don't have VCSEL lasers, the compact lasers. We have this laser, and we have optical fibers that feed into the sensor, the light, and yeah. So yeah. Okay.
Okay, dear friends, do you have any more questions? And if you would be kind enough, please could you share your thoughts on the quality of translation and your thoughts on what could be improved in YouTube chat, if you don't mind. And in general, thank you to everyone. I'm really glad we went through the first day of our conference. It was a great pleasure for me, and I'm sure for all our listeners to enjoy all your beautiful speeches. And we'll see you tomorrow in the next episode.
Okay.
Okay.
Thank you.
Okay. Thanks.
Thank you very much. Bye.
Так, ну что, разбегаемся потихонечку. Всем большое спасибо за внимание. Всем всего самого-самого хорошего.
Спасибо большое, было очень интересно.
О, я рад. Я очень рад, действительно. Да, и спасибо всем, кто на YouTube был все это время. Мне приятно, что в трансляции участвовали люди. Здорово. Всем.